Test the audio. Hello, everyone. Nice to meet everybody. I'm Luca, and I'm a PhD student in Joel Lamanno's lab at EPFL. And I'm here with my beautiful colleagues, Alessandro Valente, John Mayat, Nika Gorshek, Hannah Shede, Xinyi Huang, Jian Lin. And together with also Agostina and Laura and a few other folks, we are the organizers of the first single-cell and spatial metabolomics online day. Thank you so much for trusting us and being here with us today. We expect about 900 people throughout the day, and we're very excited to do this. So where does this come from? This comes from Rahul Sathija's single cell online day. He was the big inspiration for this event, since I think he's able to bring the community together in a very accessible way that also enables people from other communities to join. And this is indeed what's happening today, as I will give you some stats in a moment. So again, we are from the Gioele Lamanno and Giovanni D'Angelo's lab at EPFL, and we're working ourselves on spatial lipidomics and more. This event is supported not only by EPFL, but also by the sphingolipid biology community, by the Decoding Bio newsletter, by the Chan Zuckerberg Initiative, by the HuBMAP and the Human Cell Atlas, the last two, which will also deliver a talk today. You can see in screen share the schedule for the event. Now, Joel and Giovanni will deliver an introduction that will try to compress in a minimum amount of time the major achievements of the field and trends. And then we'll hear from specialists all of these facets of the field. First, Human Cell Atlas and HuBMAP, followed by super interesting talks with also a space for speed talks that were selected from a few applicants of you. So we do not have breaks, unfortunately, this year, and we look forward to all your advice for the event for next year. This is going to be an yearly event, and also we will open a Discord community for you to connect with each other after the event. For now, I just wanted to say there's a Q&A enabled on Zoom, so you can post your questions there, and the moderators in this room will collect them and ask 2 to 3 questions to each speaker. So yeah, our goal is to bring the community together and start making you interact. So the Discord is definitely there to stay, and we will only add people who really wanted to be part as we asked. So yeah, we have 900 registered users. It's very balanced. About 22% are PhDs, 21% PIs, 21% postdocs, and 21% researchers, plus some mix from industry startups and unspecified, which we hope to increase in the next few years. We are happy to have multiple communities coming together, with 40% of you saying they work in metabolomics, 41% in single cell, 17% in proteomics, a lucky 13% in machine learning, and 33% very curious other things. So we're very happy to have you all together here. Organization-wise, I want to say that our YouTube channel that set up, and we're already live streaming there. It's 25 seconds delayed, and I will now paste also the link in this channel here. The idea is that if for some sudden and unexpected reason the Zoom was to fail, you can always join the YouTube, but this should not be the case. You can feel free to also share the Zoom link with your colleagues and friends, and we really look forward to the event, and thank you so much for trusting us this first year. So I guess now the microphone will go to Joel and Giovanni, who will give a general introduction to the field. Thanks again, and enjoy the talks.
So welcome, everyone. Welcome to our single-cell and spatial metabolomics day. It's truly exciting to see so many of the community gathering online. This is our first event, so seeing so many people is really, really exciting for me. So 2024 comes to an end, and our field is coming of age. And working on in single cell and spatial metabolomics has definitely unique aspects to it. And we thought with the organizers that we really deserve a focus attention, and basically we're calling here with a dedicated forum that maybe we can have every year to get a bit updated and to get speakers and hear the latest. So this, as Luca said, is inspired from a model of the single cell genomics day that was initiated by Rahul Sathija, and similarly we're going to start with a kickstart overview. So Giovanni and I will walk you through some key developments in the field, and specifically want to highlight a selection of breakthrough in methods, technologies, and discovery. So, as I was saying, a larger community is emerging and using new methods. And one thing we have in common, I think, is tackling fundamental questions about cellular and tissue metabolism at an unprecedented resolution. We're really building bridges here across disciplines with questions that I think do not trivially fit a spatialomics meeting, the generalist genomics, or a forum that is specific to spectrometry. But here we want to ask questions I think are shared. What defines cellular metabolic states? How do metabolic states map to cell types? And does the context in the tissue constraints metabolic state? And whether we can have a system look at this and look at a complete metabolic atlases. I think this is something we are interested in the community, and what we have been seeing as major progress this year that we structure in this brief summary around 5 areas is that we really moved in different directions, from advancing single cell analysis by pushing basically the resolution limits to connecting metabolomics to other omics, proteomics, transcriptomics, by developing, for example, multimodal methods. And the field has really tackled challenges also in scalability with all organ atlasing approaches, extending focus on molecular precision with more advanced metabolite identification pipelines. And finally, I think we are also beginning to capture the dynamics of the system with tracing techniques, metabolic labeling, and much more. So let's dive into the first area of focus, super resolution. So MALDI-MSI obviously gives us an incredible molecular details, right? But it faces resolution limits. So while SIMS and other approaches that can achieve higher spatial resolution, it can't quite match MALDI's molecular coverage. So the typical resolution we are close to is approximate the 5 micro limit, and maybe a theoretical boundary is the one defined by the matrix crystal size. Nonetheless, this year we've been seeing exciting solution, maybe borrowed by the bio imaging literature ideas from microscopy that really reached our field with computational methods using deep learning to achieve super resolution, as well as tissue prep methods that have been using gel expansion ideas to and applying this to MALDI imaging. So the core of the contribution I'm going to quickly summarize here, effort from the GAO lab, Kai lab, S lab, and Koskun lab, that I think collectively basically pushing MALDI-MSI limits beyond what was possible before. So my 2 cents on what they did here. These 2 first paper, let's look first at the 2 key computational approaches to super resolution. Ozderk and colleagues developed first a guided method using a dual encoder architecture. Basically, they pair imaging mass cytometry with MSI data. And the magic happens computationally using a dual encoder that learns high-resolution features from the cytometry and basically applies them to the MSI, allowing this kind of resolution output. A similar approach, yet different in some important details, was also presented beautifully by Rapids and colleagues, where they use microscopy images, both bright field and specific stainings, MALDI-MSI and MALDI ablation marks in a weekly supervised framework where basically they apply a deep lab version 3 architecture that learns to propagate metabolite signals onto high-resolution microscopy data. So basically, the generator as an encoder, the decoder structure that generate an image per metabolite. And the key idea here is to use a probability map from ablation marks to reweight the local image generated. So then this is basically summed ablation spot by spot into field of views and to reconstruct MALDI images. The results that one gets look like this on the left. You see the pixelated lower resolution MALDI images, and this is the output, and this is a supervised example where we can see the ground truth. But typically for many molecules, like I'm showing in the right, they propose this kind of very cool reconstructions. And here I really want to stress actually is not simple linear combinations of what they see in microscopy, but they really have addressability of the single cell using the ablation probability maps, which I think was very original. So let's go on now a bit into physical approaches, in the wet lab. GAMSI, that was proposed by Chan and colleagues, achieves submicron resolution through a controlled tissue expansion. Basically, the approach makes it compatible with existing instruments, so you can basically use a tissue preparation workflow. And the process starts with a light fixation using PFA and glutaraldehyde. The authors actually comment on this being actually a fundamental step, and followed by an embedding in acryloyl X. And then light trypsinization and expansion, basically leading the tissue hydrogel composite to expand up to 3 or even 6 folds. What is cool also is that the authors propose a reversible molecular tethering system to further couple this with the cytometry and protein stainings, and they claim a 49% peaks retrieval cost compared to raw, MSI that have not been expanded. I think it was very cool. The images that they can get from setting with the instrument is set at 100 micro resolution. It's of course very much detail as compared to what you would get, vanilla putting the just spraying your sample. Another approach that is even more ambitious wants to achieve and manages to achieve tenfold expansion. It's proposed by Calab, et al., which develops XMSI, they call it, achieves a 500 nanometers resolution. The idea is to start with 15 micron tissues of quite thick tissue section. Proteins are then anchored to an hydrogel of acrylamide and vis. And this is digested and then expanded up to tenfold via basically several iterative, water washes. They do, I think, 4 cycles at 4 degrees. And the crucial trick here is also the tissue orientation. They flip the tissue face up for optimal laser access, and then they do a fine MALDI 1.5 Dan applied to with pneumatic electrospray. So this is the approach. Just I think the also data talk for themselves. Very cool subcellular resolution that they achieve here. And this would be what you would get normally from the same kind of sample without expansion. And I think as a neurobiologist, I really was excited to see, single neuron subcellular resolution. And, we can start to see with multi-MSI dendrites, which I think inside the tissue, this is a real tissue section. So this I thought was extremely exciting. So switching gears a bit, talking about whole organ atlasing is another core topic of innovation this year, I think, and centers on tools and methods for creating whole organ, whole system, whole organism, 3D or even 4D atlases over time. And the idea of building a comprehensive atlas is, we want to reveal, right, tissue heterogeneity, track dynamics over time, but also, enable meaningful comparison, for example, between individuals, analyze perturbation. So we need to compare typically in this kind of setups many samples. However, significant technical challenges, I think, persist. Both, matrix deposition and other technical variation can cause batch effects, especially during long acquisition campaigns, that are typically needed for 3D atlasing. There is, artifacts that are compounded from, even a few small artifacts and problems in pipelines compound one on top of each other, creating, for example, a not much feature space and other complication in the analysis. And eventually, at the end, ultimately, we also want to face speed of acquisition, which enables, of course, to acquire more data. So the two papers I wanted to highlight here, one is a bit self-referential from my lab and Giovanni's lab, and one from, the Zwiedler and Lam labs. Let's start with the first, where, we are aiming to propose an end-to-end platform for 3D and 4D mapping. We want to go from the sections through a system-level analysis all the way sort of to discoveries using this data. And the way, and we do it also by introducing a 4D atlas of zebrafish development to showcase these possibilities. So how does, the idea, what is the central idea here is to defer annotation of the peaks to the end, bring the peaks all the way to a combined final analysis. So basically this means doing key step, keeping this in mind, the image extraction, through peak calling, a feature space of homogenization, matching the feature so that they are the same across section, but most importantly, a more sophisticated way to normalize the data without creating artifactual signal and really also respecting the biological variation. Every peak potentially can be brought to the downstream multivariate analysis, where with this new tool, we have been adventuring in 3D and even 4D exploration in the development of the zebrafish embryo from 8 to 72 hours post-fertilization. We've been able to discover de novo key metabolic territories corresponding to prospective organs and looking at the evolution of the lipidome over time. Okay, now switching a paper, let's talk about another very exciting contribution from the Swidler and Lam labs. They really combined a lot of super interesting technology, fast acquisition MALDI imaging by shortening the transient, single cell MALDI MSI, a multimodal image registration technique for the 3D reconstruction, and a deep learning full resolution routine. And this allows them also to do large-scale atlasing of the rat brain, and they propose really a very exciting pipeline and a very cool proof of principle atlasing. I just care particularly to give you a bit of a description of what their tool called Meister does. Is, I think, a very elegant approach of deep compressive sampling of mass spectra with a 3-part deep learning system operating directly onto transients. The key idea and key insight is that the transient signals from mass spectrometry, while complex, they actually can be thought as lying in a simpler, low-dimensional manifold. And so therefore, first, they come in with an autoencoder with symmetric architecture, compress the high resolution transients into a compact 32-dimensional representation, and then a regression network learns to map this short transient directly into this low-dimensional space, so that then the decoder that they also learn in the autoencoder, they can then be used to basically extrapolate and predict really the full resolution data. So you can gain resolution in a spectrum and get amazing data in very short time. With this superpower, they can continue to study the lipid profile across 11 brain structure that they can stratify now in 3D using annotation, and they study lipid variations across different areas of the brain. Very cool. And even cooler, the possibility to then look at single cell data that they've coupled also in a particularly nice way to space using an approach of the convolution, where they can see the team cluster. And I think as a neurobiologist, I was very excited to see the extreme variation that they can see. I think that extend even beyond the team cluster that they annotate, which gives an hint to possible interesting distribution and more to discover in this axis. All right, so with this, I think I will switch here and pass the stage to Giovanni.
Yeah, so welcome everyone. This is Giovanni, and I'll take this over, from one part that also represents a problem, or if you want, an opportunity in the field of imaging metabolism, which is multimodality. So when we acquire our images, let's say metabolomics images, what we actually usually get is things that really resemble pretty much tissue architecture. Yet, it's sometimes, even in the easiest cases, it's sometimes difficult to relate what we do see with the identity of the cells or the tissue that are actually behind our metabolic imaging. So in order to get this kind of information, I think that what we have to aim for, and what actually people have been developing with more and more intensity, is modality of acquisition which will concatenate spatial metabolomics or single-cell metabolomics with other dimensions. So other dimensions, omics, let's say, techniques. And in this respect, a number of papers have been published during the last 2 years, which actually work in this direction from different labs, including the Lee, the Koshkun, the Liebeke, or the De Miranda, and the Lundberg labs. And I want to highlight some of these papers, though all of them have very important contribution, just to show what actually will bring on the table having multimodal acquisition power. And this is in this paper, for instance, by Nunes et al., what they have done has been to couple MALDI mass spectrometry imaging, time-of-flight mass spectrometry imaging, with imaging mass cytometry. The idea is that of acquiring samples first, like laying the matrix and acquiring samples by multi-imaging, and then washing out the matrix, and now apply antibodies that are meta-labeled for mass cytometry. And then registering together the images, actually you can get a sense of what are the cell types that actually are imaged by our metabolomics, spatial metabolomics techniques. So in this case, they managed to co-register lipid signature with cancer tissue and to actually describe immune phenotypes. And this is one image from that paper where they actually have combined imaging mass cytometry with multi-MSI mismatch repair deficient colorectal cancer, and where they can see that there are specific lipid signatures that are associated either with the immune stromal or cancer compartment. In a similar approach, in the lab of Ahmed Kushkun, they have actually developed a pipeline that, again, combines spatial proteomics and metabolomics. In this case, the spatial metabolomics approaches through SIMS, not through MALDI, and they actually have developed an entire pipeline which they call SpaceMet, so single-cell spatial metabolism that combines multiplex protein imaging and metabolite analysis. And in this case, they've been able to, given the high resolution of SIMS, they've been able to look into individual cells and tissue. Up to more than 30,000 cells can be at this point recognized per each tissue, where they actually again have protein markers and metabolic markers, and then they combine this information in order to extract information. This information is extracted through deep learning analysis that allows them to track trajectories and patterns in these tissues. And in this case, again, this was applied to lung cancer, so to cancer again, where they managed to confirm or to, let's say, find that glycolysis-related metabolites like lactate and adenosine are actually enriched or found more in what is the cancer tissue as compared to the stromal cells. So another chapter where we have seen, or where we need actually improvement in spatial metabolomics and single-cell metabolomics, is the interpretation of the spectra we obtain. So in many cases, we are dealing with MS1 spectra, so we have only one dimension to separate the different compounds. And so this restricts our capability to understand whether under a certain peak the molecules that we are seeing are those that we then call. So the calling becomes an urgent kind of need, and we need basically systematic approaches that at least allow us to prioritize more likely annotations over the ones that are theoretically possible. And in this respect, there have been a number of papers, some of them not necessarily related to annotation and some more dedicated to the issue, where either by computational methods that work out the most information that can be actually extracted from the data that we already have, and this allowed again to prioritize metabolites that are associated with specific peaks. Or in this case, I would like to highlight one important contribution towards democratization of technology that would allow a more than one dimension separation in single-cell mass spectrometry, that is this single-cell lipidomics through, let's say, microfluidic-based isolation of single cell, and then LCMS that actually has been published by the lab of Melanie Bailey. So the other papers that I'm going to talk about are all from the lab of Theodore Alexandrov. And, this contribution, span different kind of aspects. The first one is this, where they have actually considered the presence of more than 170 metabolites across 24 different MALDI-MSI procedures, and they have done this through by scanning a large number of acquisitions. And what they actually have extracted is the common features that they can actually apply to identify metabolites that then are reached in the different parts of the metabolome. So this is an interlaboratory survey and actually allows us to increase the number of observations that is useful actually to start teaching or actually start training models towards the recognition of specific molecules. Along similar lines, another massive kind of effort from the same lab in profiling a large number of single cell comes from this high-throughput SpaceM. SpaceM was a previous invention, let's say, from the Alexandrov lab. Here, the innovation relate to the fact that this has been taken now to real high throughput with the possibility to analyze metabolites in more than 100,000 single cells. And this actually reaches for the observation and puts the the capability of, in this case, is again multi-imaging to compare with bulk LC-MS in terms of coverage of small metabolites in individual cells. But all this leads me to probably what is the most relevant computational contribution from the group in terms of metabolite annotation, that is again a development of a previous pipeline from the lab, which was Metaspace, and in this case is a machine learning Metaspace, whereby the authors have trained the model to recognize likely from unlikely annotation based on massive data sets. And I don't want to spend too much on this because this is going to be one of the topics or one of the talks that we will hear about today. So as I told, on a different side, I mean, on a more wet side, there have been in the years different efforts in order to take single cell metabolomics to a level where one can use more than one single dimension to separate the compounds that are observed in single cells. One remarkable one is this paper where they, through microfluidic sampling, they've been able to isolate individual cells that then can be analyzed in rather standard machine setting in order to obtain single cell lipid analysis that at this point contains both retention times and MS1, and this actually increases the chances of getting a right annotation over spurious ones. Now, the last chapter I would like to talk about, which I think is very interesting for the development of the field, if you want, is the dynamical perspective. So the fact that we are at a stage now where we can try, or we can really appreciate metabolic fluxes, because metabolism in all is flux. So we might actually extract from analyzing metabolic fluxes in single cells or in space things that go well beyond what actually static metabolite levels can tell us. And so this has been matter of a couple of publications in the last year. So these are the two that we just selected as an example of what can be done with this kind of approaches. I'm going to focus again on the one from the Alexandrov lab. And here the idea is very interesting. So what they did was to feed cells with isotope labeled glucose, and actually what this glucose, which is labeled with carbon-13, would do is to go through the glycolysis and TCA cycle, and at some point the carbon atoms that are decomposed through these pathways will be used by the cells to assemble fatty acids that then will end up into lipids. Now, what they have done is to basically take a wide range in the M over Z spectrum and to fragment all the ions in that range to extract information about the isotopologue distribution of carbons into their fatty acids that they can analyze. And by doing so, they actually can estimate the metabolic flux of glucose into the de novo fatty acid synthesis as compared, for instance, to fatty acid uptake of the cells, to distinguish between cells that are grown, for instance, under normoxia or hypoxia, where the glucose usage, of course, is different, and then to assess the impact, for instance, of perturbation. In their case was silencing of ACEL on the usage of glucose for the assembly of fatty acids. The interesting thing about this technology is that these are scalable and fully transferable to all animals and all organs. So in the same paper, what they did was to feed actually mice with this isotope labeled glucose, and these mice were actually bearing glioblastoma in their brains, and what they were able to evidence with this technology is that even in an in vivo setting, they see that actually the glucose, the carbon coming from glucose, is preferentially used for some fatty acids by the cancer cells as opposed to the other cells that are found in the brain. So this is to say that we are at the stage now where we can start asking new questions in our, in our what wants to be our field. And what we do expect to come in 2025 are efforts, for instance, to try to understand better what these different metabolic configurations that probably we all see while we do our experiments, how they relate, for instance, to physiology. And to do this, we need to build atlases of single cells and of organs and of organisms. And so we do expect this effort to give some fruits in the next future. So this, of course, then begs the question of what is the relationship between metabolic configuration and cell type or cell state of the cells that we are analyzing. And again here, multimodality, which have been already looked into, might give us even more information as compared with what we have at the moment. And this is the basis of trying to understand the physiological importance, what we have named lipotypes in the domain of lipids, but that can be expanded as a concept to metabotypes, whereby we want to understand, I think we need to understand what specific metabolic configuration mean biologically and how they relate to gene regulatory networks, for instance, that are at the basis of the control of metabolic programs. So to obtain these things, we cannot do without an improvement in our tools and packages for the computational analysis of this data that we all are dealing with, which are, of course, very rich and complicated. And so I think that the discovery and the development of ad hoc tools for spatial and single-cell metabolomics remains a must also in the years to come. So with this, I want to finish, but not before thanking the people that really are those to be thanked for this event. And these are PhD postdoc students from the two labs, plus some alumni like Laura that is no longer in the lab, but still active in the field. And so I think that these people really deserve acknowledgement, and I think that their effort is going to survive the year. And so we will have this also in the years to come. And with this, I think we can finish and kick this party started, basically. So thank you.
Thanks, Joel and Giovanni. That was a nice introduction for everyone. And since we now have 10 minutes before the first talk by the Human Cell Atlas, I can see a question in the Q&A, maybe now two, and I wanted to ask you. First, we were asked if, given the fact that we heard from your talk different metabolism, glucose pathway, pentose phosphate pathway, lipid metabolism, and so on, what do you think is the need or possibility to bridge these different metabolic axes and how we can get a complete picture in the future?
Well, I'm not sure I understand this one, but I think that we just need to, for instance, in the metabolic labeling, we just need to get creative. I mean, and probably look at what people have developed in all organisms already and in bulk metabolomics to get inspiration and try to understand, how these things get actually declined at a higher granularity. So I think that the different axes need to be brought together. Space and single cell is what actually is missing in the picture, or is just started to be looked into the picture, but then it's all about how creative we get, I think.
Great. The second question, and by the way, I've seen some hands raised. Just to remind you, you can't directly ask questions. Please write in the Q&A, and we will ask the questions to our speakers. So the second question is, how does the transcriptome regulate the metabolome and vice versa, or at least what are the tools that we can use to start exploring this?
Well, I think that again, maybe you want to go for this one.
Yeah, I guess so. Well, I think methodologically, finally, starting to be possible to get close to really measure both. I think this is something definitely very exciting. I think one of the most challenging things is sort of to overcome. I think sort of the egg and the chicken problem. So which of the, in the regulation that definitely exists between, gene expression control and maybe also post, translational regulation that brings in, the regulation of the level of lipids, membrane lipids. We of course, what is difficult and challenging is to understand directionality, which come first, for example, in development, is it changes in the lipidome that trigger or just modulate the transcriptome and vice versa. And I think this, I think, involves it definitely a multidisciplinary effort, also in the sense that it most likely is not enough to just read the transcriptome and the lipidome and read them together, but also then go in with the tools of cell biology and dissect the mechanism that regulate these processes. And I think it's a very exciting avenue.
Nice. I might take the time to ask a couple more questions. By the way, I see in the chat that people are asking for the Discord. You'll be added throughout the event. Just for the moment, we were asked first if metabolites are specific for each cell types, at least from what we know now, when analyzed using HPLC MS2. And I'm not sure exactly what's the question, but do they have a specific chemical signature? That's the first one. And second, what are the major challenges that you have directly encountered so far in atlasing with MSI data?
Well, I mean, I can start from the second. I mean, the major challenge that we have been facing stays in the batch effect among acquisitions. So it's not necessarily, let's say, technical is not necessarily possible, let's say, to at least assume that all the acquisition we make with the technology that we have now stay in the same comparable space. So there is a big need for systematic regularization of the data that we acquire, which is not actually something very trivial to do. And I think that, developing algorithms that allow us to bring all the features that we measured, being these metabolites, lipids, whatever, and with multimodal analysis, let's say even more, to the same feature space. This is actually what is really required in order to make, atlasing possible. And then the other thing is, if we go multimodal, we need to go multimodal in a way that allows us to correlate the different modalities, or we have to align our acquisition to some kind of pre-existing atlas or pre-existing system that allows us to, understand exactly what kind of cell types are under our acquisition. So for the first one, I also am not sure about what the question was exactly about, whether metabolites are specific to cell types. I do think that, the many of the talks that we will hear about today speak in favor of a specificity, or let's say of metabolism associated with cells of the same type as opposed to cells of other types. Now, whether the same metabolism can be present in a subset of cells from different types, or whether you have multiple metabolic declination of the same cell type, these are things that we are actively looking into, and I think these are great avenues and great things to talk about for future development.
Maybe we have time for one last question before John Randall from Human Cell Atlas takes over. And people are curious to know more about how to study dynamics of metabolism in single cells, or at least what the field is driving towards.
I mean, this is super good questions and a question, and I think we have been, working and thinking about this with Giovanni. I think this is definitely the challenge going forward, to the next year. We've been seeing, I think, as Giovanni well introduced in the last block of the overview, methods that rely on trying to use metabolic labeling for that. And I think definitely this is key to disambiguate an otherwise, static picture that we are getting of the data. And I think, it's a similar challenge that, single cell genomics and transcriptomics field found itself, in a few years ago. And I think this has been overcome with similar tools. And I think obviously in metabolism, depend also considering, the kinetics and, the speed of those processes and, the more direct, enzymatic mediator of these processes, it might be even more challenging to technically, undertake. But I think it's going to be possible. We're going to see this, I think, in the coming years, definitely.
Thank you both for answering. And it's my great pleasure to introduce the first speaker, John Randall from Human Cell Atlas. John, the floor is yours.
Thank you very much. I'm John Randall. I'm from the Executive Office of Human Cell Atlas, and it's really a pleasure to be here. We're really pleased at HCA to be supporting and participating in this event, and I really appreciate and look forward to all the talks. I'm going to give a very brief overview or update on the status of HCA, where we're at right now, and also a preview of some of our future plans, particularly in spatial omics. I'll begin by overviewing HCA's mission, which is to create reference maps of human cells as the basis for understanding health and diagnosing and treating disease. With 37 trillion cells in the body, this is obviously a very large challenge, and so one that cannot be done by certainly one laboratory or even a small collection of laboratories. So this is really a global initiative. This map shows the number of institutions at each continent where there are scientists participating in the HCA program. It's a very large consortium. As of yesterday, we actually have an even 3,800 members at nearly 2,000 institutes in 103 countries. And so you get a sense of the scale of this project. And really, we're actually, even for the very first edition of Human Cell Atlas, we're drawing on hundreds of data sets from around the world to build a map of the human body. Very quickly, I just want to give you a brief overview of how we're organized. We are led by a 36-person organizing committee. This is a group of scientists from around the world that really set the scientific direction for HCA. They've established 5 standing working groups on analysis, ethics, equity, standards technology, and our data ecosystem. The heart of our scientific work is really our 18 biological networks. Each one of those studies a particular tissue or organ or system. But we also have regional networks on every continent, and those are really instrumental in ensuring that we have global representation, both in the participating scientists as well as the data itself, and that we're actually building a representative atlas and not just focusing, for example, on European ancestry. And all of this is supported by the executive office that I lead, which is 12 professional staff based in the United States, the UK, Singapore, and Japan. So very much a global initiative and quite large scale. We were really launched in 2017. The first few years, we focused on data generation as well as sample pipelines, analysis pipelines, and just generally building up our community. Beginning about 2 years ago, we entered a really intensive phase of data integration where each of our 18 biological networks are asked to create one or more atlases, each of which really draws on anywhere from 1 dozen to several dozen different data sets that have to be integrated, which is a huge challenge. We actually just heard a really nice overview of some of the challenges and considerations for atlasing generally. I will say most of our work so far has really drawn on single-cell transcriptomics, single-cell RNA sequencing with some ATAC-seq. And ultimately, just in this first phase of HCA, we will release V1 by the end of 2016. And I'll give more of an update about where we're at specifically in a bit. This slide shows our 18 biological networks and the number of cells that they have analyzed. Together, they profiled about 100 million cells, and actually an additional 17 million plus have been analyzed by spatial omics approaches. And so this is that latter number is certainly going to grow considerably in the couple of years ahead. Much of this work has produced, of course, publications. Our community has produced over 450 publications to date. 46 of those were published beginning last month across 8 different Nature portfolio journals. And so I really want to highlight this collection. You can find information on the Nature website about the papers in the collection. They really were categorized under Atlas integration approaches, human development, particularly prenatal development, analytical methods, and then a substantial body of work from our ethics working group and our equity working group as well. So I really encourage you to check that out. Additional papers will come out this week in Nature Medicine as well as other Nature journals. Now, the Nature collection actually, for the most part, doesn't actually describe official HCA atlases. So to date, we've completed 4 initial atlases from our eye, lung, and organoids networks, as well as our nervous system network, and that really focuses on 2 initial atlases from the BRAIN initiative, the BRAIN Initiative Cell Census Network and Cell Atlasing Network. We have partnered with them to adopt those atlases as HCA atlases as well and make sure they're harmonized with the other atlases that we're creating. Early next year, we anticipate 5 more atlases coming out, those being breast, gut, liver, kidney, and immune, and then later in 2025 and moving into 2026, then the remaining atlases will be completed. And at that point, we'll actually be at the end of the first phase, and collectively, these will constitute the first draft of the Human Cell Atlas. And all information on the atlases themselves, as well as what we call the source data sets, the 400+ data sets that are going into the first draft, are available on the HCA data portal at data.humancellatlas.org. As I've said, this is mostly spatial transcriptomics data to date. The challenges lying ahead are pretty significant for us. So beyond 2016, we'll be focusing on expanding genetic diversity and other forms of diversity in the atlas. We'll be focusing on new analysis portals, and also critically, we'll be moving to incorporate spatial data into the atlases that were created. I do want to highlight one challenge, though, that was mentioned by previous speakers, which is annotation. And here we're specifically talking about cell type annotation. We spent a considerable amount of time and effort building up a cell annotation platform, which allows scientists anywhere in the world, so hundreds of scientists, can then contribute to the annotation of the integrated atlases that are being created through HCA. And this really resolves a lot of the disagreements, the differences in the resolution of annotations across different atlases and different data sets, and is really, I think, the key to sort of getting consensus around cell type annotation for all of the atlases that we are creating. I've mentioned, of course, that one of our key priorities for the future is to incorporate spatial data. I don't need to tell this group about the value and the potential for spatial omics in atlas efforts. But what I do want to highlight is that early next year, we are launching a new major spatial program within HCA. And I really want to emphasize this is not just about doing spatial omics well, but it's really about spatial omics in the context of atlases. How do we actually think about cross-modality integration and analysis and so forth? And so there are really 3 tracks that we are going to incorporate. One is on not just optimizing technology platforms, but also matching different spatial technologies to the different needs of the biological networks, both for atlas creation, but also for querying and downstream analysis, analytical methods development, of course, and then the use of spatial data and the merging of spatial data with single cell atlases. Those are being led by different working groups within HCA. That being said, there's a lot of work that's already going on within HCA, and this is just a representation of some of the spatial atlas work that is published as part of the Nature bundle that I just described. So I will end there, but I just want to encourage people that if you have any questions about HCA, really the best entry point for those questions is to contact people in the HCA Executive Office. You can find our emails by clicking on our bios on the staff page on the HCA website. And of course, you can certainly contact me at any point if you'd like more information on anything that I've discussed. So thank you very much, and I'll stop there.
So we have the first question. Can you somehow infer single-cell composition representation of specific tissues from people's genomes?
We actually do have a lot of work. That's a really keen area of interest right now. Both particularly sort of in sort of assessing ancestry data and so forth. There's active work going on right now. I actually just saw a study using 150 different FASTQs from the HCA database. So I think that this work is really ongoing, and I think it's a really exciting area for the future. So stay tuned on that one, but it's definitely an area of active interest.
Thank you. And there is also another in the Q&A is where can we access the cell annotation tool?
I think I can put the link in the chat. The URL is celltype.info. And I certainly, there should be contact information there if you have any. I'm not actually sure that people can see that. Maybe I should just answer the question in the Q&A. I think that's probably the best way to do that. I'll put the link in the Q&A, but certainly I encourage you to explore it and to contact the cell annotation platform team if you have questions or if you'd like to use it. It's a great tool.
Okay, thank you. Maybe we can also put this into a Discord channel. And maybe the last question is what are the values of integrating all these data modalities and how you are addressing these challenges?
Well, those are fantastic questions, and it's a huge challenge. I think, the previous question with the previous talks about linking transcriptomics to metabolomics, I mean, those are the types of questions that I think we'd like to be able to ultimately answer. Certainly, when you talk about spatial omics specifically, we start, there's a lot of interest in cell programs, in cellular neighborhoods and cell-cell interactions. And that's, I think when we start to be able to build that kind of tissue map that incorporates not just the pan-transcriptome information, but also the spatial data about the organization of tissues, it becomes very powerful for studying some of those higher order questions. So a huge number of potential questions that could be asked in that regard, but it's really, I think, a critical aspect that all of us need to pay a lot of attention to.
So maybe we can close now and move to our next speaker. So our next speaker is Chen-Chen Zhu from HuBMAP. So Chen-Chen, the floor is yours.
Thank you. Let me turn on the... How do I do the presenter mode? Okay. How do I share my screen here? Can you hear me?
Yes, we can hear
share.
you.
Yes, so sorry, yeah. It's a bit early here in California, 6 a.m., 7 a.m. So I'm a little bit slow. Good. Yeah, so yeah, thanks for the invitation. So I'm presenting on our HuBMAP effort to map the human intestine with spatial transcriptomics. So I'm a research scientist at Mike Snyder's lab here at Stanford. So today I'm going to talk to you about a little bit of HuBMAP. So HuBMAP is a huge effort from NIH here in the States. So HuBMAP stands for Human Biomolecule Atlas Program. So it's a consortium consisting of multiple groups, mainly here in the States, but also some groups from Switzerland, Netherlands, and the United Kingdom. So the goal of the whole effort is really to develop and apply tools to create a very open and global atlas for the human body at the single cell, at the cellular level. And there was a publication bundle released last year, and there are more papers coming out for the second phase of HuBMAP this year and also in the next coming years. And the goal is really to use single cell and also different omics technologies to map the complexity. So here at Stanford, so we are one of the tissue mapping centers. So different groups are responsible for different tissues. So our group here in Stanford is really responsible for the human intestine. So this consists of the small bowel and also the colon. So we are mapping the different sites along the intestinal tract, starting from the duodenum and jejunum ileum all the way down to the colon, as you can see here in this graph. And our mission is to map the complexity of the whole intestinal tract. And we are using an array of omics assays, starting from the bulk assays such as RNA-seq, attack-seq, and metabolomics, lipidomics, and proteomics. But in the very recent years, we've entered an era with we've entered the era with different single cell assays. We have started with RNA-seq and the single cell attack-seq, and we've done also codex, and now we are doing spatial transcriptomics, which is what I'm talking to you about today. And the reason we are doing single cell is obviously, as I guess this audience knows pretty well, that with the bulk, you are really getting a very mixed picture of the whole tissue. And with a single cell, you are really starting and separating things. And now we are doing spatial transcriptomics at the single cell level. So you get a much clearer picture of the tissue architecture and also the interaction of the cells. And we started doing this using Codex assays. So Codex is an assay developed here at Stanford. So it's really for the spatial proteomics, which was just got selected as the method of year by Nature method. So this is spatial proteomics with using antibody staining. Essentially, we are targeting about 60 antibodies. At a time. So what we did here with this publication is really to map the complexity of the human intestine across the different sites. So you can see the small intestine and the large intestine here, and we could show the complexity at different levels. So starting from the single cell, and we can also look at the neighborhoods and how the different neighborhoods they form the communities, and how the communities they become the tissue units, and we can see how this tissue organization is happening at a very high level here. So with this, we are moving towards spatial transcriptomics. So essentially, we are moving from protein to RNA. The reason for this transition is really the RNA assay allows us to do a much higher number of targets. So hopefully, with an increased number of targets, we can do a higher resolution mapping for the cell types and also including very subtle states. And also, with RNA, because we only need to design the oligos, right? We have to choose the gene of interest. And so the target choice is much flexible than protein targets, at least with the Codex assay, because we don't have to validate the antibodies. And also with RNA, we can look at gene regulation. And then we are doing spatial transcriptomics doesn't mean that we don't do the spatial proteomics. We still do both. So we can do a multi-omic integration from the same tissue. And the way that we try to establish assays really try to compare the different spatial assays that we have. So this is a graph showing the different spatial assays at the time we started the project about 2 years ago. And so for us, the main criteria is really there are two, and we want to very high spatial resolution. So we only want assays with true single cell resolution. So we don't want any assay, for example, the Visium assay would be a lower spatial resolution. This doesn't allow us to do single cell. And the other criteria is really high number of targets. So we want to target at least hundreds of targets instead of like 10 or 12, which is enabled by RNAscope assay, which is quite popular. So here's a list of assays that we have benchmarked using our using our tissue with small intestine. So there are there are two camps. One the sequencing based. So you can see the stereo-seq at the left, and all the rest are the imaging-based spatial transcriptomics assays. All these assays they do single cell resolution. So the clear difference is really the single. First is the number of targets they do. So most of the assays they can do at least 100, and so for the Xenium they can do 480. Now there are 5K panel out there, but Cosmics can do 1,000. And with the sequencing assays, obviously you can do unlimited targets. So this is really targeting the whole transcriptome. And I'm not going through the whole details because due to limitation with the time. But I'm going to talk to two spatial assays that we did. One is the stereo-seq. The principle is sequencing, but the idea is that you have the DNA nanoballs. So basically, you transfer the sequence and the mRNA in situ onto the DNA array. And because the DNA nanoballs are so small, which is only 220 nanometers in size, so this allows you to do single cell resolution. So this is the major advantage over some of the other sequencing-based spatial assay. So this is the data we collected for our small intestine tissue. So this is the duodenum. So we did on a 1-by-1 chip. So we sequenced 4 billion reads, and totally we altogether we profiled 180,000 cells, and it's about 330,000 genes that we can profile with 60 million transcripts. It's pretty comprehensive, and we can see all the major cell types here with the tissue and with all the tissue organization. And the other assay I'm talking about, and we also liked, is the Xenium assay that among the others. So this is the same tissue. obviously the differences here, this is imaging based, and so we have much higher spatial resolution. Then you can see the whole tissue here, and with an epithelial layer, and you can still zoom in, and you can see the cell segmentation. So this is nuclear-based cell segmentation with a little bit of overexpansion of. Well, you clearly see the tissue structure just by looking at the DAPI staining here, and but we also have 377 markers, and we can use these markers to map the major cell types, and we did the same for our duodenum sample. This is essentially the same tissue section that I just showed you with the stereo-seq, and the tissue, the cell type annotation comes out very similar, but the spatial resolution is a little bit high because it's imaging based. And we are using this to map the complexity along our intestine tract for the samples that we have. You see the duodenum, ileum, and the sigmoid. So sigmoid is the large intestine. You clearly see the difference, for example, in the thickness of the muscle layer. The large intestine has a much thicker muscle because the bowel movement is stronger, and also the epithelial layer is thicker in the small intestine because most of the nutrients happen, and the absorption of the nutrients happening in the small intestine. And we do have a very thick layer of stem cells here in the duodenum and also in the ileum because the intestine tract is renewing itself within 2 or 3 days. And so that's why we need these stem cell. So our spatial transcriptomics assay clearly captured the biology and the tissue structure. And one question we had with these spatial assays is how reproducible they are. So we've done replicates for these assays, and we could clearly show that at least for these 2 assays, the replication is the reproducibility is really high. So we are reaching a very good reproducibility with 2 independent runs using the same technologies. And I'm not going into the details, but we also look at other performance of these spatial assays. So for example, the sensitivity and the specificity and how many transcripts we can profile per cell. This is summarized in this slide. And so this is suffice to say, we are okay with all the assays that we tested so far. But for our tissue mapping center, our method of choice at the very high level is really we chose the stereo-seq for discovery because it allows us to do untargeted mapping, and the Xenium for data production, and because the throughput is much higher in terms of samples we can profile per week. And these 2 assays are both work with fresh frozen samples and FFPE. So we could do whole tissue section that we showed you, and we look at the sensitivity and the reproducibility. With that, I will conclude and thank the team for their discussion and all the support. Good, thank you.
Thank you, Chen. Unfortunately, we won't have any time for questions, but if you have some, feel free to write them in the questions and answers panel. Our next speaker will be Peter Niemes from the University of Maryland. Peter, please take it away.
So if everything works well, you should be seeing me and also hearing me. And I'm just about to share the screen. All right. Could I get confirmation that this shows well?
Yes, it's perfect. Thank you.
Okay, excellent. Thank you so much. So thanks to everyone joining today. Many thanks for the organizers for bringing us together about this very tough challenge of spatial temporal single-cell omics, especially in the context of multiple omics areas. So I'm Dr. Peter Niemes from the University of Maryland. I've been doing single-cell metabolomics and proteomics for some time, and I hope to tell you a little bit of a different kind of technology and challenges that my lab has come across that we have not yet seen so far in the really nice introductory presentations we've seen otherwise so far. In case you're interested, in case you're a postdoc candidate, we have postdoc positions and also always are looking forward to PhD grad students. So please consider us. So essentially, my lab works in this fundamental question of how a single cell divides to give rise to normal cell heterogeneity to form different types of tissues. And as you can appreciate, this is a temporal and a spatial problem. So we use embryos. If you take a look at these embryos, right here, you see how the 1-cell embryo quickly divides into 2 cells, then those into 4 cells, then those 4 into 8 cells. These divisions take place every 15 to 30 minutes, so they're very fast. And on top of it, these single cells literally start mapping out the three-dimensional structure of the embryo developing. So you're no longer in a two-dimensional tissue area. You're literally in a three-dimensional organism where now you have to conduct single cell analysis. It is a tough challenge, analytically speaking, but that's what we picked when we started single cell omics more than a decade ago. And so that is not the end of development that we're interested in, because we're also interested in how these particular cells give rise to normal cell heterogeneity in the brain, specifically focusing on the neurons in a mouse brain. So these are the two different models that I will be focusing today on. Now, we know that the sequencing has been kind of given the upper hand because of molecular amplification when it comes to single cell sequencing. There have been tremendously nice developments over the years, as we have also heard from Chen just now. And so today, what I'd like to kind of shift the gear to is back to proteins and metabolites, at least how we bring this in and why it's important. So you will see that if you focus on stem cells and you really understand molecular determinants or morphogens, then you now theoretically have the option of creating cells or tissues on demand. Do you want an epidermis? Do you want neurons? Do you want hard tissues, right? Essentially, theoretically, what it really takes us is to identify these players. And we know a couple through decades of very hard and elaborate biology investigation. What if we could speed up this kind of discovery process by single cell mass spectrometry? And so my lab kind of got into this topic by using embryos, the Xenopus laevis, the frog, the mouse, and the zebrafish. We build the mass spectrometry platforms to identify, quantify metabolites all the way to proteins in single cells. And then we also loop in functional biology, specifically using microscopy, injections, electrophysiology, and even videography to monitor whole organism behaviors arising from single cell processes to truly integrate now this multi-omic information with organismal and functional identity. And so it's biological instrumental development crosses over to applied biology essentially in our lab. Our cells are from 250 microns in size, so you might consider them giant, but remember, these are in developing systems, and they also go down to the somatic cell size as we get to later stages of development. So one of the first challenges in these dynamic, live developing systems is how do you obtain the single cell contents? And that's true for metabolomics, for proteomics, for sequencing. Now, you surely can dissociate the tissues, but then you risk losing the spatial identity. There are some encoding approaches for that, of course, nowadays. So we came across this problem very early, and initially we dissected out single cells from the embryo where we could identify single cells. Nowadays, we're using capillaries that we could very finely sharpen and bring to single cells in the embryo, pierce the single cell, and then collect the content of the cell. We developed this about 8-10 years ago, and we're very happy to see this now in other labs also being adopted to move similar studies forward. Now, we've also demonstrated that this capillary microsampling approach can be scaled down to single neurons in the mouse brain, and now we're at 5 minutes down to 5 seconds per sampling event. That's what you need if you're studying dynamic systems, right? If your cell is developing, literally in a minute, you don't have time to faster around the cell. You need to collect it right now and right then. And in 5 seconds, we can do that. So then we've developed metabolomic and proteomic strategies to separate, sequence, identify, quantify small molecules to proteins. We then use network analyses and advanced statistics and bioinformatics to decipher cellular phenotypes. At the heart of our technology lies a custom-built capillary system that we actually built with Jonathan Sweether when I was a postdoc with Jonathan. And so this idea here is capillary. This is not liquid chromatography or imaging mass spec, as you will probably hear in later talks. So here we're injecting very small amounts of samples down to about 10 nanoliters into a capillary filled with an appropriate electrolyte. We're then applying up to 30,000 volts across this electrolytes, so we're electrophoretically separating out ions based on size and charge, as I will show you very quickly. And then we're generating ions, essentially an ESI source, to then identify them, quantify them by a mass spectrometer. So it is different than LC in terms of separation. It is definitely different than doing imaging-based single-cell analysis, because you've got separation here in the liquid phase, right? So this is going to take more time, but as you will also see, you're deepening the molecular coverage as a result of separation. So over the years, we've learned how to use this platform, and this is what it looks like. This is a custom-built C platform, and an Orbitrap mass spectrometer, and a microscope next to it. Now, we have used this platform for metabolomics and proteomics also, right? Because capillary is just once charged species. It doesn't matter for C if you're a protein, a peptide, or a small compound. You will separate as long as you have a charge. And so here you can see representative compounds, small metabolites being separated, being identified based on accurate mass, separation time, fragmentation pattern, isotopic distribution analysis, and more recently also mobility-based collision cross-section. So this is the deepest level of the molecular coverage in terms of confidence, I should say, that you could today attain to consider a metabolite signal confidently identified. We believe in confident identifications because to do functional biology, you need to know very confidently what metabolite you are going to be after. The platforms we built are quantitative, both for metabolites and proteins. And here you can see quantitative responses as a function of concentration versus signal abundance in terms of under the curve peak area. This is a separation technology again. We can go down to about 30 zeptomoles, which is 18,000 copies of a particular peptide, or about 25 attomoles for roughly metabolites. And we've built over the years 4 different generations of these platforms because they worked really well. Each time we learned something new. So we essentially improved sensitivity progressively and then added new measurement capabilities. But now then we got to the point a few years ago that now we needed to measure multiple cells, single cells. So now higher throughput measurements became the need, which our manually operated CEMS platforms just couldn't keep up, right? We're manually injecting in these platforms. So we most recently designed this idea that is inspired essentially by a multi-well array that can hold multiple single cells that could be sorted. And then the idea is, what if we could now make CE mass spectrometry amenable to such little amounts of samples, down to 200 nanoliters of processing samples, so we could enable a CEMS-based metabolomics approach. So we just published this in Analytical Chemistry. You will see the design there. Essentially, instead of manually lifting a stage to inject, we're enclosing into a hermetically enclosed enclosure all the 3-dimensionally translatable components of a capillary, a micro positioning system, so we could robotically, remotely bring the C capillary to the sample. Actually, we do the opposite way around, but doesn't matter. And then we apply a pressure in this system, so we could literally chase the sample into the capillary using pressure-driven injection. We have very thoroughly validated this platform. And now we know that it's quantitative, so we could literally monitor the amount of sample we could inject into the capillary, and we demonstrated across an entire day's worth of analysis, reproducible identification. So this is the first that we know of, low volume, less than 200 nanoliter volume compatible CMS platform that theoretically now opens the door to a CEMS-based alternative to what LCMS-based high-throughput proteomics can now do. So on that note, this is kind of what the platform looks like. Here's our RoboCap, as we call it, robotic capillary positioning system, coupled to a platform. And as you can see, it is completely self-standing and portable. So we hope to be able to demonstrate this. Now, we've used these platforms to enable dual proteome metabolomics. And here we were able to go to single cells, where we basically were able to aspirate contents of identified single embryonic stem cells, then take one pinch of the cytoplasm essentially for proteomics, another portion for metabolomics. So now we could do discovery deep proteomics and metabolomics of that single cell. And then we were able to raise up these embryos to ask whether we could downscale the amount of damage to the cell so much that the development is not perturbed in any way. And we succeeded. So essentially now we know that we can measure using our RoboCap kind of platform so sensitively that we could identify about 2,000, 3,000 proteins and about 200, 300 metabolite signals we're detecting in these single cells in the embryo, where we could now do dual proteometabolomic single cell in vivo mass spectrometry, giving rise to a larva and a tadpole, who, by the way, look like and function behaviorally also as they are supposed to be, with the big benefit of now getting both proteomic and metabolomic data to improve molecular pathway coverage. And then we've used that over the years to ask various questions. Now, I'll show you very quickly one example that I think you might find interesting, since the conversation of do metabolites matter comes up. I'm a firm believer that yes, they do. So this is the early development of the embryo. Here's 16 cells. We reproducibly know, well, we know the reproducible tissue fate of every single cell in the embryo. I'll spare the details how we know that. I'm happy to explain that later. But essentially, this cell is precursor to neural tissues, and this particular cell is precursor to the epidermal, so skin essentially. So we very early dissected these single cells. We did discover metabolomics using the CMS platforms I told you about. And then we obtained both statistically and fold change-wise biologically significant differences in metabolomic profiles in these neuronal versus epidermal faded cells. And then essentially we were able to assemble entire metabolic networks that operate in these cells to find that certain nodes of metabolites were more upregulated versus downregulated in this cell fate. So this was a first example of dorsal ventral axis heterogeneity at the single cell level, animal and vegetal axis single cell heterogeneity at the metabolite level, and also left-right axis single cell heterogeneity at the metabolite level. So we asked the question, do these metabolites matter for encoding the cell fates? So we first determined how much of these metabolites are present in the cells. And then we simply said, if histidine and threonine are more abundant to yay amounts, as we quantified up here, well, let us prepare this cocktail and inject them into this cell here, right? And if it does anything, we should be able to track the fate of that cell. How do you track a cell fate? Well, you express some signal in it, and very interestingly, we can just simply co-inject GFP, green fluorescent protein. So that means any tissue, for example, that descends from the D11, this neural tissue faded cell, lights up in green, and lo and behold, yes, the retina, the brain, and central somites in the control tadpole larva light up. And this is now where it gets interesting. The moment we injected the metabolites that were upregulated in the ventrally faded cell into the D11 we injected, look what happened. The cell fates totally changed. So we reprogrammed just by injecting acetylcholine, methionine, and alanine, essentially, normally neural tissue faded cells to become epidermally faded. We've since also validated this can work the opposite way around, albeit using different compounds. So since then, we have gone into cell-by-cell studies in the developing embryo to discover entire metabolic gradients that operate long-range cell-by-cell in the embryo. We also ask the questions whether these gradients are cell-autonomous, meaning the cells just do this on their own, or whether they fulfill a biological role. So we have conducted isotopic microinjection studies very early on to essentially elucidate metabolite transfer dynamics between neighboring cells. I'll spare the technical details, but essentially what we found was that depending on the compound you pick, some compounds do transfer to the neighboring cell, others don't. And then we also have been able to design pharmacological interventions to prove that these cell transfer processes are actively controlled by what currently appears to us as gap junctional proteins or some ion transfer proteins. But essentially, these gradients are established for a reason in the embryo. They're not At least we thought, right? They're not the byproduct of just something. And to prove this, we have also conducted dual color imaging experiments. So bear with me. So this is an embryo in which we can differentially, using two different color, label the D11 and the D12 cell fate. So any tissue arising from this cell is green, any tissue arising from this is red, even though these two cells are right next to each other in the early embryo. Look, one forms neural tissues, retina, brain, the other one just forms essentially your head's epidermis. So they're very differently fated. Now, based on methionine, we knew that methionine can alter cell fates because we discovered that earlier. And essentially now we found that when you inject methionine into the D11 cell, the fate of the D11 cell changes, but not that of the D12. But if you inject methionine into the D12 cell, now you alter the fate of both of these cells, and you might wonder why. So it turns out we've since also validated there are metabolic gradients that are pointing this way. So bringing methionine up to the D11 cell for mechanisms that I don't have time to go into. And essentially, if you perturb this cell, some metabolites will cross over and perturb that cell too. Right? But if you only inject here, the gradient is not pointing backward, so it's not retroactively infecting the cell phase. The cool thing is that you're now altering cell lineages, and I won't have time here to demonstrate, but we also this way altered the actual behavior of the entire tadpole in whole organism behavioral assays. Now, I don't remember how much time I have left. Hopefully I'm still okay on time, but to continue, so now in the embryo, we also are able to use capillary microsampling and literally go cell by cell as the embryo is developing to map out in space and time how the metabolome or the proteome is reorganized as entire live tissue lineages are forming in this freely developing live embryo. So this is not fixed tissue, right? This is a live embryo. And so here you can see this cell, the two offspring cells, and then one of these mapped in space-time across these spatiotemporal coordinates, essentially, of the developing neural tissue faded lineage. You can see GABA going up in abundance, aspartic acid going down, creatine staying stable. You can also now do more high-throughput cluster analyses to extract entire metabolic gradients. And then you can do the same for proteins, right? Because now we've also are able to use CEMS to do proteomics. I don't have probably too much time for that, but essentially, no matter whether we look at the metabolome or the proteome, we're finding reorganization in these single cells that are indicative of the fate, and through functional studies, we validate that they are perturbing and controlling the cell phase to some extent. Now, if you're not as much into embryos, it's okay, I don't blame you, but you might be interested in the brain. And so here's another example, because a capillary microsampling can be scaled down to a patch pipette. So now you can go to a neuron that is kept alive in a coronal brain section, as we did a few years ago. You could record the electrical activity of the neurons, tell, for example, a dopaminergic from a GABAergic neuron apart, and then we could take the patch pipette to aspirate the content of the neuronal soma and then measure just 1 picogram of the proteome using our CMS system. So this is about 1,000 to 1,000,000 times smaller amounts of proteins measured in these platforms than historically has been on CEMS. So we're actually measuring about 0.25% of the total neuronal soma, so we're kind of wasteful. We could be measuring more. But anyways, I just wanted to quickly show you an example that we're about to publish, where we went to 3 different neuronal types in the mouse brain. One was dopaminergic, paravalvumin interneuron, and serotonergic neuron. And we identify these neurons by electrophysiology, and we solely ask the question, if we conduct protein analysis on the soma, the cell body, so not even the dendrite and the axons, just the cell body of the neurons, are we able to distinguish these cell types? And the answer is, after quantifying about 800,000 different proteins, yes. So now we can literally do discovery analysis of these cell types in the mouse brain at the actual 30 to 20 micron neuronal level. And this is probably the last two vignettes of analysis toward fast CE measurements, as this is an overarching topic that will come up today, especially with imaging mass spec. So we've recently enabled the less than 15-minute total proteome analysis of a single cell by CEMS by recognizing that data-independent acquisition does not sacrifice the number of protein IDs as you shorten separation times in CE, unlike data-dependent analysis would. So this means that we could go to a very short separation time and still able to identify the same number of proteins as we would using a much longer hour, maybe even 2-hour long separation. So that means now we've got about 3,000 proteins that we can identify essentially and quantify accurately, right, in just 15 minutes. And more recently, we are able to move this also to smaller amounts, timeframes. Now, we've also introduced electrophoresis correlative data acquisition recently. This is now published in Neurochemistry. And the idea here is that unlike liquid chromatography, in CE, you're separating your compounds based on mass, but also charge, essentially by mass to charge. Like a weird liquid form of mass spectrometry, I always jokingly say, right? So here are the different M over Z values as time goes by in C separation. Essentially, what are you doing? You're pre-filtering the ions by C, right? You're telling now the mass spectrometer, look, I know what M over Z values will be coming into the mass spectrometer because the C sorts it, right, at any given time. So we propose to tailor data-independent acquisition and recently other kinds of data acquisition methods to these very specific predictable trend lines, essentially. And that allowed us to deepen the detectable proteome by 40%. More recently, we got even bigger improvements, about 60% improvements, while speeding up the analysis thanks to CE. So we're very excited because this now means that we can do CE single-cell analysis fast in an automated fashion using RoboCap in less than 15 minutes, more recently about 5 minutes, and to really deep coverage of the proteome and the metabolome. And now we've embarked on a project thanks to the Chen Zuckerberg Foundation, the initiative, where we're literally mapping out the cell atlas in the early developing embryo, cell by cell for every cell, what the proteome, what the transcriptome and the metabolome do in these cells, so we could create a language essentially to translate what happens to the transcriptome from what happens to the metabolome from the level of the transcriptome. And we're also validating this in other developmental models, for example, the mouse. So without further ado, much left. So essentially, single cell mass spectrometry, I think, is an exciting technical development that's worth keeping an eye on. It is qualitative, quantitative. It actually pioneered single-cell proteomics in the field and many areas of single-cell metabolomics, so it's important not to forget it. It is fast, it offers deep proteome coverage and metabolome coverage, and it's a nice way to complement maybe faster and even higher resolution alternatives today by mass spectrometry imaging, liquid chromatography mass spectrometry. We have used this CEMS as a tool, a primary discovery tool, to discover novel biology. I don't have really much time to go into that, but please follow our publications or reach out to me. And so we have been very happy to open new research opportunities, both from fundamental to translational research topics using CEMS-based metabolomics and proteomics, thanks to my group who have worked really hard to make all of these technical and biology applications possible, and the many funding sources and peer reviewers, I always say, who believed in our wild ideas at some time. So thank you. I really appreciate your support. Last but not least, thanks for your time, and I'll be very happy to take any questions.
Thank you very much. Yeah, so we've got 10 minutes for questions.
Oh, great. Okay. Here I was worried running out.
That's fine. So first question is, what do you think are the molecular mechanisms by which single metabolites deviate differentiation trajectories?
Yeah, how does that work? That's literally the million-dollar question as a grant, right? So essentially, we have been asking this. Let me just go back here. So this is where we demonstrate, right, that if we inject basically methionine, as we later found out, it's a very potent disruptor of normal cell fate. It very easily reprograms the normal neural tissue fate to become epidermal. But how does that work? So we have done proteomic studies and isotopic labeling to ask different questions. Here's why this becomes challenging. We inject methionine, right? You might think methionine is the one doing the action, but what if it's not methionine? What if this methionine metabolizes into something else downstream that is far more potent? So our question actually became much bigger. So first we started tracking down methionine metabolism by isobaric labeling. We identified the potential partners. We actually found some that are actually downstream produced. So now we know who that is. I'll hold the information back for a publication. And then we validated that if you inject that compound and also methionine, you're now causing post-translational modifications on certain proteins that we actually believe to be epigenetically related. So that's kind of what I can say. We hope to also bring in transcriptomics to kind of piggyback on another omic area to validate this. But yeah, so it seems like probably, perturbation of maybe some metabolite signal that maybe the cell is detecting. We kind of see the protein change aspect that might be pointing toward epigenetics. We don't have a fully developed picture of the mechanism of action yet. So if you have ideas, I would love to hear your thoughts. Thank you for the question.
All right, next question. So thinking about organisms with different developmental speeds, what do you think are the key different strategies to regulate metabolomic concentration and symmetry breaking in these different species? And are there any time constraints to building up metabolomic heterogeneity?
Oh, that's a really, really good question. Yeah. So yeah, organisms develop at different speeds. Absolutely true. And that makes the experiments sometimes maybe easier or more difficult. So we've actually worked with also zebrafish embryos, right, that have a different time frame of development, and also mouse embryos. That is a whole different story. What I can tell you is mostly about the frog, since that's where we've mostly worked. So we have asked the question, if we start probing symmetry break-in events, left, right, dorsal, ventral, front, back, or even if you go to the same exact cell that then is supposed to divide into two halves, right? One half may be giving rise to a neural tissue fate, the other to an epidermal fate, because that happens too. Can we actually find subcellular and single cell heterogeneity? And the answer is yes. So now we can also go to these much earlier than single cell forming events. Very, very weird, right? And find now molecular reorganization within the single cell, both at the level of the proteome and the metabolome. We haven't published that. I hope to be able to. Sorry, I gave a little bit more broad answer. Hopefully I answered some of the question.
So next question, what do you think about the complementarity of CMS to MALDI-MSI, and were the main areas where CMS work while MALDI-MSI doesn't?
Yeah, that's a great question. So I suppose you might be an MSI person asking this. Maybe reach out to me. I would love to actually team up to do MSI and kind of really answer the question. But joking aside or not, I'm happy to collaborate. I think the main difference will be definitely speed and spatial resolution, obviously, right? I mean, with MSI developments nowadays enabling 5-micron resolutions, or sometimes even better, I hear. And I'm sure that Jonathan Sweedler and Elizabeth Neumann and Theo are gonna talk about this later too, right? You've got really high sensitivity that you're able to monitor cells embedded even in a tissue. I think the challenge becomes, what do you do when your organism is not flat? Right? I mean, most organisms are not flat, right? Otherwise, it would be worms. So, and in the embryo, it's 3-dimensional. So we don't really have the ability to, you could section the embryo, but then you're still missing out cells, right? Into different sections. So we had to go with more C and manual sampling of the cells that are occupying in different 3-dimensional objects. But with MSI, you could do the imaging very fast at much higher spatial resolution, as long as you have the correct sample. Now, I know that there are really nice tricks that Jonathan Sweetler probably will tell us about, maybe dissociating, for example, tissues, right? And then you're able to trace also cells using optical cues and other data. So there's many different ways of doing this. Yeah. Maybe another difference could be metabolome coverage, right? How good are we at identifying signals in CMS versus MSI? Metabolomics is going to be challenging no matter what, right? As long as you do metabolomics, you will know the challenges. But CE-based challenges are oftentimes different than MSI-based identification challenges are, right? We're also obtaining different amounts of information. So I think CE might add a little bit more information to aid metabolite identifications, but I also recognize it's much slower. And if you're seeking to measure a lot more cells, either you maybe pick a RoboCap, hopefully we'll get there soon, or you really go with the MSI approaches that are really mature and nicely performing today.
Great. I think we have time for one more question before we move on to our next speaker.
Mhm.
So metabolomic methods are often tissue destructive. What are your approaches for multimodality studies combining spatial transcriptomics, proteomics with metabolomics in the same sample?
Yeah, they're a very good one. So the closest we could probably get is, I'm trying to find, can you still see my slides?
Need to share.
Need to share.
Oh, I still have to share. Sorry about that. The closest I can get to is when we were doing the multi-omics, proteomics, and let me see. Where is it? No, I'm not finding. I don't know where I put it. So basically, when we are able to pick a capillary, oh, here, it was right in front of me. So when we're able to go to a single cell, and so I guess the first question is, would you like the cell to survive the analysis? Right? Do you need to have in vivo analysis? And maybe the answer can be no. Then it's much simpler. We intentionally tried to demonstrate an example here where we enabled in vivo conditions. And our trick was to downscale the amount of damage because the cell's so small to such a small level that the cell will allegedly shrug it off and develop, right? And we demonstrate that's feasible. Now, these cells are really large. So, these are about 250 micron, if not 350 microns in size. And we sampled a very tiny, maybe a nanoliter, 2 nanoliters portion, one's for metabolomics, one for proteomics. So that cell probably could tolerate that, right? If you're now going to a mammalian cell, this becomes increasingly difficult. So maybe that's why for the cell to survive, maybe is not probably as important, and maybe then you can think about sequential analysis where you could extract the metabolome first, do proteomics, then so maybe that's an alternative. You could also use imaging-based approaches, right? Metabolomics versus proteomics. There's so many ways we can do this nowadays.
Thank you, Prof. Nims. That was quite a journey. And anyway, if anyone has more questions, feel free to reach out over the Discord we're going to open soon. Quick announcement for those posting in the Q&A, feel free to add your name and affiliation so we can announce you properly. And for now, I guess it's the turn for Melanie Bailey, Professor of the Department of Chemistry at the University of Surrey. Melanie, the floor is yours. Thank you for being here. Okay, we are in touch with Professor Bailey. She's about to join, so please bear with us for a couple minutes while we set up all. Thank you. All right, we briefly reorganized, and I think Janshu from Harvard for Broad Institute and MGH can take the lead for now, and then we'll coordinate for the next talks. Thanks again for your patience.
Hello. Can you guys see my slide?
Yes.
Okay. Thank you for inviting me to give you an overview of some of the work that we've been doing in the lab. So my lab is very interested in developing tools to map the cell fates. So in order to do this, we're very interested in developing new genotyping technologies, new phenotyping technologies, and new perturbation tools, and then working this on the new model systems. So this is the lab and also the funding support. So we, as I mentioned, that we're very interested in multiplexed decoding and engineering cell fates, but due to the time constraint, I don't have time to talk all of them. Just to give you three main topics that we've been very interested in the lab. The first one would be the multiplexed genotyping. We've been working on in situ sequencing methods to map the genetic variants, mutations, microorganisms. And on the second part of it, which is the multiplexed phenotyping, and I will talk about this today, and that is the multimodal phenotyping and data integration and translation. And third part is the multiplex, the perturbation. How do we actually add multiplex the perturbations to the genotyping, phenotyping effort that we've been working on? So I will focus on the second part, the multiplex, the phenotyping. And the fundamental grand challenge and the questions that not just us, like everyone probably in the audience is interested is how can we actually map cell phenotypes? Cells were initially discovered through microscopy by looking at the morphologies. And in recent effort that the Human Cell Atlas, HuBMAP, and many other consortia are trying to categorize the cells through single-cell sequencing technologies. But there's the pros and cons of each of these methods. For example, the sequencing-based methods are usually destructive and expensive and also low throughput. On the other hand, imaging-based phenotyping approach can be non-destructive, easy to do. But the challenge is that it's actually very hard to interpret what does it mean with those imaging features. So we wonder, can we actually combine the best part of these two technologies so we can map the cells at large scale and more efficiently? So the solution would be, how can we just combine imaging data with different kind of single cell omics data through machine learning or generative AI? Because the technology foundations behind this is the recent advancements in AI and machine translations. Some of the examples that I listed here, such as ImageNet, AlphaFold, and DALI, they're all doing this translation and tasks in the ImageNet. And we're now people are trying to teach the computer to recognize what is a dog, what is a cat, right? And in the AlphaFold, people, not just AlphaFold protein structure prediction, people develop different kind of machine models to predict protein structure from sequences. And in the recent effort that, OpenAI's DALI or even Sora, they're trying to generate text or videos from, images and videos from text. So we ask the same questions, that can we actually do this machine translation in biology across different scales? And then there are a lot of great effort and pioneering work focusing on the DNA and protein structure prediction. So we wanted to move to the next scale. Can we do this at the cell and organ level? So we wanted to use the same strategy to combine high-dimensional imaging with high-dimensional single-cell multi-omics measurement. And the first example, first tool that we developed is called Raman2RNA. This is in collaboration with Dr. Aviv Regev and Peter and Tommaso at Genentech. And the idea is, can we build an AI model to predict single-cell RNA-seq from non-destructive label-free measurement? And the imaging that we tried was Raman hyperspectral imaging. So the idea is that you just take pictures using Raman microscopy and then connect that with the single-cell RNA-seq through some anchor genes that are measured by single molecule fish. And then in the end, you can predict the single cell or generate the single cell RNA-seq from just taking pictures or time series videos. So why are we starting with chemical image with Raman microscopy? Because Raman microscopy belongs to one big category of imaging that is called chemical imaging. Chemical imaging actually is fairly new to the biology community, and it measures the chemical fingerprints or profiles in a non-destructive and label-free manner, and also high-dimensional. Because if you think about single-cell gene expression or single-cell multi-omics measurement, or multi even, it is relatively high-dimensional. So from the imaging space, we need to use the high-dimensional imaging measurement as well. So from this point of view, that's why we started with the Raman microscopy, which can measure up to 1,000 dimensions per pixel. If you think about a fluorescent imaging, which can give you maybe 5 to maximum 10 colors or channels per pixel. So this is 100 times more information and in a label-free manner. So which means that you can do this in live cells or even in vivo settings in humans. So here's what it looks, how it works. So first we use the iPSL reprogramming process as a model system. And then we took pictures using Raman microscopy, and then we chose a few genes and then did single molecule fish on the same dish, and then across different time points. And then we build a neural network, and then integrate this with the single-cell RNA-seq data using Tangram. So, and then later on in the inference stage, we just predict the single-cell RNA-seq from the Raman pictures. And then you can see that this is what the embedding look like with just the Raman images. And this is the single-cell RNA-seq embeddings. And then these are a few genes that we predict, like demonstrated that you can actually do prediction pretty well, but not all the genes, because this is just a proof of principle study how we can actually generate single-cell RNA-seq data from any of this imaging modalities, especially, for example, this gene's COL1A, which is a fibroblast marker genes, but it is not very specific to just the fibroblast, and it's also expressed in many other cell types at different levels. So that's why the performance is a little bit low in these genes. But if you look at the cell type level, and that the prediction works pretty well. So that's the first methods that we developed to generate single-cell gene expression using Raman microscopy. And inspired by this, we started to work on other imaging modalities which are more clinical relevant and easy to generate. And then we started to work on the histology images. And this is, again, together with Dr. Aviv Regev and Colin Stoltz at MIT. So we developed this method called SHAF. And it's the same concept. Can we just predict single-cell RNA-seq or spatial transcriptomics data from histology images? And in this case, there are two scenarios. One scenario is that you have paired data from the histology images and the spatial transcriptomics data from the same slide. So then you can use the paired model. But if you don't have this paired settings, paired data, you have HE staining from one slice or one sample, and you have single-cell RNA-seq or spatial transcriptomics data from another sample, which is actually the case in most cases. And we can use the unpaired model to do this prediction. And just let me just to show you some of these examples. And this is in the paired settings that the HE and spatial transcriptomics data are from the same slice. And you can see this is the HE stains, and then the middle panel shows you the cell types predicted by SHAF, and the third column is the ground truth measured by 10x Genomics Xenium platform. So you can see that at the cell type level, this looks pretty good. And then we asked, can we actually predict individual genes using SHAF? And then here is just some of the examples that SHAF can do pretty well. Some of these genes, but not all of the genes, and because the data is just not enough. And so that's the paired settings. So and we also asked the questions that can we actually do this in the unpaired setting where HE staining, HE images, and single-cell RNA-seq are from the two different samples. So we use this in one of the new multimodal multi-omics atlas that we generated. And to get as a part of the Human Cell Atlas Consortium, which also like the first paper bundle just came out a few weeks ago, we generated a single-cell multi-omics, spatial multi-omics atlas of the human placenta, and using single-cell multiome and 3 complementary spatial transcriptomics method, slide tags, StarMap ISS and StarMap ISH. So these are the data that we're using to train the model, but this is in the unpaired setting because the HE staining and then the single cell and spatial transcriptomics data are from the different slices. So let me just show you the results. This is the cell types inferred by Schaff. And this is the ground truth cell type annotated by pathologists. So you can see this looks pretty similar. So this is at the cell type level. And then we also ask the same question and how well we can do at the individual gene level. So you can see that some of the genes that we listed here are different kind of marker genes for different cell types in the placenta. And in the top row is the genes that we inferred by Schaff. And then the second row is the genes inferred by the ground truth gene expression measured by StarMap, which is a targeted in situ sequencing method. So I hope that I can just give you an idea how we can generate different kind of single cell and spatial transcriptomics data from different kinds of imaging modalities, which we showed. The first case was the Raman imaging, and the second case is the HE staining. But these are the imaging modalities are, every imaging modality has pros and cons. And that's why we are still exploring. And I welcome people who are in the microscopy world to reach out to us if you have some cool imaging data. I would love to, collaborate and work with you. So beyond the Raman imaging and HE imaging, we're also testing other imaging modalities. For example, the left figure shows you another label-free imaging, chemical imaging methods, and we don't need to do any staining, and this is just purely just clustering based on the imaging features. But the advantage of doing this imaging, the chemical imaging, is it's extremely fast and cheap. It only takes less than 10 minutes to generate these images. And without any staining or without any, labeling. And on the right hand, which is another type of imaging method called quantitative phase. These are all the wonderful collaborations with doctors Wei Ming at Columbia University, Peter Su at MIT, and also Dr. Colin Stoll. So hopefully we can share more results using this 2 different imaging modalities to generate single cell and spatial multi-omics data with you. So as I showed you that we demonstrated that you can actually generate single cell and spatial transcriptomics data from images, we asked the reverse direction. If you have, a massive amount, because there are already a lot of more than probably like 100 million single cell gene expression profiles, can we actually generate the cellular or tissue pictures across different modalities? The motivation was because when we started to talk to, pathologists, and we thought that, if we give you a single cell gene expression, then you can actually tell us what is the pathological features in this. But it actually turns out to be very, very difficult because it's kind of like people speak different kind of languages. So pathologists, they are not familiar with, a HuBMAP or like a single cell gene expression profiles. So they wanted to have a histology images so they can look at. But oftentimes in clinical settings, you just don't have the opportunity to do different kind of measurements. Probably you can only do one measurement. So you have to choose whether you want to do, single cell RNA-seq, or you wanted to do, MALDI, or you have to do, IF staining, but you only have one chance to do this in one slice. So that's why that motivates us to develop this method called Generative Da Vinci, to do this image, like a gene to image generation. And the input for Generative could be single cell, single nuclei, RNA-seq, spatial transcriptomics, and in the special case with in the neurology, it's the patch-seq data, and then we put it in Generative, which is a diffusion model, and then we can generate different kind of single cell single cell images or tissue images that include HT staining, DAPI staining, IF staining, neuromorphologies in electron microscopy images at the single cell resolution, and also at the HT staining at the multi-cell and whole slide resolution. So let me just give you a few examples. And here, the first column is the ground truth, and then the next 5 columns are that we generated multiple pictures from the gene expression as an input. But when we train this, the model never sees the ground truth images. So and then we ask the model to generate those pictures. And you can see that we can generate HT staining, DAPI, immunofluorescent, even just electron microscopy data, and also neuron morphologies. And just zoom in to see, just to give you one example, how we can translate the gene expression to the neuromorphologies. And then the first column is the ground truth, and then the next 5 are the Generative generated cellular pictures. Because the goal is to understand the relationship between gene expression and the cell morphology, because the cell morphology is linked to function. So how can we build build this translator to actually learn the relationship between this two? And then you can think about what if there's a perturbation in the gene expression space, what the morphology might look like, and how that will lead to different kind of pathology or and function. So that's the Generative. And we also have quantitative metrics to show that the generated pictures are actually very similar to the ground truth images using 2 different scores. So one is called FID, and the other one is called saying similarities. So that's a brief overview of some of these tools that we developed in the lab to combine imaging with the genomics. And I hope that I just give you this, an initial evidence and proof of principle evidence that we can build a translator between different kind of languages in biology. We show that you can generate or translate between RNAs and different kind of image modalities. But this is just at the beginning, I think, of a very exciting era for digital biology. How can we, work all together to build maybe like in the future a Google Translate for different kind of languages in biology, especially for a lot of the consortia that we'll talk about today, like HuBMAP, CZI, and Human Cell Atlas. How can we work together to fill up the gaps across the different kind of data modalities, and then hopefully in the future that we can achieve this Google Translate of biology languages. But the problem is that, and I think that echoes with everyone's effort here, and people are trying to build organize the database across different kind of modalities and consortia, but that is actually still a huge challenge for developing better computational tools. And because unlike, for example, AlphaFold, there's a well-organized protein data bank, but we still don't have this multimodal database that is highly organized. That's why that's essential for HuBMAP, CZI, and Human Cell Atlas and other consortia to work together to build this organized database, but also include other data modalities in addition to just the single cell multiomics or metabolic data. So hopefully in the future, that I think the solution should be that we need a company like Scale AI to organize and curate the data for biology, and also that there's a huge need for computing resource. So maybe I would just end here, and thank you so much. And this is my contact information, and thank you. And we're also looking for talented postdocs. So if you're interested, just reach out to us.
Thank you, Prof. Xu. That was truly inspiring. And now, unfortunately, we do not have time for questions, but we're in perfect timing. It's 5 p.m. and it's the turn of Jonathan Swidler, one of the true huge pioneers of this field. professor in neurochemistry and much more at the Illinois Urbana-Champaign University. Please, Jonathan, the floor is yours. Thank you again for joining us.
Thank you. Can you see my slides just to check?
We do. Thank you.
Good. So welcome, everyone. It's a great pleasure to be here. It's an amazing day. Here I thought I would be in Illinois on a cold winter day doing, doing some reviews, and instead I get to be listening and learning about some spatial metabolomics and single-cell measurements. So it's a great improvement. Thank you for organizing this. So my group has been in this field, it's almost hard to say, single-cell measurements for a third of a century. I think we published our first paper long before people told me I was crazy to try it. And of course, it was one metabolite at a time, and things has changed. And so today, I want to sort of take you through some of our approaches. I don't have time to go through too many. It's sort of interesting to think about a history. This is a very incomplete timeline of single-cell measurements. And I like to put it up for two reasons. Microscopy came first. Everybody knows that. But if you go to the brain, the microscopist got to name cell types. So you think of Hooke or Leeuwenhoek, they said, oh, those cells are star-shaped, stellate or pyramid. And at some point, people thought the shape defined function. And so if you think of the cell atlas, that was one way to do it. Chemists came along and said, well, let's look at serotonergic cells, fancy way of saying they use serotonin, or dopaminergic. And it turns out those could be different shapes. And even sometimes some cell could have one chemical some of the time. And so cell state versus cell types are something to think about. But it really wasn't until the 70s that single cell analytical chemistry really came of age. And then the 90s when transcriptomics, I think the first example was Jim Eberwein with single cell transcriptomics in 91, where you got wholesale measurements. And what's changed is these people on this call, things have gotten much more robust, much easier. It's not easy now, but it's certainly, it's more doable in large numbers. It is worth pointing out is if you look carefully, you'll find examples of GC, gas chromatography, mass spec of a single cell in the 70s, a slab electrophoresis of a single large cell. And then I love his work of the 70s from Frans Hillenkamp. This is long before he discovered MALDI and reported MALDI, but this was actually using LAMA. And he had 3 to 5 micron resolution. Okay. The mass spectrometry wasn't as good because, of course, it was the 1970s. But there's a lot of things that haven't really, changed in terms of the need. And you can actually see where his cells head. So you can find some examples long ago. So I'm going to start with 2 major approaches. One, I don't have to go into much detail because of Peter Nemisch did a great job. But you can isolate a cell. That could be by hand, by tweezers. It could be laser capture, microdissection. It doesn't really matter. You put it in a vial, and then you can measure it. And Peter showed some great examples. This is just one. We've been doing this actually with different detectors, mass spec, since the late 90s. But if you can isolate the cell and you use mass spectrometry as the detection limit with the attomole to zeptomole, you can see things in it. And this includes amino acids, peptides, and others. And this is just showing you one example. The entire important part of this is to prevent dilution. If you take a picoliter cell and dilute it to a microliter, that 6 order of magnitude makes mass spectrometry's job hard. And so we oftentimes, let me get a better laser pointer, we use very small samples where we just put it on a steel, in a nanovial of steel sheet, so we can actually minimize dilution and then get higher detectability, and then sample stacking and others. And so this works, and we can see differences. It is serial. And so in some ways, even with Peter's amazing increases in throughput, it's still fairly slow. So I thought I would spend most of my day, remaining time, talking about peptides. Neuropeptides are gene products. And so you can say, why not use transcriptomics? Well, it's DNA, RNA, protein, a prohormone, but there's so many enzymatic post-translational modifications that the protein to peptide conversion can't be predicted. It's different depending on the cell, and that changes the peptide's biological activity. You have to make the measurement. This is another way of showing it. You have this long prohormone with amino acids. You cleave it at KR sites, and you add amidation, phosphorylation, pyroglutamate, lots of others. They can be released at a cell soma. Oftentimes they are. It can be released at a terminal. It can be released in lots of different ways. And these are the molecules of life in the sense that neuropeptides are what determine whether you're attracted to somebody, whether you want to eat. I think it's been exciting. I've been working on these a while. When I went to the Society of Neuroscience meeting in October in Chicago, tens of thousands of people, this is the time of neuropeptides because methods to modulate these, the drugs to do this, are moving up. to the point where they're weight loss drugs, they're diabetes drugs. A large number of the top 20 pharmaceuticals in the world right now work on or mimic these peptides. So let me show you a historical slide, and then I'll show you what we really do. But this sort of is kind of fun because this is some work from 20-something years ago. You can take a group of cells from transcriptomics and stain them. You can pop off a single cell, and you can measure it. And if you put it on a target, right there, it's under cell culture, you hit it with a laser. This is our picture of Star Wars. I guess it's really multi-mass spectrometry, and you get a profile of the peptides in there. And even a chemist can say, if this single cell as opposed to the others has a peptide that's different, and this cell, by physiology, drives this particular creature, a Nobel Prize winning sea slug, Ecclesia californica. Eric Cannell got a Nobel Prize for doing some work on defined networks. If this cell drives swimming behavior, maybe that peptide drives swimming behavior. So you can go from a single cell to a function. And by the way, our cell is different. Every one of these cells have different peptides, different transmitters. They're all different. Not by little bits, but a complete metabolism in this creature. The cells are incredibly distinct, and it also validates our approach because the physiology and the single cell measurements match and align. Okay, let's make it a little faster. This is what we do now. We could use the Zenobi approach at ETH, but we just scatter the cells everywhere. We image them, and then we hit them. And one of the things I want to talk about this approach, which is not mass spec imaging, is that instead of lining up like this with MALDI, what we can do is we just hit the individual cells. We never miss a cell. So it's more reproducible. Each spot is a cell. We don't measure the empty spaces. If you have 1,000 by 1,000 mass spec image and you have 1,000 objects you want to measure, you're only measuring 0.1%. So this is maybe 1,000 times faster. Now that's not true because stage movement is about 100 times faster. Speed matters. And then the spatial resolution is completely independent of the objects being measured if they're far apart. I'll show you that later. This is the software we have available, and this is how we do it. This is actually about 10,000 cells. We have lots of fiducial marks, and now we just hit the laser, and there's different numbers on each one, and this happens to be a cancer xenograph, and so each one of these cells is a cancer model, and they've been labeled with fluorescence, so we know where to hit, and then we can do measurements that look like this. Now, when I first started to do this, people said, how do you know it works? Well, of course it works. We did it in a sea slug, but people said, no, prove it. So I said, let's go to an islet of Langerhans because the peptides define the cell site. So beta cell is insulin, alpha cell is glucagon. And so when we did this from a rat in this particular case, even with classified learning, we separate the cell types to alpha, beta, gamma, delta based on whether they have insulin or glucagon or somatostatin. And the ratios are the same. We can pull out individual 400 or 500 micron islets, separate the 5 to 10 micron cells, and we get great data. Let me tell you, though, when you know something about neuropeptide processing, you learn things. This is the single letter abbreviation code for the pancreatic hormone. There is a dibasic site there, which is cleaved at glycine, which means this is amidated, and there you go. Pancreatic hormone weighs 43.99. There's a dibasic site there. Arginine, arginine are cleaved about 20% of the time in mammals. Is this cleaved? Well, if it did, it would make a peptide of 1296. It turns out these are individual dots or single cells. In the ventral area, some 3 or 4 cells per islet are cleaved, the rest are not. What was fun about this is we already know what this peptide does because somebody said maybe it's cleaved. They determined it binds to a neuropeptide Y4 receptor and it potentiates insulin secretion. Then they did HPLC and said, oops, we're wrong. We don't detect it. I guess it's not cleaved. Well, it is cleaved in a few cells. Presumably those cells have different processing enzymes and make a different peptide. One of the fun things, and it was mentioned, we need non-destructive approaches to do multimodal. MALDI sort of is non-destructive. Here is some single cells. This is on an ultraflex stream. We can then carry the cell to another building where we have our solarics. Now we get better resolution, but on the exact same cell. Because we don't miss a cell with these fiducial marks around the slide, we can do this. We can do SIMS in this case, and then MALDI. We can even do something strange. We can do capillary electrophoresis after MALDI. So we built a CE system, sort of robotized like Peter was talking about. In this particular case, we tell it where to go. And we can say, find cell 1172 by the MALDI image. And we know the XY position. This will go down, grab the cell, suck it up, and put it in a vial. And then we do capillary electrophoresis. This is a CE metabolome of a cell after we did MALDI peptidome. And it turns out if we use, okay, we're kind of crazy, radioactive peptides, 85 to 90% of the cell is left behind. We verify that with the phosphorimeter just a proof of concept. And so 90% of the material is left behind to do the CE metabolomics afterward. Here's a better example. There's lipids. This is multi-mass spectrometry. These say that these 3 cells are selected because they have glucagon. They don't have insulin or somatostatin. And these are the electropherograms. And one of the cells, for example, had authentic dopamine, and we could spike dopamine in. This particular time, we didn't have our CE didn't have good tandem MS. Now it does. What was fun about this is that transcriptomics said that some alpha cells had the enzymes to make dopamine, but nobody had ever measured authentic dopamine. Now we can say it's really there. After we did this in a rat, we then were offered to work on humans. These are human transplant quality islets. They're not as good as our rat islets, but they're obviously coming from a human. They're shipped by FedEx from the University of Pennsylvania to us. We can dissociate the cells. And this is now 4 islets from 4 individuals. And each dot is a cell. Islet 4 has lots of somatostatin cells. Islet 1 has glucagon cells. Can we tell them apart? What are the peptides? And so now we're going to do a timsTOF Pro measurement on individual islets. These are small, but not single cell levels. We ended up detecting thousands of peptides, and hundreds of them are from prohormones. And this just for one prohormone, glucagon, this is what a textbook says. And you see in an islet alpha cell, you should see glucagon, but in an you see oxyntomodulin and others in the gut, in enteroendocrine cells. But we find a couple of islet cells that actually have oxyntomodulin, glucagon. We see other, in purple, unusual peptides with alternative signal peptide sites. Are these real? This is where single cell gets interesting. This is from a human that has type 2 diabetes. And in this particular islet, we just sort of spread the cells out. This one cell has all the oxyntomodulin. It's a huge signal. All the other cells have none. So oxyntomodulin is not supposed to be in an islet. It's supposed to be only in the gut, enteroendocrine cells from this prohormone. And the difference is about 1,000-fold. So we're not looking at subtle changes. People always say, what kind of statistics do you use? We don't need very fancy statistics. So we're talking about 1,000-fold change. Insulin, by the way, is everywhere, and as it should be, in lots and lots of beta cells. So let's move on. I think the next speaker, or depending, or maybe 2 from now, will be Elizabeth Newman, a former student who's doing a fantastic job at UC Davis. This is when she was in the group. We did lipids in this particular case. And we're looking at lots of lipid cells done with Celarix, and the predominant lipid is shown in different colors. We get great data, and so we see a lot of lipid heterogeneity. She was able to see different lipids between astrocytes and neurons, for example. Let's move on. You can ask, is this data robust and reproducible? So after we acquired and published 5 or 6 different papers on this, I had a bioengineer who wanted to do some informatics and looked at our previous data sets. Elizabeth Newman's data is about 80% classified using some follow-up studies, probably because of immunohistochemistry. But if you compare hippocampus versus cerebellum, 96% of the cells can be told that they're either from the hippocampus or cerebellum. That's a surprising result, because this includes microglia, astrocytes, and neurons. Turns out that differences in lipids are bigger from brain region than cell type. And it's a little strange if you look at the fragmented data. SIMS data is even greater differences between our ability to tell apart between different brain regions. So I just wanted to show you a couple examples, and then I'll tie it back in with some imaging. One more example from Elizabeth's data. This was actually looking at developing human brain. It's 100,000 cells, and this was actually done in an ultraflex stream. And it was done in collaboration with Arnold Creakston's lab. And it was just showing you we can do high numbers. And so they prepared the cells. Here they are shown, using this approach where we disperse the cells, we figure out where they are, and then we target them in a high throughput manner. And this is looking at heterogeneity by clusters, and then you can see very large differences between different clusters from different cell types, and they're reproducible depending on what brain region you look at. And so I put this on really just to say large numbers of cells. Since the Creakston lab does a lot of single cell transcriptomics, they actually have this data on their transcript cell reader. And so it's kind of fun to look at. Can you go smaller? Inside, everybody likes to say the cell is the fundamental unit of life, but what about a nucleus or a mitochondria or whatever you want to talk about? So we're looking at single dense core vesicles. and looking at peptide heterogeneity. Now, again, going back to aplasia, these can be anywhere from about 300 or 400 nanometers to about 2 microns, mostly about half a micron. We can disperse them and we can image them. If we spread them out well, then we can hit them with our laser, boom, 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 and then get data. And I'll show you that in the next slide. But if you want, if they get too small, and we're now doing this, I'm not going to show you any data, it's unpublished, but getting close, single mitochondria, you can actually image the spots with electron microscopy, and the resolution can be great, and then we take that and do mass spec. And if they're far enough apart, even with a 5 or 10 micron laser spot, you can still do very small objects. This is some of our data that was published in that Nature Methods paper. We get lipid data, we get peptide data, and we actually, within dense core vesicles in the atrial gland, we actually have 3 different vesicles that have different peptides. You can see them by separated by k-means clustering. Here's violin plots and saying that these peptides here, these are the mass to charge. They're not completely separate, but mostly you see certain peptides of cluster 2 or 3 or 1, and there's a little bit of bleed over. This is one of the first times I know of where you're actually seeing peptidergic vesicles from the same cell type are actually differentially packaged like this. The data is of such high quality that we're actually able to find an unassigned peptide, actually, that we couldn't identify. And we actually then, because the aplasia genome had been sequenced, we're able to pull this up from an unassigned section of DNA and say, oh, actually, it looks like a gene product, and not like that. It's a prohormone that we found in this case. And so we were able to discover a new prohormone that actually wasn't even known to be a protein. And so the data is of that quality. When we tried to publish this, they prove it works for different vesicles. So we did a poorer vesicle isolation. And then we actually got both 2 different types, electron-lucent vesicles and dense core vesicles. And this is just showing you differences. And you can see in gray and sort of this weird brownish purple color that the lipids are very distinct from these. We can tell them apart. And so we're getting lipid differences in vesicles from the same group of cells. If you're interested in single-cell measurements like this, Richard and the group published this DAT Sigma. It's a data-driven machine learning-based framework. You can use this to help guide your instrument. You can use it for the post-processing. When we put this up, we obviously know how to use it. We've had a few groups saying, could you explain a little bit more? We're happy to help you walk through this, but it really is bundling all the tools I just talked about for this single-cell data. I want to make sure I can get to everything. Now, it turns out I don't have to go into this much detail because this slide was introduced in the beginning. Thank you very much. This is sort of putting a lot of different things together. It's sort of a ridiculous slide, but we do single cell measurements. I didn't talk about our mass spec imaging because it's a little closer to what most people do. But we're working with an informatics specialist in a mass MRI expert, Van Lam. We combined everything. And so you can take a brain, you can serial section it, you can take one section and dissociate the cells, and you can do single cell measurements. You can take the next section and do mass spec imaging. We're using an ICR, which is slow, so we've already described how we can speed up the acquisition using lots of different approaches, the subsapes approach and then this Meister approach, and it can actually make things about 20-fold faster. What limits us here, I think, is sort of unusual. It's no longer the acquisition time of the instrument, but it's actually the stage movement time. I don't know why, but Bruker and others use fairly slow stages, and that becomes an appreciable amount of the overall acquisition time. But what we do then is we make a spectral library. We have individual points and we have the cells, and we can say what cells sort of make up that pixel. And so we can actually then say, well, this area seems to have things that weren't made up of cells. Maybe it's extracellular, for example, plaques or blood vessels or others. The other thing is, this is going to sound, my brain is a bar of butter. I don't know about yours, but lipids are the most common class. The dry weight is over 50 to 70% of the brain. And so when you slice it, you can distort it. And so what we're also doing is we're taking mass spec imaging data, but before we're doing that, we're taking an MRI atlas of the brain while it's intact in the skull. And then afterward, what we can do is we can take any distortions in our slices and fix them and anchor them to this gold standard of that particular animal. Now, in this case, we didn't quite do that. We used a gold standard from another animal, but we're actually getting this so it's all in the same animal. So we register it, we do mass spec imaging, we do single cells, and we put it all together to make a three-dimensional map and a single cell map. The most interesting thing about this in terms of data, and this is just showing you one set of the data, and it's too crowded, but for time reasons, I'll only show one thing, and that is we highlighted 6 specific regions, and we look at some of the lipids. And it turns out, just like we saw when Richard's data was, and we looked at our data, the lipids seem to give us a zip code in brain region. The lipids are different in the hippocampus, and they're different in the hypothalamus, they're different in the cerebral cortex. And so the lipids seem to be region specific. Now that may make sense a little bit. I can't, as opposed to cell specific. If you think about it, a cell can't make its lipids. It does some of it, but some of the fatty acids and the other precursors come from the liver. They come from the gut microbiome. They come from your diet. And if maybe you have the wrong fatty acids presence, maybe another one is substituted, and you can't tell that from transcriptomics, but it could easily be that depending on when certain cells are their lipids are being regenerated, that these change. Again, I want to also point out my neurons are over 60 years old. We'll just leave it at that. The lipids have to be replaced every month. Maybe even so a significant fraction of the ATP that my brain burns is not for learning and memory or behavior, it's actually replacing those lipids. And so cells last a long time, so the lipids are always being replaced, and that's maybe why you get these differential lipids in different brain regions. I don't have too much more time, but I wanted to end with one more example. This is some work on an Alzheimer's model. Now it's an extreme model. It's in a mouse, so it's a humanized, and it has 5 mutations to give a familial or an early onset Alzheimer's. And it means that mice get plaques very early. So if you want to study, you don't have to work on 1 to 2 year old animals with some of the more less robust, but the 5 fat animals. And then what we do is we can image the brain. We get our mass spec imaging. We actually can then do staining for the plaques using thioflavin S, and then we can then look at areas. We can co-register and then look at the areas in the plaques versus other areas, and we can do this regionally. And when we do that, we actually see both, which others have shown, plaque enriched and plaque depleted lipids. Because it's now we're using timsTOF flex with ion mobility, we can actually get isomers that are hard to differentiate. And the other thing we're doing, this is now using strictly imaging, but if you look at the plaques with single cells, you could actually consider this a like single cells maps. You could map 1,000 plaques and just acquire data on the plaques using our micro-MS approach. And so there's a lot of different things that can be done. And this is just showing you a little bit greater detail looking at plaque-associated lipids. And because we have MALDI2, because we have ion mobility, we're actually able to enhance the detection of quite a few of these. And we're actually seeing some subtle differences, some difficult to tell isomers. And so again, this is there's a paper on ChemArchive if you're interested, but this is some data that we're just starting. And now we're actually then going to be looking at some models that are a little bit less change, because again, if you go to this many plaques, then there's probably a lot of secondary changes in the brain, and we're not sure if it's just caused by the Alzheimer's or all the changes caused by the plaques damaging the brain region. And then the other thing, of course, is this is N of equal 3, and we need to increase the numbers to get more biological significance. So in this sort of whirlwind, I love answering questions, so I hope you have a few. In this whirlwind view of single cells to mass spec imaging, I just wanted to really highlight some of them. We talked about a fairly high throughput single cell mass spec approach, not counting the biology to make the experiment, not counting the deposition of the cells on the target and any of the multi-omics. And we have, if you look at some of our papers, have done this with Raman, with Rohit Bhargava and Paul Bone. We've done this with lots of different immunohistochemistry. We've done it with lots of different approaches. But if you don't count that, just the mass spec, it's about a second to cell. And so if you have 1,000 to 10,000 cells, it becomes a reasonable experiment. It's non-destructive. Now, of course, it's not. MALDI uses up some of the molecules, but we really can use multi-omics on the same slice. And that allows us to use even different types of approaches, like, as Peter talked about, liquid phase time of flight, that's a capillary electrophoresis, as well as gas phase time of flight. It does, when doing this, improves the robustness and completeness of the data for selected cells. The other thing I wanted to say is that, I really do like the enhanced image resolution using MRI on the exact same animal, because that really is helping some of the issues we've seen. And then we're making cell atlases where we're doing both mass spec imaging as well as single cells. And that work is really just beginning, but it gives us a lot of data on that. And then we're starting to try to understand the differences between what we see from a metabolomics point of view from a peptidomics and lipidomics point of view compared to the transcriptomics. And again, well, I absolutely acknowledge the incredible power of transcriptomics. The metabolomics really is telling you the cell state. It's telling you what the cell is doing, how it's interacting with the environment. So as much as you know, the transcriptomic data is useful, we really do need to have this molecular detail because it is one of the things that's telling us how the cells are actually interacting and working. And then the most important slide, and that is I get to sit here and talk to you. And yet I did very little of the research. This is just showing you the group through the ages that I talked about. This is one of the more recent talks. And then some of the models, you know, I didn't talk about most of them, but I think I can say the Sweetla group is really a zoo. In the last 15 years, we worked on animals ranging from comb jellies and jellyfish to sea slugs and echinoderms to a range of mammals and fish and others. And we do that because each one has a different type of biological driver that we can address. And the really wonderful thing is mass spectrometry is pretty ambivalent to the model system being used. And with that, I have time to answer any questions you have.
Thank you, Professor Zwider. That was amazing. Maybe I can start with Olga Chaim's question. Lipid differentiation comprehended per tissue area more than per cell type is a relevant concept. Would you understand its function on by composition while localized in among extracellular matrix components and or tissue areas with acellularity?
That's a great question. I don't understand. So, you know, so I don't understand something called cell state versus cell type. And what I'm going to first back up, you can imagine an astrocyte. People talk about lots of different types of astrocytes, but an astrocyte next to a dopamine cell upregulates the catecholamine processing enzymes of the transport. And so it's different than if it was next to, say, glutamate cells, where the glutamate cycle is upregulated. And so you could imagine the same astrocyte, which you would say was distinct type, is actually a cell state. It can change. And so now when you say lipid heterogeneity, is that a cell type difference or is it a cell state? It depends on which area of the brain it's in. And so, you know, I don't know the function. I don't know if it's if cell area is dictating because of the delivery of precursor molecules. If you think about why you need so many lipids in the brain, it's probably because of the range of shapes. You have, you know, fairly flat cell membranes from 20 micron or 10 micron cells, and you have tubules coming out that are, you know, submicron, and you need to have curvature. And then when you have curves that go this way as opposed to this way, convex or concave, if you're not going to have them leak calcium and other ions, you actually have to have very different head groups and tail groups. And so it may be, though, that of the 10,000 different lipids that are reported to be in the human brain, maybe you have a little bit of flexibility. You can grab one with a different set of tails and things. And so the differences really are depending on your diet and the liver function in that particular day when you're deciding to replace the lipids in your cerebral cortex. And maybe that, you know, and you do it not all the time, but you sort of do neighborhoods. Or it could be that it has to do with the extracellular matrix. I don't know of any papers, and I tried to look, that the extracellular matrix proteins are that distinct in, for example, the superoptic nucleus versus the suprachiasmatic nucleus or other areas of the hypothalamus? Is it distinct if you go to the different regions of the hippocampus, you know, the CA1 or some of the others? And so I don't know enough about that to answer that question, but it's a great question. And it's something that our group is turning more resources towards understanding. I will say the last thing is this is why cell culture doesn't work as well. Because when you culture the cell, you're tending to use a set of fatty acids and precursors, and that's, of course, what the cells can use, and they live. But inside the body, when you have an intact animal, then you're getting this heterogeneity caused by the rest of the bodies and the circadian rhythms all feeding into this heterogeneity. So in this case, you really do have to work in vivo or ex vivo model systems or freshly isolated.
Amazing. Before jumping to Professor Nim's question, I want to briefly acknowledge a couple of events organized by the Sphingolipid Biology, our partner. They are both posted in the chat, so feel free to check them out and reach out to us in case you have any question. So time for Professor Nim's question. He says, Jonathan, bravo on the nice work from your lab again. As you demonstrated, it is often just a few cells, maybe just one, in a population that exhibit a specific chemical profile. For example, peptide hormone in your case. In the spirit of reproducibility and rigor, I am wondering about how to address related questions. If an event is so rare that only one cell exhibits the event, I do not even understand the concept of reproducibility. In your view, what does reproducibility mean for one from an analytical point of view? Thank you for your insights.
So Peter asked the impossible question. So I'm going to answer this in one very strange Sweedler way, and then I'll get back to the science. I will never forget when Nature published all the rigor and reproducible standards about how you had to do things, and they're great. They also had a paper in that same issue about a group of ornithologists that discovered a new species of bird on Madagascar. And I say that because there was a picture of the bird and they had a feather and they did DNA and it was obviously a new bird. There was no two birds. They didn't say go out and catch 10. It was a new species. They had infinite data on that one. And so when you talk about rigor and reproducible for rare events, it's kind of a hard question. You know, if you think of astronomy, they talk about a supernova. They don't say, oh, I'm going to wait 150 years to publish the work until I see another one. So sometimes rare events, you don't have reproducibility. And that's the way science has always been. Now, I mean, it just is. And I'm going to say in analytical chemistry, somebody finds a dinosaur bone and does proteomics on it and gets 1,000 measurements on it, and they're able to see some proteins. You can't say, well, why not the next dinosaur bone? Maybe it didn't fall in the same mud pit. So rigor and reproducibility has to go hand in hand with the question being asked. With that said, one of the reasons I didn't publish that work already is we're trying to see more. And it turns out, unfortunately, we're seeing more of those. Actually, it's a really cool scientific finding of the oxyntomodulin in type 2 diabetes single cells from humans. The reason I say unfortunately is that these are isolated by a transplant specialist. You know, unfortunately, when somebody dies unexpectedly, they look at the islets. If they're high quality, then they isolate them and they do transplant, and we get some of the cells. If they have diabetes, they don't care. They don't want those. They're not going to transplant those into somebody. So 99% of the time when they find a diabetic human, they don't isolate the cells and we don't get them. So the problem is we have a lot of healthy human donor cells. We have very few islet samples. And so we need a few more to see. And we have now 5 or 6 examples, 5 or 6 people where we see an occasional cell. So it's starting to become a little bit better. But it is a hard thing. I'll give you another example that's more robust, but still very frustrating. Nitric oxide is 1 in 1,000 cells in your brain in some brain regions. Serotonin, 1 in 100. Serotonin, NO can react with serotonin to form nitrososerotonin. We're not going to do the number of cells required to see a statistically good number of those. And so if we can't label those cells, I'm not trying that experiment because it would be millions of cells required to do that. And so sometimes you're right. If you want reproducible results for measuring something like that, you really have to think, is this possible? So it goes from doable to what are you trying to show? And you're going to have to figure this out. It's a great question. Rare events are difficult.
All right, great question and great answer. Thanks again, Professor Sweedler. Now I guess it's time for Melanie Bailey.
Wonderful.
Please, Melanie, join us. And she's from the University of Surrey, from the Department of Chemistry. Thanks again for everyone. I think you're muted.
Hi, everybody. Thank you for the invitation to come and talk today. I am just trying to figure out how to share my screen with you. I'm having I'm sorry?
You are already sharing. We just don't see your PowerPoint yet.
No, I don't see it either. It's missing. One moment. Here. Right, do you see my PowerPoint now?
Yes.
Yes. Okay. Thank you. Thank you. I'm so sorry for that. I've my university seemed to strike me out of Zoom. So thank you so much for the previous speakers for moving their talks along so that we could accommodate this slight technical malfunction. So hi, my name is Professor Melanie Bailey. I'm from the University of Surrey in the UK, and I'm going to be talking to you about the seismic facility, which is a facility that we're setting up at Surrey for spatially resolved single and subcellular analysis. And what we're trying to establish is a facility that helps bioscientists from all over the UK to do single and subcellular analysis based on a new capillary sampling technique. So it's kind of a collaboration of people who work in omics, biologists with test applications, and then people who do capillary sampling and mass spectrometry. So what we have is we have a number of different pieces of instrumentation that you can use single cell analysis, but the subject of my talk today is about capillary sampling, and we have instrumentation from the Yokogawa Corporation, which has is sort of been touched on. We've seen Peter Nemes' lovely talk on using capillary sampling. This is actually a commercialized system which works under a confocal microscope, and everything is automated. So the idea is that you can go and select a cell under confocal microscopy and aspirate it into a capillary, so you can extract single cells retaining the neighbors, and you can extract subcellular compartments. This comes with a little bit of a health warning, so we can certainly extract parts of cells, but quite how spatially selective that extraction is up for discussion. But here you can see some sampling of nucleolus, and then on the right-hand side of the slide, you can see that we've done some extraction from the mitochondrial region of a cell. We've also been able to extract BCG from within the phagosome of the cell, and then on the right you can see selective extraction of individual stress granules. So what you have, we've kind of figured out how to extract cells and bits of cells, and then how to transfer the material. You've got a tiny quantity of material, which we're trying to very carefully transfer either onto a surface for analysis or into a vial. And because we can select and transfer this material, we've got the opportunity to analyze for all sorts of different analytes. So we've been looking for metabolites, elements, lipids, proteins. Of course, it would also be possible to look for transcripts and genes. Because this talk today is about metabolomics and we've only got limited time, I'm going to be focusing on lipidomics and a little bit on metabolomics. So once you have that material aspirated into your capillary, I guess you've got two options. You can actually spray it directly into the mass spectrometer through nanospray ionization. That's really fast. It's very sensitive, and it doesn't use so many consumables. But of course, you have no separation of analytes prior to analysis. So we've been looking at trying to transfer the cells or bits of cells into vials so that we can carry out liquid chromatography analysis. And part of the motivation for doing that is because there are automated processes. You can put your samples into your vials one by one and send them away to the LC-MS for automatic analysis. And the software for data analysis is much more mature. But as well as that, you've got the opportunity of seeing different isobaric species. So, for example, lipids, but also opens up the opportunity of doing some proteomics as well. So this is just showing the difference between a nanospray ionization method and an LCMS method. This is actually a helic chromatography method applied to single cells to look for amino acids. And basically what you can see on the left is that with nanospray ionization, which is in blue, we're getting much better sensitivity than with LCMS, as you might expect, because you're not diluting the sample so much, you're not having to transfer the sample from the capillary to the vial. But the nanospray really lacks precision. One of the issues that we have is trying to integrate an internal standard alongside the cell for analysis. So we get much higher relative standard deviations for our nanospray method. So we've been looking to see what we can do with LCMS, and we've developed a quantitative methodology, for example, for looking at drugs, doing a targeted analysis. So, you know, it's possible to quantify the level of picogram per cell of drug, for example. And when we actually did this analysis, we were looking at, you know, drug uptake in single cells. We were able to see cells that had high and low drug uptake. And then we just screened for differences in the data. And in the mass spectral data, the peaks that we were seeing that were different between high and low drug uptake corresponded to, or were identified, the features were identified as being lipids. So we thought, oh, single cell lipidomics. So we decided to see what we could do along those lines. So Kyle, one of my PhD students, has been working over the last couple of years to see what we can develop in terms of single cell lipidomics. So this is selecting a cell using capillary sampling, transferring the cell from the tip to an LCMS file, and then seeing how many lipids and what types of lipids we can see using mass spectrometry analysis. So we started off this work using an Orbitrap Q-Exactive Plus mass spectrometer, which is available in lots of different laboratories, where we're using analytical flow chromatography. And as you can see in the graph in the middle here, we can see sort of 400 or so features identified as lipids per single cell, but that's just based on MS1 spectra. We get very, very few features, as you can see in green, that we can actually identify using both the retention time, the MS1 and MS2 spectra. So analytical flow, it's quite difficult to get a very comprehensive assignment of our lipid features. Nonetheless, we are actually able to see differences, for example, in lipids between cells that have been drugged and are not drugged, for example, with chemotherapeutic drugs. And when we did that analysis, we were actually able to show that the lipid features that were dysregulated as a consequence of the drug exposure corresponded with what had been seen previously in literature. So moving on from that, we were able to scale down to microflow chromatography to increase our sensitivity. And we also used a Xenotoph mass spectrometer. And then we were able to develop a methodology that allowed us to do DDA lipidomics with MS2 spectra, and then we're actually able to get the fragmentation spectra from the lipids. So now we're actually able to identify lipids through their retention time, their accurate mass, and their MS2 spectra. And then moving on from there, we have been working with different vendors to see what the newest mass spectrometers and the sort of lowest flow rate, highest sensitivity chromatography methods available can actually achieve. And I guess perhaps unsurprisingly, if you scale down your flow rates, if you go to positive negative mode switching, or if you introduce ion mobility into your workflow, then you can see more and more lipids per single cell. So we're getting up to around 400 lipids per single cell with MST spectra and retention time. The other thing that we're doing is we're being quite restrictive about what we're calling a lipid. So if the lipid class isn't represented in our internal standard, which is EquiSplash, then we don't count it. So that's the way of sort of quantifying or qualifying lipids per single cell. So one of the questions that we have had about capillary sampling is the fact that we have a manual system as well as the automated system. And if you get the you can force the sampling to go wrong, I guess is the way to say this. So you can actually you can aspirate a lot of cell media at the same time as the cell. And of course, you can overcome this problem by resuspending the cells in PBS, but biologists will tell you that the cells don't really like to be in PBS. And so we've been looking to see how to perform the blank correction. You know, are we really measuring lipids in the cell, or actually are we taking a measurement of the local cell media? And I think actually, unless you apply a blank correction, your measurement probably is a combination of very local cell media plus cell. And for some applications, this might be interesting, but we've been looking to see how we can perform a blank correction. So what we've been doing is looking at where the meniscus is in our capillary tip, and then blank subtracting according to that, using blank samples. Now we've actually compared, using this methodology, we've compared a microfluidic sampling method to a capillary sampling method. And in the heat map on the right, what you can see is that first of all, our blanks and our cells are different, but also, remarkably, the capillary sampled cells give a very similar lipid profile to the microfluidic cells. And I think that's a really nice validation that we're measuring what we think we should be, that two completely different methods of blank correction will actually give us very similar lipid profiles. So I'm now going to go on to describe a very quick application. I know I don't have very long left for my talk, but I wanted to show you what we've done recently on looking at radiation-induced bystander effects, because again, I think this is a really nice validation of spatially resolved single-cell live cell lipidomics. Of course, one of the advantages of using this methodology is that you can sample the cells while they're in their living state. So we're part of an International Atomic Energy Agency program that are looking at methodologies for live cell irradiation and single cell analysis using accelerators and using synchrotrons. And so this work kind of falls under that umbrella. So what we've been doing is we've been looking at bystander effects in irradiated cells, because it's well known in radiation biology that if you irradiate an area of cells, actually the neighboring cells can also it can induce stress in the neighboring cells, and this has implications for the treatment of patients. Particularly when you're talking about sort of spatial fractionation radiotherapy. So we set up an experiment to see whether we could observe bystander effects in single cell lipid profiles. So what we did was we had a dish of cultured cells, we had a lead shielding on top of the dish, and we irradiated the cells through a pinhole. And we monitored lipid droplets using live cell imaging over time to pick a particular point in time at which we could see a divergence between control and irradiated cells. That was around 48 hours based on their lipid droplets. And then we sampled cells that were directly irradiated, their neighbours, and then cells from a control dish. And we actually split our cells into two different batches to try to have a go at an interlaboratory comparison. So half of the cells were analysed at Surrey and half of the cells were analysed at Thermo Fisher in San Jose. So what we did to prepare the cells for analysis is we actually freeze-dried them and shipped them at room temperature in vials, and then upon arrival, Thermo Fisher reconstituted them and ran them through a nanoflow LC-MS method. So we were actually able, both labs were able to see differences between irradiated and control cells. And I think what's particularly cool is that both labs saw very similar things. So we were seeing differences in arachidonic acid containing lipids. We were also able to see, or not able to see, so our bystander cells, so the ones that were next to the cells that were irradiated, but not irradiated themselves, also responded to the radiation. So the bystander and irradiated cells were completely indistinguishable. And this was completely corroborated by lipid droplet analysis that was performed on the same cells that we sampled, but also as a population. And so I think it's a nice validation, firstly, that of bystander effects in irradiated cells, because to my knowledge, this hasn't been looked at before. But it's also a validation that you can actually ship cells to another laboratory for analysis. So if you want to test the capability, for example, of your mass spectrometer to see if it's possible for you to perform single cell analysis, but you don't have the ability to isolate single cells yourselves, then we've been able to show that it's possible to do that. And it's also possible to sort of mix and match, I guess, cell sampling facilities and mass spectrometry facilities. So in conclusions, what we've been able to show is that LC-MS can reduce the variability for single cell analysis compared to nanospray. Certainly not faster, but future work, we're looking at how we can increase the throughput of measurements, and it can certainly help with peak assignment. We found that with appropriate blank correction, the capillary sampling and the microfluidics were giving very similar lipid profiles, which I think is a really nice validation of the single cell lipidomics work. And because we're looking at live cells and we can do confocal microscopy prior to picking the cells, we can perform lipid droplet analysis through live cell imaging before the single cell lipidomics. And what we've also been able to show is that we can ship single cell samples to other labs for analysis and still detect a biological response. So I'd like to thank all the contributors to this work from the various different institutes that we're collaborating with. I did want to put out a quick advert because we've got funding for 2 PhD places in our lab. So we're building the world's first MEV and KEV ion beam imaging system. So what we're doing is we're getting a water cluster SIMS, secondary ion mass spectrometry system from Ionoptica, and we're bolting it on the end of our accelerator. And the reason that we're doing that is because we think that we should be able to increase the coverage of analytes that we can see with mass spectrometry imaging, but also with MEV beams, you can focus them to 100 nanometers in spatial resolution. And so it's possible that we will be able to image metabolites in cell samples at very high resolution, and possibly even proteins. So that's what the PhDs are all about. And then finally, just a quick advert for analysts. I've recently taken over as editor-in-chief, and we would welcome any articles from the single cell community. And thank you to my team who did all of the work, and thank you very much for listening.
Thank you very much, Melanie. And our next speaker is going to be Elizabeth Newman. Elizabeth is a professor at the UC Davis University, and the floor is yours.
Hi, everyone. I'm going to take a second to share my screen and then hide the floating controls. Hopefully everyone can see me okay. and hear me okay. So I'm Elizabeth Newman. I'm at UC Davis. I'm really honored to present here. I was really excited about this opportunity to talk and show what some of the things my lab was working on. And so I'm primarily an imaging person, although we're trying to converge the imaging space with the single cell mass spectrometry and omics space. And so while I'm going to give some really discrete examples, what I'd like to really hammer in is that we are capable of analyzing many different kinds of samples, even things encased in bone. And so I'm an analytical chemist by training, and so I also like biology. And so what I think about at night is, how do you actually study biological systems? And usually people break this down into thinking about DNA, RNA, and proteins. But I know for the biochemistry people in the room, that's like really an very exaggerated and very simple look at biological systems. But I'm an analytical chemist, so I'm going to tack on metabolites on the end here because I've captured you all today. And out of politeness, you're going to hear me out that, in fact, metabolites is important. And while I know it's more complicated than this simple diagram, from an analytical chemistry perspective, I think about what tools do we have that we can use to study things like DNA and metabolites. And since my day job is really teaching undergraduates that really just want to know what's on the exam, here's a non-exhaustive list of what's on the exam at the end of my presentation today. But joking aside, if we think about things like DNA, we really have next generation sequencing, and it's very high throughput. Back in the day, we had some Sanger sequencing, and PCR is still a workhorse. But broadly speaking, we don't have many tools to actually study DNA, and we don't really need them. But as soon as we start going to things like proteins, we have things like LC-MS, spectroscopy, X-ray crystallography, and then we get to metabolites. And like the list just is huge. And this is actually really, really short. But even though you see things like LC-MS to study proteins and LC-MS to study metabolites, the methods to actually study both proteins and metabolites by LC-MS are very different, and in fact, usually require different kinds of PhDs. So what keeps me up at night is, how do you actually study all of these different omics spaces to understand biology? And so the question that becomes, and in the focus of my lab, is how can we measure this all, preferably all at once, all in the same tissue, and even all in the same instrument? And so I'm a fairly new PI at University of California, Davis. My office is on the 5th floor. Someone thought it was a joke to try to put a mass spectrometer on the 5th floor, but we broke the elevator, but it's successfully there. My first experiment was actually to knock out cabinets and make it actually convert from a rock-based lab to a bioanalytical lab. And here's a token picture of my office, because like many of you in the room, probably my parents didn't really understand what this job was until I told them that the office had a door on it. So I always include this in case anyone in the room doesn't understand what being a professor is all about. And so we use a Bruker timsTOF Flex. His name is Bruce. He's dressed like a shark. He's still dressed like a shark. In fact, he has braces because we had a high school student add to our costume decorations. And I'd like to, before I get into the science, really thank my group. Starting a new lab's not easy. Being the first graduate student in the lab's not easy. And then being the senior graduate student during your second year is not easy. So right now we have 9 graduate students. Some of them are here. We're about to take a new group picture. Yes, those are board games in the background. I'm a giant nerd. But really, all of the kudos and all of the effort that you see here is thanks to them, right? And so I just want you to remember their faces. They're in the top right corner of every slide. But I really, as a PI, really am thankful for the team that I work with. And it allows us to do really, really cool science, as I hope you'll appreciate. And so again, we use matrix-assisted laser desorption ionization mass spectrometry imaging. And you've probably already seen a slide about this, but I'm on West Coast time, so I've actually just recently joined. 5 a.m. my time was a little too early. But basically, we take a UV laser, we ablate a spot on the slide, and then we collect a max spectra. And we refer to this as a pixel. And this complexity, I've made a cute cartoon, but it's really in the hundreds of thousands of molecules. We can then raster this UV laser across the tissue, collecting mass spectra at each one of these pixels, allowing us to build a heat map of where these different molecules actually lie. And in our case, we couple this to other things like immunocytochemistry, immunohistochemistry, or transcriptomics to really get that single cell piece. So one of the first projects I did with my graduate student, Caitlin, is studied Alzheimer's disease in conjunction with Li Wei-Jin at UC Davis. He runs the Alzheimer's Center here. And so we know that diet is actually, in sex, are very important indicators of both disease severity as well as likelihood of developing disease. If we actually just look at a cross-section of a mouse brain with controlled diet and Western diet, you can think of like sugar and hamburgers, very American diet. Controlled diet would be like normal mouse chow, what the mouse is supposed to be eating. There's probably some fiber in there somewhere. But markedly, there's very little differences actually as a function of diet and even as a function of sex when you consider the entire brain at once. But you can see there's actually some like nominally some color differences here. So when we segmented out, say, the cortex from the corpus callosum and the hippocampus, the changes started, or the statistical changes started to emerge, except for in the hippocampus. Now, when you look at like greater literature, the hippocampus actually is very easy to induce change. Its function is on memory and has a play in cognition. So you would imagine that actually it would be very flexible. And when you feed an animal a new diet, it would actually change. But in fact, we found that that wasn't true in actually fairly large cohorts. And so one of our hypotheses is actually, if we go to the single cell level and start integrating cell type, are there cell types that are actually buffering these or other kinds of effects? So in fact, we did that, and we're actually using an Alzheimer's model here, 5xFAD, and we're integrating immunofluorescence, or I1AB, which stains microglia. And here we've zoomed in on what is a plaque structure. We've separately stained for plaques. We find an integration of these microglia at this plaque structure. And indeed, we can actually make out a high abundance of these plaques with PI384. So one of the questions that became emerging, both in the question of Alzheimer's disease, but later in the function of diet, is this PI384 related to the plaque structure itself, or could it be possibly related to these microglia and their accumulation at the plaque interface? And so what we've been doing is looking at plaques, and I'm showing an example here, but we're doing this for hundreds of plaques within murine samples, is we're actually looking at, say, the interface of the plaque that we've stained with a plaque stain, and then 10 microns out. So the next single cell layer out, and then the next one following that, and actually seeing how these molecules actually vary as a function of distance from this plaque. And then again, overlaid with the immunofluorescence, how this correlates with microglia infiltration of these plaques. So actually, this is our good friend PI384. It actually, we've seen with immunofluorescence, this is an accumulation of microglia at the plaque and at the plaque interface. So this PI384 is actually probably related to the microglia, not the plaque itself, which is interesting. And so what we're seeing is, as we modulate this as a function of diet, or even consider the function of sex, how do things like keto diet affect the abundance of this PI384? So keto diet, or high-fat diet, again, you need to have some fiber in there, but we're not drinking milkshakes or eating a bunch of candy. A keto diet actually reduces the plaque burden of the brain, and this is actually fairly well studied. But what's interesting, it actually reduces the level of PI384, which again is related to our microglia. And so what we're wondering and hypothesize for the next step is, does a keto diet actually also reduce activated microglia? And is this PI lipid actually related to that function? And then again, we're exploring this not only in mouse models, but in human AD. So here's an example of a phosphatidylserine that's actually very high abundance within human Alzheimer's samples compared to their healthy or without cognitive defect samples. We've shown that this lipid is actually fundamental in about 40 human samples now. And so we're trying to figure out how to design a mouse model that actually explores the function of this lipid in the context of Alzheimer's disease. And while I've shown you a lot of neuroscience, we also, again, can pivot to a lot of different organ systems with these kind of multi-omics approaches. And so Kyle, another talented student, and I have been working with Deanna Williams, who's an expert at HIV. Now, I'm very terrified of HIV, as most people are, and the idea of students working with HIV is actually something that would keep me legitimately up at night. And so we work with an analog, SIV, which infects macaque monkeys, but doesn't infect humans. And what this allows us to do is take a model that actually is pretty similar to humans, but study the effects of HIV on things like the kidney. We're also doing the brain, but for the sake of diversity, I'm going to show you kidney data today. So here, for the non-renal pathologists in the room, you have the cortex and the medulla and the glomeruli are lit up here. And so we have macaque monkeys that are uninfected. We've then extracted their kidneys, some that are HIV infected, and then some that are HIV infected, but actually treated with a retrovirus or an antiretroviral. And so what we're trying to understand is not only the effects of HIV on kidney health, but also do antiretrovirals actually completely restore function within the kidney when some evidence says they don't? And if it really doesn't, what is actually going on here? And so what we've done is actually done highly multiplex immunofluorescence to automatically segment our mass spectrometry data into the workhorses of the kidney, such as distal tubules, collecting ducts, and glomeruli. And you get a nice, pretty rainbow here. And we've segmented our cohort of 4 monkeys in each of these groups. I'm showing several here because it's otherwise too crowded. But what you can see in each of our tissue segments, we broadly have most of the different segments, allowing us to then extract the mass spectrometry after we've validated with immunofluorescence. And indeed, when we think about glomeruli, we can actually see there's an elevation of this lipid within glomeruli that are treated or infected with SIV, but this actually decreases upon an antiretroviral, but not to normal levels. And again, this is a composite of 4 monkeys per bar graph, and also hundreds of glomeruli per tissue section we're looking at. And then there's instances where this PA lipid actually increases upon treatment with an antiviral. And so some of these questions that emerge, are these artifactual, or how are the lipids interplaying with the efficacy of this antiretroviral? And so some of the next steps are actually, can we design these experiments and move them towards mouse models to explore knockouts? And then also add a transcriptomics piece. And I'll tell you a little towards the end how we're going to add that arm. The other 2 ends of my research is actually exploring things from very large to very small. Again, because these systems aren't isolated. And so while the other 2 examples, we're looking at the brain in isolation or the kidney, we're exploring, can we actually look at whole body systems all at once to see global effects with the when we introduce drugs, or in this other case, plastics. And so polystyrene's well known as styrofoam. And so you can think of styrofoam as something you might take on a camping trip. And so what's interesting, if we feed mouse chow that's been stored in a polystyrene container, we can actually detect the polystyrene within these whole body systems to see not only where is the polystyrene going, but how is it actually being modified to traffic through these different organ systems. So to orient you, we've cut the head off so it fits on the slide. Again, we're kind of ornery in that way. But this is the picture of the mouse with the head. And so your head is on this end, and then your tail would be on this end, where high abundance of this polystyrene peak, and at this point it's got 5 benzenes on it, so it's a 5-mer, is located primarily in the kidneys here and then in the liver. And what's interesting is as we explore different polystyrene lengths, we can see it organize into different organ systems, and we're also exploring the metabolomic efforts. And while whole organ systems are important, here's another example where we're using eye mobility to separate out the background from polystyrene, clean up our blanks, but you can actually see accumulation in even more organs as you, again, traverse these different lengths of polystyrene. But the other end of this is, while whole systems are important, we're also exploring these small nanobodies like EVs or extracellular vesicles within murine systems. And so EVs, for those unfamiliar, are ways of trafficking molecules from cell to cell. And where they actually are probably most important is in metastasis of cancer. So you have an original tumor, and then the tumor, to again spread, shoots out these EVs that contain things like mRNA or proteins or other things that contribute to metastasis of cancer. And so Randy Cardney and I were talking, he's an EV expert, with Caden, one of my students, and we were saying, fundamentally, a mass spectrometer should be sensitive enough to study these extracellular vesicles and at least the lipids in their membranes, although really it's a sampling issue. And so we've creatively developed a way of using electron microscopy to actually validate that there's a single EV within our sample. And again, this is based heavily on the work Jonathan Sweetler does. Again, he was my PI, and so I've borrowed some of the concepts I learned from him. And so we validated that there's single EVs within the pixels that we take from multi-mass spectrometry. And in fact, we get actually usually between 5 to 15 lipids from a single EV. And while this is interesting in and of itself from a biophysics standpoint, we wondered, do any of these lipids actually correlate to cancer? And can we actually figure that out? And so here on the top is a multi-mass spectrometry image of an ovarian cancer tissue. Here would be where the tumor is located. Again, it's quite robust. And then there's some microenvironment around the tumor, and then there's healthy ovarian tissue surrounding it. And so what we did was we took blood that was taken from this patient before their ovarian cancer was removed. We extracted the EVs and then performed this single EV analysis. And in fact, we found lipids that correlate both to the tumor as well as the tumor or the area around the tumor. And while these EVs may be 1 in 10,000 EVs, what's interesting from a diagnostic standpoint is we might be able to diagnose cancer and determine tissue of origin by drawing blood. And so far, this is validated with about 30 different patient samples where we're actually able to bin them into stage of ovarian cancer, as well as remove benign tumor cases, just based again on the EVs present in blood with these highly multi-omic experiments. And then finally, what I'll end the slide on in the last couple minutes is I've showed you a lot of cool cell typing, but what protein in the antibody work gets you is really just a cell name. Transcriptomics gives you cell function. And so Kyle, you've seen him on an earlier slide, is actually a classically trained organic chemist who has an interest in biology. So him and I were trying to think of a way to creatively assess gene expression without sequencing, such that it was actually scalable and much cheaper. We could actually go after all of these genes, again, in very large tissue sections. And so him and I are not that creative. And so we're like, you know, the original way of assessing gene expression is actually fluorescence in situ hybridization. But the key is, we wondered, instead of a fluorophore, could we add something like a MAPS tag? And in this case, it's a peptide. We've also played with like small molecule metabolites or lipids to, again, allow multi-omic measurements. We can then photolytically cleave these or cleave these based on pH changes. There's lots of different options. But once they're free, we can actually detect them by mass spectrometry, figuring out where these probes have actually been hybridized using very traditional ISH-based techniques. And our Gen 1 was actually very expensive. It wasn't much of a savings. It was $500 per probe for about 100 tissues, so like $5 per tissue. But still, you're not going to be able to look at 20,000 genes in one tissue section, which is our ultimate vision. And so him and I really thought, and I said, like, you know, part of the reason was we're actually leaving way too heavily on the biologists. Why don't we lean more heavily on the chemistry and think about how other chemists are handling similar problems? And by using different versions of click chemistry and like even metals, we've talked to an inorganic chemist, her name's Marie Heffern. We've got this down actually to $15 per gene for, again, 100 tissues. We're looking at cents to actually study where these genes are expressed. And here's a version of FOXP2. Again, it's in the cerebellum and then the hippocampus, a little bit in the olfactory bulb. But the benefits of actually doing this all in a mass spectrometer is we can actually get the localization of different genes, such as this for an oligodendrocyte, as well as lipids or other molecules, including, I don't show this data, but some signaling molecules and amino acids and such. Glutamate is another example. We can actually say, oh, my labels didn't show up. That's cool. This pixel has an abundance of this mRNA, and then here's its resulting metabolomics, allowing us to really go between these two spaces very fluidly, again, on one instrument for much cheaper than spatial transcriptomics allows you to do traditionally. And so with that, I'd like to thank our funding sources as well as our collaborators, many of which were featured here, but I didn't have time to talk about everything. I just mostly wanted to give a survey of some of the stuff our lab is working on. And again, I'd really like to thank the students who have really worked very, very hard to get a lot of this data and not only get the data, but also build a lab up from nothing with me. So with that, I'm happy to take any questions, although I went a couple minutes over. Thank you for your attention.
Thank you, Elizabeth. That was a lovely talk. Got maybe one question before we have to move on to the next speaker. So impressive image quality for teeny tiny differences. Were you and your colleagues able to observe macrophages in or around necrotic spots for hyperproliferative primary tumor areas or micrometastatic spotted areas?
That is something we're actually working on. So I didn't get to talk about this project, but we do more traditional cancer where we're actually looking at immune cell infiltration into these tumors and also how different genes actually interplay with that, along with things like diet. And so the answer is yes, and I can talk offline and share some data if that's of interest. But yeah, that's also an exciting direction. Immune cell infiltration is highly complicated.
Well, thank you so much. Thank you so much for the really lovely talk. And now let's move to a new format of presentation, which is 5 minutes speed talk from the speaker about the latest research in the field. And the first speakers would be Alina Segwat, a postdoc from Emma Longboard Lab from KTH Royal Institute.
Good afternoon. My name is Alina Sigaeva. I'm a postdoc at Emma Lundberg's group, and it is my absolute pleasure to be here today to talk about our research. Just like other speakers, we are interested in the complex and fascinating topic of cell metabolism, but we approach it from a bit of a different angle. We are a part of the Human Protein Atlas, which is a large initiative that was founded 20 years ago, and it aims to characterize the whole human proteome. We use highly validated antibodies to map the proteins on the subcellular level. We capture both the expression levels and the distribution of proteins within the cell. We can also get additional information such as native morphology of a specific cell or the number and properties of its neighbors. We have discovered that even genetically identical cells that are grown and imaged under the same conditions show variable expression of more than 2,000 proteins. 28% of those proteins correlate with the cell cycle, but 72% have unexplained variability. It appears in cell lines as seen on the left, tissue samples like in the center, and it also shows up in clonal experiments where one mother cell gives rise to a heterogeneous population, as shown on the right. A large portion of the unexplained variable proteins are metabolic enzymes, such as GAPDH, that is shown here. We currently work on dissecting the underlying mechanisms of it and understanding the biological consequences of single-cell variability. We have analyzed the co-expression of variable mitochondrial proteins, and as one would expect, the enzymes from the same metabolic pathway tend to correlate in their expression. Here is the example of IDH3A and DLST, and they are both parts of TCA cycle. Actually, a lot of the components of TCA cycle, and specifically those highlighted in blue on the right, correlate in expression on the single cell level with IDH3A, which we used as a marker. But this is not an absolute rule, and some enzymes within the same pathway don't really correlate well. At the same time, we see correlations in the expression of enzymes from different pathways. These findings highlight the complexity and interconnectedness of metabolic networks. To test the functional meaning of this variability, we looked at the expression of several thousand proteins that are
Sorry for the interruption.
on the single cell level with IDH3A, which we used as a marker. But this is not an absolute rule, and some enzymes within the same pathway don't really correlate well. At the same time, we see correlations in the expression of enzymes from different pathways. These findings highlight the complexity and interconnectedness of metabolic networks. To test the functional meaning of this variability, we looked at the expression of several thousand proteins that are known to be associated with redox metabolism, and we also recorded the levels of oxidative stress within the same cells. There are strong correlations between the oxidative stress and expression of known antioxidant enzymes within the same cell, but at the same time, other metabolic proteins, such as HADH and other enzymes from the beta oxidation pathway, also strongly correlate with ROS levels. It again highlights how intertwined cell metabolism is. Our hypothesis has been that this variability reflects the existence of distinct metabolic states within the same cell population. These metabolic states might not even be captured if we only do metabolic measurements. Imagine two cells. One of them produces low amounts of ROS and has low levels of antioxidant enzymes, while the other one has high antioxidant defenses and high levels of ROS. If we look at the net oxidative stress levels, they might be similar between the two cells, but the cell with high antioxidant capacity might easier adapt to additional oxidative stress. At the same time, the other cell might not be able to sustain additional stress and might die. If we want to correctly predict the response to, for example, therapy, we need to know both the levels of metabolites in a given cell and the whole underlying landscape of enzymes, such as enzyme activity, concentrations, and possibly even things like mRNA levels and the potential gene expression. The correlations that we find in our work suggest that we should also be looking at the interplay between different metabolic pathways and not just focus on one of them. This is a complex challenge, which is why we work on developing pipelines for multimodal measurements. We want to combine spatial proteomics with functional metabolic measurements on the single cell level and bring together different pieces of the puzzle. In our work so far, we show that imaging-based spatial proteomics can reliably capture single-cell variability, that correlation in expression reflects functional connections between proteins, that there is biological significance of the observed protein variability, and we propose that this variability needs to be mapped in space and time, as well as in relation to external stimuli and internal metabolic states of individual cells. This can only be achieved as a community effort, so I am very grateful for this opportunity to connect with all of you, and we're looking forward to the future developments in the field.
Cool. If you have any question, please feel free to put up in the Q&A button, and we will forward to the speaker. And the next speaker is Horace Chen, a PhD student from Rishan Gao's lab and from University of Illinois, Chicago.
Hello, everyone. My name is Horace. Thank you so much for providing me the opportunity to present today. And today I will talk about the technical details of gel-assisted mass spectrometry imaging, GAMSI, which is a sample preparation method that allows spatial lipidomics with enhanced spatial resolution. First, I would like to start with a short introduction to mass spectrometry imaging, or MSI. It is a label-free imaging approach that allows spatial investigation of biomolecules across the surface of an intact tissue. However, in comparison with other imaging modalities, such as fluorescence microscopy or electron microscopy, the spatial resolution of MSI, or especially multi-MSI, is limited to around 5 micrometer, which is larger than a single cell. Extensive instrument improvement have been done in the field to boost the spatial resolution of MSI to cellular or even subcellular scale. Here we present GAMSI as another direction to improve the spatial resolution by combining hydrogel-based tissue expansion. And this improvement in spatial resolution is solely achieved by sample preparation. Hence, no modification of the instruments or analysis pipeline is required. So here I would like to give a quick walkthrough on the GAMC protocol. So first we have the tissue and it will undergo a short PFA fixation step to enhance the structural integrity for the subsequent step. This fixation also maintains a lipidomic profile comparable to that of the fresh frozen sample. Next, it will undergo a molecular anchoring step in which we will introduce a small molecule called ACX to the samples. So this ACX molecule contains an NHS ester group which will react and covalently conjugate to the primary amine in the proteins. It also contains this carbon-carbon double bond that will participate in the polymerization step later. And then after that, we will incubate the tissue with the monomer solution. So this monomer solution contains sodium acrylate and acrylamide. And then after that, the sample will undergo in situ free radical chain growth polymerization, in which all the carbon-carbon double bond in the sample will start to polymerize together to form a polymer network. Since this small molecule linker also contains the carbon-carbon double bond, which means the tissue or the proteins will be embedded in this polymer network covalently as a result. On top of that, since the polymer network contains sodium acrylates, that will make this polymer swallowable in water. So after that, the sample will undergo proteolysis step to mechanically homogenize the structural integrity of the gel specimen so that it can expand evenly in water without any tears or wrinkles. We have optimized these steps for better lipid retention by using trypsin and surfactant-free environments. We hypothesize that lipids are retained indirectly through this process by the protein-lipid interaction or hydrophobic interactions. Although the protein-lipid interaction is affected due to trypsin digestions, part of the interaction is still retained due to the mild digestions. On the other hand, since we use a surfactant-free digestion condition, the hydrophobic interaction between lipids will also be largely retained. Next, we applied GAMC to a mouse cerebellum sagittal sections, as well as a fresh frozen sections as a control to showcase the improvement of spatial resolution by GAMC. As shown in this figure, it is important to note that this improvement of spatial resolution is corresponded to nearly an order increase in magnitude in terms of the pixel density. Next, using a smaller pixel size, we're able to observe the enrichment of the lipids and Purkinje cells at single cell resolution. So with that, I would like to conclude the presentation today that we established the general concept and protocol of GAMC. We also demonstrated the spatial resolution of multi-MSI can be improved by GAMC. We're also able to observe enrichment of lipids in Purkinje cells at single cell resolution. So with that, I would like to thank my PI, Dr. Gao, and my group for their support throughout this project. Dr. Gao is also giving a presentation later today, but he will focus more on the details and findings of the GAMC project. So please stay tuned to that. Last but not least, feel free to check out our protocol and the GAMC paper using the QR code here. And thank you all for your attention.
Okay.
Hello, everyone. My name is Horace.
Sorry. Now, let's welcome the final speaker, Laura Isabel Malcolm from San Diego State University.
Why does this hurt? It's pretty much what every patient says when they go to the doctor. It matters from their perspective, but it also matters from a clinician's perspective because it will shape how they treat a patient. A skin infection is treated very differently than liver disease. And it's also an important question from a fundamental point of view to understand why disease develops. And so a key question of my work is to understand what determines disease symptom localization. Sometimes it's easy. So for example, if you think about influenza, it's a respiratory virus. You find it at high levels in the lung. Patients develop respiratory symptoms. And in the same regions where you have lots of virus, you also have major impacts on tissue metabolism. But sometimes it's more complicated. So for example, in the case of the parasite Trypanosoma cruzi, which causes Chagas disease, patients will develop these very characteristic lesions at the bottom of the heart, in the colon, and then the esophagus. But by the time this damage develops, there's almost no parasites detected at these locations anymore. So what my lab does to address this is we build spatial maps of metabolism. So we will take organs, dissect them in a way that reflects organ physiology as well as disease processes. We build 3D maps that show the position of each of these segments in 3D space. And then from each segment, we extract metabolites. For each extract, we perform liquid chromatography tandem mass spectrometry. And then we rebuild the data back into 3D, which lets us visualize metabolite distribution and link that to aspects like tissue damage, pathogen load, local inflammation, etc. And so applying this technique in Chagas disease, what we found is that there is a disconnect between where the parasite is and where metabolism is changing. And we find most strikingly that the only spot with significant metabolite changes is the heart apex, so matching very nicely with where symptoms develop. If we look in the gastrointestinal tract, we're interested in understanding why symptoms develop in the esophagus and the colon, but not elsewhere. And we see a similar disconnect where we have lots of parasites everywhere in the acute stage, but significant metabolic changes only in the esophagus, large intestine, and colon. And then as we transition to the chronic stage, parasite numbers get controlled, small intestine goes back to normal. And so this can help explain why symptoms do not develop in the small intestine. On the other hand, even though parasite numbers are also being controlled in the esophagus and the colon, metabolism is getting worse at both of these locations. And so this can help explain why symptoms do develop at these locations. Now, at this point, you may be wondering if this is just something that's weird with Chagas disease. And actually, we've been studying multiple other conditions and finding very similar results. So here's an example from SARS-CoV-2 infection. And so this is also a respiratory pathogen. Patients will develop in some cases, these persistent lesions in the lung periphery. And what we find is greater metabolic impact of infection also in the periphery of the lungs. And that's not accompanied with a pattern of higher viral load at these sites. So matching with what we saw in Chagas disease. And so this work is possible thanks to the wonderful people in my group, our great collaborators, the funding agencies, and all the wonderful software that people have been developing that support this research. And with that, I look forward to answering your questions.
Well, thank you so much for all the speakers. And now it's my pleasure to introduce the next speaker, Professor Rachel Gao from University of Illinois, Chicago. And Professor Gao, please take your floor. Professor Gao, I think you're muted.
Yeah, thank you. Can you hear me?
Yes, perfect.
Great. Yeah, thank you so much for the organizer for having me here, and also for all the inspiring talks from the previous speakers. My name is Ray Gao. I'm assistant professor of chemistry and biological sciences at the University of Illinois, Chicago. Before I get started, I just want to show you one slide of our campus. On a nice day like this, if you're from the highest building from our campus, or about 100 meters above the campus, you'll be able to see actually Lake Michigan. Our campus is right next to the Chicago downtown. You can see also the beautiful skyline. So my lab here at UIC focuses on multiple fronts. One of our major efforts is to develop molecular imaging tools for spatial biology applications. And we use these developed tools to study neuronal functions as well as dysfunctions. And hopefully, in the next 10 or 20 minutes or so, I'll be able to convey some of the recent efforts related to spatial metabolomics methods we're developing. So one of the reasons we became interested in spatial metabolomics, and especially lipidomics, is in the context of brain function dysfunction. As we all know here, lipids are a chemically very diverse and complex group of metabolites that play a critical role in maintaining neural functions or causing dysfunctions. And from previous studies, we know that endogenous lipids show spatially distinctive distribution, dynamics, as well as metabolic pathway in the brain, which is a highly important and also spatially heterogeneous part of the central nervous system. So here, showing this slide are two phospholipids. As you can see here, the chemical difference is very subtle. And if you look at one of the side chains here, is whether it has this carbon-carbon double bond or not. And as we have seen, many of the imaging methods, including light microscopy and electron microscopy, historically struggled to molecularly identify and spatially map lipid molecules in biological specimens. And on the other end, the development in the mass spectrometry imaging has become a very powerful technique to study native lipidomic profile in biological specimens in a molecularly multiplex manner. So if you look at the spatial distribution of these two phospholipids, you'll be able to see using mass spectrometry imaging, there's distinctive distributions across mouse cerebellum. This is a tissue that's prepared from a section from the mouse. As you can see here, some of these lipids are distributed more densely in one part of the brain, whereas the other lipid has a distinct distribution from the first one. So I would say, perhaps without introduction, the MALDI-MSI is one of the very powerful tools within the MSI techniques. And using a mass spectrum imaging, we'll be able to actually get both the molecular as well as the spatial information. And here, the principle of mass spectrometry imaging is to collect mass spectrum from each pixel from across the cell or the tissue that is being affixed or immobilized on the surface. So one of the, I would say, limitations of the current mass spectrometry imaging methods, especially for MALDI-MSI, is spatial resolution. And earlier, one of my students, Horace, has presented this slide as well. And I believe this specs are slightly updated by now. But if you look at the spatial resolution of MALDI-MSI, it's typically in the range of 5 to 10 microns or above. And as we saw over the past number of talks as well as publications, there's a substantial advancement in instrument as well as the computational end to improve both the spatial resolution as well as the molecular resolution of MALDI-MSI. However, one of the efforts that we embarked on is to ask this question: can we actually enhance the spatial resolution of MALDI-MSI to the single cell or subcell level without modifying the existing mass spectrometry hardware or analysis pipeline? So in order to introduce the approach that we're taking on, first of all, I would like to introduce some of the limitations that currently sets the physical limitation of MALDI-MSI spatial resolution. And one of these limitations comes from the spot size as well as the stage and laser step size of the currently available mass spectrometers. In MALDI-MSI, as we know, it uses laser to basically ionize the ions that come from the sample. However, these lasers are typically in the UV regime, which is considered challenging from the optics point of view, to focus down to a very small spot. So in most of the commercially available MALDI-MSI or setups, the laser spot size is typically around 5 to 50 microns. And in conjunction with this spot size, the stage as well as the laser step size is also sort of confined in the range of 5 to 50 microns. Another aspect of this, actually the limitation comes from the matrix itself. So when we perform a multi-MSI, we have to apply this thin layer of organic molecules called matrix in order to help ionize, dissolve, and desorb and ionize the ions. However, when we apply this matrix, it forms these crystals of certain sizes. So if we use, for example, one of the commonly method called spraying, the crystals will actually come off as 5 to 10 microns. If we switch to sublimation, it generally provides finer crystals in the regime of sub-microns to a few microns, however, it's limited in the matrix selection. So this type of matrix application method also sets up a limit to the achievable spatial resolution for multi-MSI, which is also independent from the laser spot size and the stage precision issue I just mentioned. So given these limitations, we thought about what are some approaches that can really help us overcome this spatial resolution limit. So one of the methods that we use in my group is called expansion microscopy. And this is a technique to introduce a swellable polymer into cell or tissue samples, and then use this swelling property of the polymers to physically modify biological samples. And then the first actual introduction of this method was done back in 2015 by two brilliant grad students at the time from the Winning Group at MIT. And the idea behind this, again, as I mentioned, is to introduce this type of polymer. Here is a polyacrylamide sodium polyacrylate polymer, and basically form this polymer across intact specimen. In this case, shown here is a coronal slice of a mouse. And you can, as you can see here, is the pre and post expansion images, and you can see this physical magnification of the sample. I just also want to quickly show a time lapse. Some of you may have seen this time lapse before. This is hydro-embedded tissue that is going through this expansion process. It's basically, we're looking at it, this exchange of solvent from buffered solvent to water. So this process has been developed by many groups over the last couple of years. And the general principle or workflow is shown here. So you start with a fixed sample, can be a fixed cell or tissue. And then we apply basically this anchoring step to introduce a small molecular anchor to a subgroup of biomolecules of interest. And then, as I mentioned, we form this swellable polymer all across, both inside as well as outside of the tissue or the cell. And then the tissue or the cell goes through this step called homogenization. We also call it a digestion step, to basically kind of break it down so that it can actually go through the expansion in the next step. The last step is this expansion step by introducing water into the system. So we're exchanging out the buffer solution to water. And then this polymer, which is synthesized using monomers, can actually absorb water and then pull things apart at the molecular level to increase the physical size of the sample. Some of the basic specs of expansion microscopy: the expansion factor, this is the linear expansion factor, can be anywhere from 2 to 60-fold. It has been a range of chemistry has been developed on improving the expansion factor. The effective resolution. So here we're talking about the fluorescence microscopy resolution. So if we assume IB resolution about 300 nanometers, we're talking about 5 to 150 nanometers at the end of the expansion. The expansion isotropy, this is actually a really interesting aspect of this because people ask how isotropic this process is so that the spatial resolution, spatial information can be better preserved. This really depends on the hydrochemistry that we use. It's typically about 10 to 15 nanometers for radical chain growth polymerization, and it can actually be pushed under a 10 nanometer regime by using a different type of hydrochemistry. And overall, this type of approach is fairly cost-effective and can be highly accessible. It doesn't require, again, actually modifying the fluorescence microscope hardware or the computational pipeline. So I just also want to show an image from fluorescence microscopy. This is imaging using microscope called lattice light sheet microscope, which is also combined with expansion here. What we're seeing here is from a mouse primary somatosensory cortex, we're looking at a subset of pyramidal neurons and two synaptic markers here. And one of the caveats, or actually advantage of applying this expansion is its optical clearing capability. And what is interesting here is if you look at the expanded sample, pre-expansion as well as the post-expanded images of the same sample, you can see here that this piece of a brain slice becomes optically transparent. This is because, as I mentioned earlier, there's a step called homogenization or digestion step. In this step, we actively use proteolysis to break down proteins and at the same time to remove lipids using surfactants. So the sample expansion decreases optical scattering because it removes and dilutes of the scattering centers, in this case, mostly coming from endogenous lipids. So for us, one of the challenges of adopting this type of method to mass spectrometry, as well as the lipidomics, is to actually come up with an approach or method to preserve the lipids during this expansion process rather than removing it. So here's the basic workflow of GAMSI, or we call it gel-assisted mass spectrometry imaging. And earlier, you heard a short talk by Horace, and I would leave the details of each step to Horace's talk. I highly recommend his talk. But I just want to emphasize there are multiple steps that we really sort of optimize over the time that's actually going to be different from the conventional expansion microscopy workflow. So in GAMSI, the spatial resolution MSI is enhanced by polymerization and subsequent physical expansion of the sample, which is mediated by this reversible retention of the target analytes. In this case, we're interested in the lipids, so we're particularly actually interested in retaining as well as releasing these lipid molecules. As I mentioned, GAMSI does not require modifying existing mass spectrometer hardware or analysis pipeline. So it's basically, in principle, compatible with any mass spectrometer or actually MSI techniques. So the first experiment we did is to validate how much of the lipid is retained going across once the sample goes across this GAMSI workflow. So what we prepared here at the beginning is a fresh frozen brain slice, and then a slice that is processed using the GAMSI workflow. And we then applied this dye that stains actually pan-phospholipidly across the tissue. And then we characterize the fluorescence intensity per unit tissue area and basically compare how much phospholipid is retained before and after applying this GAMSI process. And here's the result from our validation. In short, actually what we saw is in a white matter, about 80% of the phospholipid are retained. In molecular layer, also 80%. In the granular layer, and these are different anatomical layers from the mouse cerebellum, we saw about 100, actually close to 100% of the phospholipids are retained. So given these results, we're becoming more confident that not only phospholipids, perhaps most of the other lipids can be retained during this process. And then we went on to do another validation, and this is related to the expansion isotropy that I mentioned earlier. This is to basically confirm how actually isotropic or well homogeneous this expansion process is. And in previous expansion microscopy techniques or publications, people have reported this sort of general area of expansion is about 1 to 5% in length. We also characterized the same sort of spatial errors before and after GAMSI. And then we found that the error is about 1 to 4%, which is comparable to the conventional or previously reported expansion microscopy techniques. So having these validations in hand, we decided to try this technique and see if it actually can indeed improve the spatial resolution on a mass spectrometry imaging setup. So the first mass spectrometer that we tested this is from one of close collaborators, Dr. Stephanie Colonna's lab. And what we basically did is we use a fresh frozen sample, and then we use a sample that is processed by GAMZ, and basically look at the spatial distribution as well as the qualitatively the spatial resolution of it before and after the process. And shown here, so these samples were expanded threefold. So basically it provides linearly a threefold increase in effective resolution. But also I just want to also emphasize this corresponds to ninefold increase in the pixel density. And perhaps you can see here qualitatively, we did see increase in the spatial resolution. And to our also, I guess, pleasant surprise, we did see the spatial distributions of each where most of the major lipid peaks have been preserved, or largely being preserved before and after the SCAMZ process. So here's another slide showing other lipid peaks. And again, the same spatial resolution improvement was observed across all the major lipid peaks. And then we decided to try other mass spectrometers, given that in principle, this method or approach should be applicable to any mass spectrometers, regardless of the hardware or the instruments or the computational pipeline. What you see here is using Bruker Flex multi-tissue typer. And you can see that before and after GAMZ, you can see a qualitatively an increase in the spatial resolution. And these samples were prepared using 4-fold expansion. So it corresponds to a 16-fold increase in the pixel density, and the effective pixel size here that is about 12.5 microns. And one thing I also want to point out is when we look at the biological insights, when we do this type of spatial characterizations of lipids, oftentimes that we think about the resolution on the molecular end, but also the spatial resolution is really important. If you look at this specific lipid peak that corresponds to one type of PE, you can see here in the fresh frozen sample, it seems to be co-localized or locally distributed in the white matter. But if you actually increase the spatial resolution, you can see actually this peak is not in the white matter, but it's in a different anatomical layer. Some of actually the important aspect of this process is how well it can actually preserve the lipidomic profile, especially this chemical breakdown before and after the process. For this purpose, we looked into the lipid peaks that were detected using the conventional gold standard fresh frozen sample versus GAMZ, and we created this pie chart to show the chemical breakdown of the detected lipids in the fresh frozen only sample and GAMZ only, as well as the lipid peaks that were detected commonly across the fresh frozen and GAMZ. So from this kind of characterizations, we basically saw that most of the lipids are preserved or retained in the GAMZ process, whereas there's indeed some sort of small changes in the breakdown in the compositions of the lipid that are detected. But also, again, as we explore this more, I think we're having more insights into what are more pronounced, what are actually less pronounced in terms of the lipidomic profiles that we can detect using GAMZ versus other type of sample preparation procedures. So what is the limits that we can actually push this technique to? So this is where we decided to go for higher expansion, as well as to push the pixel size or the raster distance to its limit. So these images were also taken using a commercially available instrument, Bruker Flex. And you can see here is we're using a 10 micron pixel size. And when we have the full 4-fold expansion, the effective pixel size becomes a 2.5 microns. So this is sort of barely at the borderline of single cell resolution, but you can see here, so these structures or morphology corresponds to individual cells called Purkinje cells. These are very large inhibitory neurons that you can find in a mouse cerebellum. You can see that we can start to see the lipidomic profile of individual cells within intact tissue sample. And then we decided to test on state-of-the-art mass spectrometers. So these images were taken using a Bruker-Timstorf. We only used a multi-1 mode in this case. What we did is to decrease the instrument pixel size from 20 microns to 5 microns and see how well actually we can image or get information from the tissue sample. So with fourfold expansion, the effective pixel size is about 1.3 to 5 microns in this case. And you can see here that we can indeed actually increase or decrease the pixel size down to 5 microns using this approach. We can also change the expansion factor. This is by, as I mentioned earlier, changing the polymer chemistry that we apply. So we can really push the expansion factor to even larger, so from 4-fold to 6-fold. And if you look at the effective pixel size, the 5 microns divided by 6, so we're talking about somewhere slightly below a micron in terms of pixel size. And what we saw here is, so this is at the borderline of white matter and these granular layer. And what you can see here is these distinct lipidomic profiles that corresponds to the nuclei of individual neurons or astrocytes within the white matter. So that's actually all the slides I have to just quickly summarize my talk. Unfortunately, I convey the message that lipids are chemically complex and diverse group of metabolites involved in multiple cellular and subcellular structures and processes essential to life, especially in the context of neural functions and dysfunctions. Mass spectrometry imaging is a powerful technique to study native lipidomic profile in intact biological specimens in a highly multiplex manner, but its spatial resolution limit often precluded from single cell and subcellular applications. Hence, we have developed this technique called GAMZ to overcome the spatial resolution limit of mass spectrometers. So with GAMZ, the spatial resolution model can be enhanced multiple fold to the sub-micrometer level without changing the existing mass spectrometry hardware or analysis pipeline. So what's next? So some of the things we're exploring is, of course, can we actually further increase the expansion factor? There's a range of chemistry that I mentioned that can really bring up the expansion factor to even larger than what we have demonstrated in the paper. And also at the same time to enhance lipid retention as we are hitting the sort of a detection limit or sensitivity limit as we dilute and expand these samples. Some of these methods or techniques have already been published. I just want to mention to you, if you're interested in learning more about this general principle of expansion microscopy, here are two. There are actually a lot more review papers that you can refer to. I also just want to bring to your attention the original publication of GAMZ can be found here. And also the protocol, recently we provided this protocol through protocols.io, so you can check out the protocols if you're interested in repeating or using this technique. Last but not least, I'd like to acknowledge the people that are involved in this project. Without my group and our collaborators, this was not possible. I just want to mention the big portion of my talk is based on my student Horace Chan's work, and also Maddie is also part of the subgroup that is working on this method. I would also like to acknowledge our close collaborator, Dr. Colonna, and her lab for co-developing this method, and also our colleagues from Bruker, and also the dedicated staff from IMSCERT from Northwestern University. And last but not least, I'd like to thank also colleagues and friends in the mass spectrometry community and many other labs, this space of combining mass spectrometry, imaging, and the expansion microscopy techniques. So thank you all.
Amazing. Thanks, Professor Gao. We have time only for one short question, one short answer. So it's from Olga Chen. Thanks for approaching the limitations. On regards to matrix crystal interference, could you comment whether this is biased for MZ data on glycoconjugates with sulfation content or other molecular post-translational differences? Any solution to suggest?
Yeah, that's a great question. So if I can repeat, the question is if can we actually preserve the glycosylated molecules? And in this context, do we have any bias towards preserving or removing some of these molecular modifications? I think that's a great question. We haven't really sort of closely characterized on glycosylated molecules at this point. There is, of course, concern. We're also interested in characterizing such molecular information. But if you actually look closely into the sort of the biochemical backend of this the method, many of the sort of the chemical process are sort of fairly gentle. So that's one of the design principles that we had when we designed this entire workflow. So I guess I'm hopeful in a way that many of these sort of the perturbations or chemical modifications are preserved. And I think there will be additional developments within the field to better preserve these modifications. And I think I just leave it there, a little bit open-ended, but hopefully now that's partially at least addresses the question.
Thank you so much. So now I am pleased to introduce the next speaker, Ryan Kelly, who is a professor at the Brigham Young University. Ryan, the stage is yours.
Great, thank you. Let's see. Hopefully you can hear me okay.
Yes, perfect.
Okay, great. So I do not do currently lipidomics or metabolomics. So hopefully some of what I talk about in regarding single cell proteomics will be of some interest to some of you. So one of the things that we have found is that making a method accessible is really important, right? So we originally were kind of building our own liquid handling robots to prepare single cells in really tiny volumes. Kind of the feedback was, well, that's great that you can do it. What about everybody else? So we've really worked on making our methods accessible to others, primarily relying on commercial equipment. So, and our workflow kind of looks like this. You need some way of getting a cell into a well. And now typically our well is just a 384 well plate, like a PCR plate, kind of small volume. And if you just use the very bottom of that 384 well plate, it's not different in terms of surface contact from like a microfabricated nano well. So we can get cells into a well using FACS or the Cell-in-1 or this Tecan Uno single cell dispenser. And then we dispense reagents, typically using that dispenser. We incubate to digest our proteins into peptides, lysing cells, all that takes place together. Then we subject that to a low flow separation and then followed by mass spectrometry analysis. So we have this recently published kind of guide for getting started with single cell proteomics. And one other thing that we recommend is that is starting with label-free single cell proteomics rather than kind of barcoded multiplexed analyses. And the reason for that is that the sample prep is a lot easier. We've seen over many publications that you can quantify more proteins in a label-free analysis than using a multiplexed workflow. Data analysis is pretty straightforward, and we're hoping to make the throughput fairly comparable, like the number of samples we can measure in a day. So our original workflow using these custom robots and microfabricated chips also used a number of steps, even to do a label-free analysis. You would have to add 3 or 4 additional steps for label-based workflow. So it was done in one pot to minimize losses from transferring one sample from one well to another. But it was multi-steps with incubations in between. So we had this goal to keep it one pot, but also make it one step accomplished in 1 hour and with the same kind of proteome coverage that we've had previously. And what we found was just using this high temperature protease. It's called it's from Promega and it's called rapid trypsin lyc. They've engineered that to be stable at 70 C. And so the kinetics are much better, and then it's cross-linked so it doesn't denature at these high temperatures. And we found just that protease alone or with some non-ionic surfactant that is DDM. It's commonly used for single-cell proteomics to assist with cell lysis and protein solubilization. Doing that, we can get to 1 hour. And then using the TCAN Uno for both optionally cell dispensing and then definitely for reagent dispensing, we can fill a 384 well plate worth of single cells in just a few minutes and add this one-step mixture, the rapid protease, and then within an hour we have our well plate full of cells ready to analyze. So measurement throughput is also really important, and especially for label-free single-cell proteomics in the early days, this was really pathetic. It was like manual sample injection. We didn't have auto samplers that were capable of handling single-cell size samples. So it ended up being, and the mass spectrometers were slower back then. So we were only analyzing 8 to 12 cells per day. So you can think of that as like a farmer harvesting wheat by hand, right? So then automation has started to kick in, and auto samplers were kind of developed or adapted to work with these small samples. And then also this latest generation of mass spectrometers like the timsTOF Ultra and the Orbitrap Astral mass spectrometers, they're just way faster. So we've started to see kind of 24 to 80 cells per day become more common. And you can see, so we're saying we're getting a little bit automated, like switching from hand harvesting to a tractor. And we're trying to push that further. I would say this kind of overstates the level of gains switching to a full combine. But what we're doing now with a 2-column LC system, it's not accessible yet, but we're working on that. But a 2-column LC system where each one runs a 5-minute gradient and they're staggered in time so we can analyze for 5 minutes with no gap in between. So that allows us to analyze 288 cells per day. And so this is just some of the geometry of this 2-column system, but we don't need to go into all of this, but basically we're pushing sample through one column while another sample is being transferred to a storage loop and another sample is being pulled in from a well. And so that just allows us to go one sample after another. And you can see because 288 samples per day allows us to analyze 2,000 samples in just 1 week. All right, so this is a lot slower than mass spec imaging that essentially can just zap a pixel across a tissue, but for LC-based analyses that can go really deep in an analysis, it's pretty fast. And you can see that here we're just on an LTQ to make sure the LC system is looking okay. We were just tracking a few peptides eluting with their retention times. And you can see we did have a couple of days where these columns are not temperature controlled, and the room cooled down a little bit too much overnight, a couple days, but the system recovered, and no harm done there. And you can see here, just analyzing a single cell sized amount of HeLa. For these quantitative experiments, we sometimes do a mixed proteome with some human, yeast, and E. coli all mixed together. And this is 200 picograms of HeLa and 308 picograms of sample overall. And if you add kind of the human A, yeast A, and bacteria B, or the B samples, we're getting over 5,000 proteins from these 3 species every 5 minutes using the Orbitrap Astral mass spectrometer with good coefficients of variation. And just the human part of that is about 3,400 proteins per single cell size sample. So just to give a feel for this, we've had different users. This is kind of our home-built data analysis program. And this is how many runs each user has performed since we've been tracking for a couple of years. And Chow, who's doing the 2-column 5-minute system, he's way past 12,000 samples now. And you can see this is how many runs per day. We were typically getting just a few dozen runs per day or something. But once we get on to the Orbitrap Astral with this 5-minute system, it's 250 to 300 samples per day. So we're pretty excited about the throughput. And here's just a month view where we had this 5-minute system running for part of July and August, and then again for part of September and October. So just within 26 days, spanning September and October, we analyzed about 6,200 samples on that 5-minute system. So we're pretty excited about this level of throughput for LCMS-based analyses. So just to wrap up, hopefully we've seen some advances in sample preparation, separations, mass spec acquisition, and data, let's say data management with this software that we're developing that should be published soon. Works well for single cells and other low input samples. Single cell proteomics is becoming increasingly robust, quantitative, and easy to perform. And this has actually improved now for single cells. We're quantifying over 3,000 proteins per actual single cell that we analyze now with this throughput. And we didn't talk too much about this MS Connect software, but those are the screenshots that we showed here. It can automate data and management and analysis. You can imagine if you're collecting, close to 10,000 samples, analyses in a month, you need software to help with the analysis. And just wanted to thank my wonderful research group and National Institutes of Health for funding, and be happy to answer any questions.
So it seems like we don't have any questions. So I think we'll move
Perfect.
on to the next speaker in the interest of time. Next speaker is Manuel Liebeke from the Max Planck Institute for Marine Biology and University of Kiel. The floor is yours.
Kelly, you need to. Sorry,
yeah,
to stop sharing.
There we are.
Okay, perfect. Okay. Now it should work and you should see my presentation. Okay, so was great. Thanks for the invitation, and it's a pleasure to now talk about microbial cells because that's what our lab is focusing on. So we are interested in host microbe interactions and how we can use mass spectrometry imaging to kind of look with a microscope into that interaction. And before I start with the science, I would like to thank the lab. These are the people who are doing the work. And in general, we are probing microbial, plant, and animal biochemistry using mass specs. And our device, which we have, is an atmospheric pressure multi-system, and we put in diverse samples directly from the environment. And I will come to those examples in a minute. So we are convinced in the lab that the chemical mediators, metabolites, play a really crucial role in interactions between microbes and higher organisms. They exchange metabolites with other microorganisms, with their host and with the environment. So they are really, metabolites are central for all those interactions from microbes with plants, microbes with animals, and humans. And we study those for a couple of reasons, because we think they play a role in communication, cooperation, or also chemical defense. And some examples from the lab in the past years, I brought them up to show you what we are approaching, just kind of as a teaser. We can have animals which are symbiotic or aposymbiotic, which means they have no symbionts available. We can use metabolomics to see those metabolic impacts on the host. Then if we move into imaging, we can go to organismal level and detect new metabolites in certain animal groups. And we also apply this to plant systems, where we look into the rhizosphere, where there's a high amount of microbial species making the chemistry, and we wanted to see how this is developing when we look into the root system. There are other ways where we looked into the toxin way of metabolites. There we collaborated with a lab, Timo Niedermeyer, where we detected a toxin on plant leaves in aquatic plants. And we had the topic today was an atlas. That's something we did for an animal, an earthworm, where we combined multimodal system to have a 3D worm, where we looked into microbes, parasitic interactions, and histology with many different modalities. And then also recently, when we kind of have a better understanding how we analyze our samples, we also can find novel biochemistry, where we found a new sterile pathway in animals in conjunction with the microbes. And when we talk about microbial systems, that's actually how they look like. And these are microscopy images from other labs, which I really enjoy looking at, because each color here, it's a one microbial species. And what we're using is, in many cases, 16S rRNA-based probes. So we have a probe which is fluorescently labeled that binds to the 16S sequence of the microbial species. And then we can do microscopy. And with this high resolution, we can start to see how microbial cells are inside, to how they are structured, how are the community structures. And this is a mouse tongue here from Jessica Mark-Welch's lab, where there's lots of structuring of certain microbes on this tissue surface. If we move to the gut system, the mouse gut, we have the epithelia here, the gut epithelia, and then it's a very mixed community in the mouse gut. And then we move to plant roots, where we're on the plant hair, we can see microbes sitting on top. So we have these bacterial communities actually on tissue surfaces. And the 16S method is a very, very good one, because we have the high resolution and very high specificity. And that helps us to look into this interaction between hosts and bacteria. And I will come in a minute to a system where we work on. This is also epithelial cells, but in this case, the bacteria are growing and dividing within epithelial cells. So these are muscle cells. And that helps us to see those bacterial communities in tissues, actually. And I took another example, again from Jessica Mark-Welch's group, which I find really fascinating, because here on this biofilm, oral biofilm, we have one organism kind of dominating this region here, and then it starts to interact with the green one on top. And if you look really close, you start to see that some of the green ones, they are surrounded by an orange bacterium. So there needs to be an interaction going on, making this structure happening. And we think that chemical cues are actually have a very important role shaping such spatial composition. And we want to understand what are these chemical cues. And the idea is to translate the landscape of microbes at the micron scale to a landscape of metabolites using mass spectrometry imaging. And I haven't put in a mass spec imaging slide here just to because I thought this is covered during today's talks. But I want to take you now into the deep sea. That's where we got some of the samples from. And what you see here in the shimmering water are deep sea mussels. and we are interested in their intracellular symbionts. And the question is now, why going to the deep sea and doing mass spec imaging? Here are these animals. This is a micro CT scan from our lab to see the histology. And if you zoom in into this system to better understand histology and how cells are kind of structured, you start to see these blobs, and each individual blob is a host cell. And if we cut through and use 16S RNA-based fluorescence in situ hybridization, we can visualize certain bacterial cells. And we know from metagenomics that this is a very low complex microbial community. And you can see that some of the host cells, they are filled just with the green bacterium, and others are filled with two with the green and the purple one, which are symbiotic microbes making a benefit for the muscle in the deep sea. And when we wanted to look with that with multi-imaging, this is spearheaded by Benedict Geyer and Patrick Bourseau from my group. We have a typical workflow from taking a sample, cryo embedded in carboxymethylcellulose, then we do cryosectioning, put the matrix on top with a spraying device, and do mass spec imaging. So then we get our first modality, we get the chemical landscape measured. And then the next step is we take this very same tissue section from that slide, we wash off the matrix, we do a post-fixation, and then we incubate with our probes, do the labeling, and can do microscopy. And from that very same section, we then get the spatial microbiome covered. And in one case, Benedict did this. He's now a postdoc in Stanford. Here we have these multitude of different metabolite maps from the tissue. We can put together like 3 different lipids, which then represent what we know the histology. Then we do the fish with, and we add also a Darby staining to show the nuclei, and then we have these 2 symbionts. And that is actually the very same tissue section again, where we have spatial information from the metabolites and also in a targeted way from single bacterial cells with a link to the genome. So we also know what metabolic capabilities are present in those microbial cells. And what I wanted to bring here, because this is kind of workshop-like and we saw many different methods, if you want to combine mass spec imaging with a different modality, with fish microscopy, for example, we really emphasize to test your laser system, so really check the laser intensity. And we did this here in our protocols paper, where we increased the intensity of the laser and the shot numbers, where we can see we get more metabolite signal, but on the other hand, we lose the signal from the fluorescence microscopy. And that we emphasize should be done for every system. And I'm talking about combinations of mass spectrometer and tissue type. And we did this also not only for our RP-SMALDI, but also for the newest Bruker-Timstorff device, where we then increased intensity of pixels, the shot per pixels, etc., to check on one example or on a couple of examples that microscopy is still going very well, and that you kind of fine-tune both modalities to a good outcome. Now back to the muscle system. Here I show you a zoom in of our epithelial cells, and we highlighted 3 different host cells here in white, which are colonized, and we show this with fish with that bacterium. And what we expected, because this is the same 16S, so one single bacterial strain, we expected that each host cell gives us the same signal. And when we looked into the mass spectrometry data, we actually saw that there are differences. We have the yellow and the blue compounds, which show really a heterogenetic pattern. And when we uploaded our data to Theodoros' tool, the Metaspace tool, we could also identify them. These are sterol-like compounds, hopanoids called. Very similar structure, those 2 compounds, and it was always believed that this is something bacterial cells express in a similar matter and no difference. And what we actually see is on this microscale space, which we have like a few hundred micrometer, the bacterial cells in tissues actually express a different sterile component, which makes us thinking maybe there's an oxygen gradient or other environmental gradients which lead to that change in chemistry of the symbionts. So that's not all the same, as really a heterogeneity of bacterial metabolism within a host. And to kind of follow up on those hopanoids, on these differences we saw, we also combined our spatial metabolomics with spatial transcriptomics. And what we did is we we first phenotyped our tissue, and here again we have in purple and in orange the phenotype of these sterols. So they are across a bigger tissue section, show also the diverse patterns. And what Dennis then did is he structured the data and made a kind of image analysis, created regions for dissection, and we went with that tissue section, which was under the MALDI, to the laser capture microdissection microscope, cut it out, many of these orange regions, selected them in one vial and the purple ones. And then we, with the help of Hara Gruber-Wodeczka, also in Kiel now, we did low input spatial transcriptomics. So these are now transcripts of the microbial cells, not transcripts of the host cell. And what I displayed here is the difference between those two regions. And these genes here, they're actually biosynthetic genes for these hoppanoids. So they differ in those regions, and now we can put those things together and think about which gene is doing this chemical modification. The Y is still under progress. And that this is working across the transcriptome shows this brief chart here of the top 25 genes. There's a quite good correlation from bulk analysis when we took a muscle, cryofixed it, and then did transcriptomics and compare this with the transcripts we received from tissue sections. Now going back to the image here from the muscle system, some of you may have realized there's a yellow dot here. So host cell filled with a yellow signal, and actually this is a parasitic bacterium, and we called it, or the group called it, Candidatus endonucleobacter, because Miguel, he published this year in Nature Microbiology, because they found out how this bacterium is actually growing in the nucleus, is growing their nucleus to a huge size, and we wanted to know, can we actually also see a metabolic signature of this intranuclear parasite? And we went to Klaus Dreisewert's lab, to the high-resolution T-MALDI system, brought a big tissue section towards the lab, and when Vincent kind of called me, said, oh, I can't see any signals, I'm actually not sure if there's an infection going on. And then we went back to the microscope and did spatial microbiome first to find those microbial cells. And in our lucky case, this individual was really kind of low infected. So he only found 3 host cells infected with a parasite, but then he took the neighboring section, flipped it, and could do a spatial metabolome with a 2 micrometer pixel size, showing different metabolites in high resolution. And I like to zoom this in, because what we have now is that very high spatial resolution. We have metabolites in microbiomes on the micron scale. And what we actually have is like this yellow signal here are specific lipids from those infected cells, and you see that it's still pixelated, but that also means each pixel is a separate measurement. So we have many measurements from a single host cell which is infected with that parasite. We get those phenotypes and can put them, once we identified the molecules, back together and just start to understand the metabolism in situ of those parasitic organisms, which we cannot culture at all, not the host and not the bacteria. So Morlan in the lab took this further. She now compared other animals, other deep sea mussels, which also got this infection, but a higher infection load. Here you can see with the fish signal that all this area, all these epithelial cells are positively infected with a parasite. And the same section went for MALDI imaging. And our workflow is then to process the mass spec data, process the microscopy data, and then define regions of interest. So at the end, Morlan came up with around 40 infected regions, 10 regions where we have beneficial symbionts sitting in, and then also we had a few cells, 10 uninfected regions. And then we can co-localize the mass spec data with the microscopy data to really go on single cell level and see the infection going on. If she then took this data from all these infected regions and symbiont regions, she identified also with LCMS from bulk data certain lipids which are significantly changed in those infected regions, and going always back to the image, because that's our proof where things happening, that those lipids are also abundant in the infected regions. Those ones are look to us like they are everywhere present where we have bacterial infection. So these are the regions with beneficial microbes, and these are the regions with a parasite. So this seems to be either a response from the host towards all bacteria, or these are bacterial lipids just from bacteria in general. Now, can we apply our methods from the deep sea also to more complex microbiomes? And this is here again the deep sea muscle epithelia filled with microbial cells. And next to it is actually a mouse intestine. Also, kind of the size and arrangement is very similar. So we moved on now with a new lab at the Kiel University with a mouse system where we want to understand what is actually the metabolic route through a mouse gut with the microbes and the metabolites together. And a very nice system is if you kind of cut out the ileum or the colon, you kind of make a Swiss roll, that's how it's called, cut through it, and what you get is actually a cross-section through the whole ileum or colon. You can do histology and also the imaging and kind of get a look across that whole metabolic route through the gut. And when Morlan did this now for the colon, we can spatially map in also in high resolution images from mice, which were kind of has this pathogen-free microbiome. And then we also compare this to germ-free mice, where we kind of have very similar pattern because these are metabolic signals for certain gland cells, for mucosa and for the submucosa, not yet differences in the microbial metabolites. That's where we kind of right now are working on. But what we can do once we have this gut roll looked at with the MALDI, we can also do the fish microscopy. Here we used a EUB probe, so a very general eubacteria probe. And then we can, as similar as I showed you for the deep sea mussel, go in, take a single pixel and kind of retrieve certain, what we think are bacterial lipids, have this distribution and can combine this with the tissue and cell distribution and the fish signal. And that's actually our future plan where we want to fingerprint or metabolic fingerprint hundreds of different gut microbial species, get their MALDI fingerprint to go back to our MALDI data of gut systems, gut tissues, and say, can we use that as a guidance for where are certain bacterial cells? And this is working like a biomarker way we showed together with Strittmatter in Munich with DAISY MSI, where this is actually working for microbes in tissue. And with that, I'm at the end. And what I'm kind of hope I could show you today is that we can, if we have spatial metabolome and the spatial microbiome from the same tissue section, and maybe combine it with transcriptomics, we get an insight into the metabolic interactions when we have host and bacteria sitting next to each other or nested within each other to get really insight to phenotypes. And with that, I would like to thank again my group and our collaboration partners and you for your attention.
Thank you very much, Manuel, for the fantastic talk. Unfortunately, we don't have time for question. Now it's my turn to introduce Marlene Knobloch, which is a professor in the University of UNIL. Please, Marlene, the floor is yours.
Can you hear me and you see my slides?
Yes.
Okay, perfect. So thank you very much for the organizers for this fantastic meeting. I'm coming a bit from a different angle, but I hope I can convince you why we should look at lipid droplets in the brain. Now I don't see my... why we should look at lipid droplets in the brain and why they are probably more important than previously thought. So our model systems are neural stem cells, and they form the brain from the early development on, and they can also reside in the adulthood and form actually continuously new neurons. And I have been interested for a very long time in the question how metabolism influences NSPC behavior. And we have been focusing also on lipid metabolism and could show that both the de novo lipogenesis and the breakdown of lipids by fatty acid beta oxidation is actually a crucial regulator of these neural stem cell states and also how they behave. And when I started my own lab, we got really interested in these storage organelles, the lipid droplets, and this was work pioneered by two PhD students, Margim Ramosai and Sophia Madsen. And as you can see here on this scheme, this is really, they are really at the center place of these lipid metabolic pathways. So de novo lipogenesis can feed into this, and they can be a reservoir for fatty acid beta oxidation. So what are lipid droplets? They are the cellular lipid storage organelles, and they are best known from adipocytes, but they are also abundant in other tissue involved in lipid metabolism. And in principle, every cell can form lipid droplets, but not so many cells do. And these lipid droplets are having a neutral lipid core, triacylglyceride rich and cholesterol ester rich. Then they have a phospholipid monolayer, and I will come back to this. And they are heavily coated with so-called aldehcode proteins. And lipid droplets have many functions, not only energy storage, but they are also protective organelles against lipotoxicity and many other functions. So when we studied lipid droplets in neural stem cells, as you can see here in primary cells isolated from the adult mouse brain, these are really very abundant organelles, and you can also see that they are coming in various shapes. And I will not have time to go into this, but if you're interested, this is published work. We could show that these organelles can directly influence their cell metabolism and actually their proliferative capacity. So they are really crucial organelles for these neural stem cells. But what we also realized is that these droplets differ a lot depending on the state of the cells. So here you see proliferating stem cells, here you see quiescent cells, and you can appreciate that the area changes a lot and also the way they look. And when we differentiate these stem cells into astrocytes and neurons, which occurs spontaneously, you can also see that they are very different. So astrocytes have a lot of small droplets, whereas neurons have almost no or very few droplets. So the question comes, of course, do they have the same role and function in these different cell types? And do they maybe influence these cells? And to do this, we are actually very far from single cell approaches yet, but maybe we will get there. So we established a protocol to extract these lipid droplets. This is the project of Diana Panfilova and was initiated by Mergim Ramazai, where we actually use stepwise ultracentrifugation approaches, and we get really extremely pure fractions, but we lose a lot of these droplets on the way. So we are actually starting with 100 million cells per replicate. And what Diana did, she used proliferating NSPCs, and then we have a way to make them quiescent, and we induce differentiation into astrocytes, and we had like 4 biological replicates per cell type. And then we did lipidomics and proteomics on these isolated lipid droplets and asked whether they are actually different between these different cell types. And to make a story short that is ongoing and is very exciting is when we look at the lipidome of these lipid droplets from different cell sites or at the proteome of these lipid droplets, you can see here very nicely that the cell types separate very clearly the 4 replicates that were done independently. They separate very clearly based on the content. So these lipid droplets seem to be really cell type specific compositions. And we found many interesting candidates, especially on the proteome, but also on the lipidome, that might actually influence the cellular state, and we are following this up. But lipid droplets are very dynamic organelles, and we wanted to have a tool to study them in a more dynamic way, in a staining-free manner. And there had been no actually reporter line for the mammalian system. So Sophia Madsen did this novel endogenous ALDI reporter mouse line, where we actually used CRISPR-Cas9 to endogenously tag one of the most ubiquitous lipid droplet coat protein called perilipin-2 or plin-2. So we added a TD tomato with a linker in the endogenous locus under the endogenous promoter, and which results in all lipid droplets that express plin-2, they will be fluorescently labeled. And this is happening in all tissues that express plin-2. And this is actually a huge advantage because now you don't have to do any staining or any visualization. And we are interested in the brain, and we actually developed this tool to look at neural stem cells and lipid droplets. But then when we looked at unstained sections of 2-month-old healthy mice of this strain, we were really surprised how many lipid droplets are actually in the brain. And you can see this here. This is the endogenous fluorescence of this reporter line. And if you zoom in into a region as the lateral wall, you can see these beautiful ring-like structures that we can now visualize. And when we look at many different cell types, we can actually see that these organelles are really in all kind of flavors in these different cell types. And this came a bit against the dogma because people have claimed that lipid droplets are not present in the healthy brain and they will only appear with disease. So one of the big advantage of this mouse model is actually that we can now unbiasedly separate cells based on their lipid droplet content. And I want to emphasize here that this is not based on any expression level on a genetic level, but this is really the presence and the accumulation of these organelles. And because we tagged it with a red fluorophore, we can actually cross it to various GFP reporter lines. And we did so with the astrocytic line, neuronal line, and neurostem cell line. And we can use flow cytometry to get a quantitative view who and how many of the cells have LDs. But we can also use this in a more unbiased way and sort the cells for downstream application. And we did this, so we made single cell suspensions and then used fluorescent sorting, where we could say, does a cell has enough detectable lipid droplets? Yes, it is a positive cell, or has it a negative signal, and then it's a negative cell. And then we did this with 10x genomics and processed with single cell RNA sequencing. And I have to show you here, we focused on microglia. This was actually due to a technical problem. So we definitely have to optimize the isolation protocol to get all the cells covered. But for the microglia, we had by far the best reads. And when we did unsupervised clustering, we had like 3 nice clusters shown here. And when we then overlaid the information, whether it was originally a positive or a negative cell for this accumulation of lipid droplets, we found very interestingly this one cluster here that was almost uniquely composed of cells, of microglia that were having lipid droplets. This is also reflected mostly on the expression level, but again, I want to emphasize that gene expression, especially when it comes to lipid accumulation, is not necessarily the best readout. So this is really on a level above. So we wanted to compare this cluster 2 with this cluster 1 that has relatively little cells with lipid droplets. And I will not go into this in details, but you can see that we found a very different profile of this cluster 2 cells, as you can also see here in the volcano plot. And this is actually something that has been becoming a very hot topic about lipid droplet accumulation in disease. And there has been this description of microglia that are lipid droplets laden. They are called LDEM microglia, but also in other diseases that lipid droplets accumulate. So we compared the signature with this established signature, and we could see that indeed this cluster 2 we found in the 2-month-old healthy mouse brain actually shared quite some signature with these diseased signatures, but also some of the homeostatic genes were still upregulated. So we think that already at 2 months, we might have a subpopulation of cells that shares disease-like signature, but is still not yet in a diseased state. So we were really excited about this, and we are, of course, following this up in more details. So why do we see what others have not seen? You could say, okay, this might be an artifact of the mouse line. That was also the reviewer's comment. Or you can see it more positively. You can say we have a technical advantage because we actually don't need to stain. And I said this before, the phospholipid monolayer makes these organelles really fragile. So if people come with classical ways, detergents, they are actually destroying these structures relatively easily. And you can use lipophilic dyes trying to reveal the core, or you can actually use antibodies against these LD code proteins. And I don't have time here, but we spend a lot of time, especially Alicia Ray and Francesco Petrelli in my lab, to prove that what we see is real. And we managed, and this is now on bioRxiv and on the revision, we managed to optimize a method to now visualize these lipid droplets in wild-type brains. And we see that these lipid droplets are indeed very abundant in the healthy brain. And what we also found, and I think this is very intriguing, is that when we now wait 2 years, these are still healthy mice, 2-year-old TD Tomplin 2 mice, we see this massive accumulation of lipid droplets compared to 2 months. You can see this here quantified. And this is probably also why people are able to detect with classical methods lipid accumulation in the aged brain, because it's so massive, but they have underestimated completely to which extent this accumulation is so massive. And just to convince you, this is actually a wild type brain with our optimized protocol, where we can now also reveal these lipid accumulation in very similar extents. So which cells accumulate lipid droplets over time? So I showed you we did this single cell sequencing before, but we had problems to really see all the cell types. Now we have a new approach. We will actually use this flex approach, that it's now possible to use fixed tissue to begin with. So we have now a very nice cohort of mice, 2-month-old, 1-year, and 2-year-old mice, and we have males and females because we are also interested whether there is any sex difference in this accumulation of lipids. And we will use this fixed approach where we can actually directly fix the brain right after isolation, and then make single cell suspension and sort the fixed cells based on LD content. And then we have all these downstream options to actually characterize who is accumulating these droplets over the time and how does the profile change of these cells. And we have started a collaboration with Giovanni's lab, where we will do multi-imaging of these old brains and try to address what is the lipid droplet content. And of course, what would be beautiful is to match it afterwards with imaging technique, where we can visualize these lipid droplets. And just for the very shortly, we also see that lipid droplets accumulate highly or are highly abundant during brain development. Here you see the ventricular zone of the developing cortex with all these lipid droplets. And we are establishing a lipid droplet atlas over developmental time and regions. This is the project of Marie Eaglebuscher in my lab, where we have now collected at all the different time points embryos from this reporter line. And just to show you here, and I will not go into details, this beautiful accumulation of lipid droplets. And you can appreciate it's very different in different regions, and it also varies a lot over time. And we are, of course, very interested to combine this with multi-imaging or with single-cell sequencing to try and understand what are these droplets doing during brain development, and why is there such a huge difference in accumulation. And with this, I'm coming to the acknowledgement. Of course, my lab, I mentioned the people on the way of my presentation, and also the former members, and of course, all the collaborators and the funding agencies. And with this, I'm happy to take questions.
Thanks, Marlon, for the beautiful talk. Unfortunately, we do not have time for questions. And it is my pleasure to introduce the next speaker, pioneer in spatial and single-cell metabolomics, Fyodor Alexandrov, now at UCSD, together with his PhD student, Bishoy Badi. Thanks, Fyodor and Bishoy, for joining us, and the floor is yours.
Do you see my screen?
Yes, we do.
Okay, great. Thank you, everyone, so much for organizing this great event. I think it's actually definitely like our immersion field needs more of this, and I very much like how it was organized and particularly by graduate students. And thank you so much for inviting us to present it. From our team, we'll present two of us. I will present the introduction. Afterwards, our senior PhD student, Bishoy Badi, will present some of his results as well. So our presentation today will be on spatial and single-cell metabolomics. We'll try to cover both aspects. And so we recently moved to University of California, San Diego, where we're now based at Departments of Pharmacology and Bioengineering. And it was amazing 10 years at Amble previously, but this year, me and 6 of our team members, we moved to UCSD School of Medicine, where we'll still be developing technologies for spatial and single-cell metabolomics. pretty much the same focus using imaging mass spec as foundational technology, both spatial and single cell omics. And as always, what we do is with some link and focus on big data and software. And our lab manager, Shabraz, software developer, Metaspace, Lucas, Gianni, who's now one of our key data scientists, Louisa, expert in immunology, and two graduate students, Sharat, working on single cell multi-omics, and Bishoy, who works on lots of things that he'll present later today. So before we kick off with our presentation, I want to put it a little bit into the broader context. And so if we go to PubMed and we pull number of publications for single cell metabolomics, we'll see some numbers, and we start compare this to single cell transcriptomics. We see that, so single cell metabolomics, I mean, that field that here we're talking a lot about is where single cell RNA-seq was 10 years ago, 2014. And if you remember, like if you were already back in science back then and you remember what's going on, so single cell RNA-seq was about 2 years before big papers, before it was on the covers of Science and Cell and other journals. It was really this exciting time where everyone understood that, so the single cell biology and single cell technologies are coming, and afterwards they deliver it. So I think that we are right now pretty much in this same time with the single cell metabolomics as well. Another interesting aspect also to reflect here is, so if we look at number of publications, these are probably some of those that you know very well. Jonathan Swidler and Renato Zenobi, they were one of the pioneers, and they published papers in very leading journals back then already. This was an original, this was a review. So another question, like, what was preventing the whole field from growth? And so I do think my personal opinion is that this is sensitivity of mass spectrometry, but I would be very interested to hear your reflections on this. So, but now we see that it's so amazing to see so many new groups come into this area with absolutely commercial, not custom-made setups, and it really warrants the growth of the field. If you're interested to follow the growth of this field, then I also invite you to check out and join our conference, which organized by a new foreign society of international single cell mass spec. So we did already 2 editions in the US and Denmark, and next one will be in the fall of the coming year. We still need to decide where it will be. So now moving towards our presentation. So for us, enabling technology for spatial and single cell metabolomics is multi-imaging spectrometry. Here is the scientific officer placing this tissue section to imaging mass spec setup that we have. And you probably all know here very well that imaging mass spec, in particular multi-imaging mass spec, became the major technology for spatial metabolomics. Around 2011 or something, like we quantified this, and it was clear already back then. But of course, like the properties of the technology and capacities improved tremendously since then. So now the question is, like, will multi-imaging become the major technology also for single cell metabolomics? I think this yet to be, like, remain to be seen, but it has pretty good chance for it. So we started working in this field, in particular spatial metabolomics, with software tools, and one of our key software tools is Metaspace. And Metaspace is software for metabolite identification. This is how we started, but now it can do much more. You can do data QC, visualization, analysis, sharing. And also what's very important is it became knowledge base of spatial metabolomes. And Bishoy Vadi, once again, our senior PhD student in our team and one of key data developers and scientists for Metaspace, will present on this later. But what's cool is that right now we have a lot of data there, we have a lot of users, and finally we started having strong scientific impact with more than 500 publications, and most of them, they were in the last 4 years. So we have more than 100 publications of users, Metaspace users per year. In particular, because this is a very collaborative platform and it's free, open source, there is a lot of open data, and we also contribute to several NIH atlases. So this is how we started. But when we joined Amble, this was one of our first projects, but there was one elephant in the room that really kept us a little bit questioning. And in particular, the field as well in technology, and that elephant in the room is which metabolites are detectable by multi-imaging mass spectrometry. And we tried to use metaspace and all this big data and a lot of metabolites on the data there to learn about this, but definitely wanted to have a bit more quantitative, a bit more ground truth about this. So, and why is the elephant in the room? Because if I ask you specifically which metabolites are detectable, whether that metabolite is detectable or not, so it will be very hard for you to answer this because there are no theoretical models and the data is very limited, and even collection of data sets are very, very limited. So this, I would like to present the project where our PhD student, radio student Veronica, teamed up with mass spectrometrist Mons and with software developers Lucas and Lachlan to answer specifically this question. And I'm showing it as the first part of our presentation because I think it's very important for anyone doing multi-imaging and imaging mass spectrometry as well. So this work is preprinted on bioRxiv and currently under revision. What Veronica did here, she, together with Mons and others, they created standard samples with biologically relevant metabolites. So this is single pure metabolites spotted on glass slide. They developed setup. They published also the whole kind of like 3D like blueprints, how to do this. Anyone who is interested can reproduce it, information about solubility, solvents, and so on. But long story short, they created more than 200 slides where 172 metabolites were spotted on every slide. And metabolites shipped with quite a lot of effort so that they will be representative of molecular classes, metabolic pathways. And once we have this standard samples, which was a lot of effort to put, but once we have it, now finally we can get ground truth and objective quantitative information about the detectability of that metabolite by MALDI, by a particular technology, or by a particular protocol. And this is what we did. And I'll show you just one excerpt from this big paper. So where we did 24 protocols in our lab, 12 matrices, 2 polarities. And on top of this, there was tremendous effort of complementing this with interlab validation by 10 labs using 12 protocols. But now what we see is that, look, so what we show here is that here are the different matrices. Do you see my pointer? No? Okay, so probably here you see my pointer. So here you see different matrices. Here you see different metabolic pathways. This is just an excerpt from the whole data. But it's illustrative why, because so the dot here, it shows, with the color, it shows intensity of the metabolite. What will be intensity of your metabolic class if you put it in MALDI? And this fraction of compound detected per class. So this is like the whole class was detected very well, and this maybe not so much. This maybe not so much. And if we now compare this, we see that for fatty oxidation, positive mode is much better, and those matrices will be your best bet. But it's not always the case. For example, here, if we want to detect citric acid cycle, you actually can do it with a positive mode. You need to go to negative mode. But the good news here that if you want to do primary metabolism profiling like citric TCA cycle glycolysis, you can do it with the same matrices. And now you can infer all this information. You can do it with a user-friendly web application. If you go to Metaspace 2020.eu, detectability, and you can follow all these steps, then you can create these plots, and you can pick for any class of your interest or metabolite of your interest. You can get so much information about which protocol to use, what is normal ionization pathway, which are adducts, or so sometimes modification of the molecules that can be used to detect this molecule. So finally, I think we made the first step towards really addressing this long-standing problem, which metabolites are detectable. And the good news is that almost all of them are detectable. This is what we show. Yet they require specific protocols. Now we put up this as a resource and web app where anyone can go and find the best protocol for their metabolites of interest. So this was the first part. How can we understand detectability? Now let me move on more towards like the of interest of this workshop is single-cell metabolomics, and in particular high-throughput small molecule single-cell metabolomics. Because several years ago, we developed a method called SpaceM. And this method stands for Spatial Single-Cell Metabolomics, was developed by first PhD student in our team, Luca Rapes, who published this in Nature Methods, and it was so great to see actually his recent preprint on this super resolution that he's really like developing things in this area. So he is extremely bright and talented. And and really bursting with this idea. So what he developed is the method for single-cell metabolomics, where the cells are plated on glass. Microscopy is done to find where the cells are, to phenotype them. And multi-imaging is done with identification with metaspace to find these pixelated metabolite images, locally develop the way how to overlay these types of data with subcellular precision. And now, past normalization and single to pixel to cell deconvolution, we can finally have single cell information. So we published it back then, and we showed it that it can be used in particular for NAFLD and NASH to really find the differences between induced models of homeostatic states, NAFLD hepatocytes, NASH hepatocytes, and also reveal metabolic mechanism of action of anti-inflammatory molecules, which inhibits NF-kappa B. So all great. I mean, this was really great result for us. It was really like so cool to see how these pixels, they correlate with the cells. However, we had a few key challenges back then. And some of them, they overlap with what was discussed previously. What are the key challenges in spatial metabolomics as well? So batch effects was mentioned. So for us specifically, the challenges were poor detection of small molecules. That's why most of this 700 metabolites and lipids were lipids. Throughput, because it took us like more than a month to really like run it and even more to process it, and variability from one experiment to another. And we worked over the past 5 years to really change this. And now we have a new method, which is called high-throughput SpaceM. And we just preprinted this a bit earlier this year. And now we can put a lot of samples on one slide. And this is more of 5 times increase of throughput. Finally, at last, we have protocol for cell prep and for small molecule metabolites. And this is also supported with custom glass slides, because you see we also etch the areas here and batch processing software. And finally, at last, we have the method which can detect small molecules. Here, I'll just demonstrate you for a model system of HeLa and NH monocultured. We have a bunch of replicates here to study variability. And we were detecting 135 metabolite ions. This is only M plus H. And if we look where the metabolites from these ions are, we see that they cover pretty much all metabolic classes: amino acids, lipid-like molecules, carbohydrates, nucleosides, purine, pyrimidines, and so on and so forth. So this is great. And on top of this, they also cover pretty much all metabolic pathways of interest: amino acid metabolism, glycine, serine, histidine, arginine, purine metabolism, pentose phosphate pathway, TCA, glycolysis, and so on and so forth. So finally, we have a method to reprobe metabolism and not only lipid metabolism of the cells. And this is what we did. And here I show you just a cancer case study where we considered 9 cell lines and HeLa. So we can find now cell type specific markers. And it's very interesting to see that sometimes these cell type specific markers, they express only small subpopulations, less than 20% of cells, which shows the advantage of using single cell metabolomics. And on top of this, not only we can do cell type specific markers, but we can also use the unique capacity of single cell metabolomics, namely calculate the metabolite abundance across physiologically heterogeneous cell populations. And this is what you can't really do with bulk. I mean, to do perturbations for this, but then you will be limited towards the scope of your perturbations. So, but here every cell is different, and we can find metabolites which really go hand in hand between each other. For example, here adenine and guanine. And surprisingly, this co-abundance is pretty common, even above the 0.8% correlation. But there is also an inversely of co-abundance. And now we can finally build these single cell co-abundance metabolite networks. And why is it useful? Because so we can use them for many different things. But what we are looking now into, how can we infer metabolites which are important for this network, so-called metabolic hubs? And how, knowing that metabolic hubs are not metabolic markers, how can we use them for target identification drug discovery? And with this, I would like to wrap up. I would like my part of the presentation. Bishoy, our senior PhD student, will present the next one. But I still would like to thank the whole team who joined us, who moved to UCSD, our collaborators, in particular here in our lab detectability studies. More than 10 labs contributed to this, and selected alumni. And with this, I'm passing over to Bishoy.
Yep. Thanks a lot to you. Always interesting to see the presentation. I always learn something new. Okay, let me share my screen. Can you see my screen? Okay.
Yes, no.
Right. So I'll present a bit more about the Metaspace ML and beyond, and what can you do with Metaspace and integration with other omics. But as Theo explained, Metaspace is the main platform that we currently use for metabolite annotation, and it acts really as a huge knowledge base. There's a lot of insights and information from different data sets covering different species, different tissues, animals and plants, and it's really a huge community. And maybe a bit of like shameless promotion here. I would like to basically encourage everyone who is interested to use Metaspace to engage in the community that we have. We recently have this announcement on GitHub where we have Q&A. If you have feedback, if there's an annotation that you think is incorrect, or there's something that you would like to see change, please join on the main discussion, and let's talk and help build a Metaspace community together. So Metaspace mainly lies in a long long workflow of metabolomics data analysis, and it mainly lies in the molecular identification, and there are many different approaches. And then you can take these annotations and do different types of validations like bulk, LCMS, and targeted as well. But one thing we noticed with Metaspace when I joined and started working on Metaspace ML is that even though that some instruments have incredible capabilities in terms of resolution and resolving power, we sometimes get underwhelming annotations, like number of annotations is really low. So this was the motivation to start working on a new model or a new annotation pipeline, where basically when you have your data set, you can use different features as already was available in the Metaspace model as a rule-based approach. So these features include like spatial correlation with isotopes, information on the image in terms of the spatial localization of the ions throughout the tissue, and correlation between theoretical and observed spectral intensities, and many more features. This model was able to learn using three to five different features, actually mostly those 5 different features, to help separate between target and decoy ions. This is the main approach to help annotate more low FDR-based annotations, basically. And the beauty of this model is that it was trained on multiple data sets. It's highly contextualizable. You can customize it for different contexts regarding species or tissue of origin, and even protocol. And this was trained on thousands of data sets and was evaluated rigorously with other testing data sets. So this is already is available on Nature Communications as of a couple months ago. And if check it out, and if you have any questions, I'd be happy to talk later. So the main thing is now, how can we take this further after Metaspace? So you have your own data sets, you get your annotations. Let's try to see how we can take this into the single cell and spatial realm. So for this, we have another tool to convert the Metaspace datasets into common objects that are available in the SCVerse ecosystem, mostly like ScanPy for single cell analysis or spatial data frameworks. And then you can use the annotations from Metaspace into different objects so that you can follow along with single cell and spatial analysis pipelines. And on top of this, we also have another package, an enrichment package, to perform single cell and spatial metabolomics enrichment that take into account the ambiguity of the annotations in terms of isomers and isobars. So just to summarize, so now that we have spatial metabolomics data sets are getting increasingly produced, and they are highly sparse and noisy, and we can only annotate 10% of MSI data, and that becomes more challenging for small molecules and lipids, as Theo has alluded to earlier. Metaspace offers more of a knowledge base for building and customizing ML models. And the Metaspace ML model was much higher and much more precise than its predecessor in terms of better precision, throughput, can detect low intensity and biologically relevant metabolites. And now with the Metaspace converter, you can take your data sets from Metaspace and start building or running single cell pipelines. So for the next part, I would like to talk more about how we can combine spatial metabolomics with spatial transcriptomics. That also aligns with the theme of this day. So why do we need to actually integrate metabolomics with transcriptomics? Transcriptomics has always been the gold standard for cell type annotation, but it's really hard to get more information about the metabolic states just from transcriptomic information. You can indirectly infer it, but it's unable to delineate this metabolic states. On the other hand, spatial and single-cell metabolomics can provide more precise characterization of metabolic states, yet cell type annotation is limited unless you have it a priori, as Theo mentioned in the other HT-SpaceM project. So what I worked on is mainly a alignment of spatial metabolomics to SM-FISH spatial transcriptomics data. So the data basically, I have mouse brain data sets that are adjacent sections, so 10 micrometers apart. One section goes for multi-imaging, and another section goes for spatial transcriptomics, which is an imaging-based single molecule FISH. So we perform metaspace metabolite identification on the multi-imaging sections, and then use the spatial transcriptomics to perform cell segmentation, and then having the information from metaspace with the ion images and the cell segmentation based on the transcriptomics, we can now register or align the images together and try to deconvolve the metabolite intensities from the ion images into the cells based on spatial transcriptomics. And since this is brain images, we can also align them or register them with the Allen Brain Atlas to get more information about anatomical regions so that we can study this even more in a region-specific manner. So by doing this, we end up with paired metabolite and gene counts data sets that now we can use more downstream function analysis. And just to give you an idea of what possible analysis could be done, you can, now that you have paired data sets, you can investigate metabolite RNA correlations. You can do cell type annotation. You can project the cell types into the metabolic space. You can understand spatial marker interactions. There are different methods that are already developed and out there to probe spatial transcriptomics, and it's about time now that we can integrate the metabolomics and start investigating how they work together. So just to show you an example of some of the results. So here, for example, this is the Allen Brain Atlas anatomical region on top of the cell mask based on transcriptomics. So now we know which cells are in which regions. And with the spatial data framework, now you can appreciate how we can combine different types of elements based on the data that we have. So here I'm showing a spatial data objects where you have the images in terms of atlas section, the bright field MALDI images, the DAPI images, and the ion images. And then you have labels in terms of the cell mask and ions. You have polygons if you want to study, for example, the composition of cells and atlas regions. So you have the atlas annotations, you have the cell polygons, and you have the ion images, pixels, and on top of that, you have the end data objects providing, for example, the gene counts and the ions. And what spatial data does is it's trying to align everything into one coordinate system. So here you have the metabolites is a one coordinate system because the ion images have different spatial resolution than the RNA. You have the RNA with its own coordinate system, and then you can align both of them given the type of transformation that you already did. And spatial data allows you to merge all of these elements into one object and make it possible to query different type of transformations and operations. So to give you a bit of a glimpse about the results and what they look like, we were able to see spatial localization of cell types. That has been already done before, so that is not new. But when we look at the metabotypes or the metabolic the cell population defined on the metabolic space, we can also see a really nice spatial localization as well. And if you look at top markers compared to the gene expression, we can also see really nice metabolite gene correlations. And you can really appreciate how striking the correlation is, or how positive the correlation is. And this is based on single cell. So it will probably get much higher if you constrain this into a region-specific manner. So if you focus on a specific region and try to get metabolite RNA correlations. And now you can define better region-specific and cell type-specific metabolic populations. On top of this, we can take this a step further and try to explore marker interactions. So what this means is that now we have a new tool called MISTY, and what MISTY does is try to take a specific region or an area in the tissue and start to explore or ask the question, can gene expression explain metabolite abundance and vice versa? So that's the predictor and target, and in different spatial views. So there's the intrinsics, if you only look at the cell and intracellular expression only. There is the juxta view or local view. So that means you take a radius, or you draw a radius, and then you try to investigate marker interactions within that specific radius. There is a para view, so that's much further apart effects between metabolites and RNA, and you can add other views as well. So we performed this, and what we can get, sorry if the font is not clear, but what you can see here is that if we consider gene expression as a target, and we ask what is the effect of metabolite abundance on gene expression in different views. So, and what is the contribution of these different views? So intracellular versus local neighborhood versus para, and so on. And one thing you can focus on, so for example, here I'm showing the effect of gene expression on metabolites on the x-axis, and this is the para view, so meaning that there are more spatial distance apart. And if we look at these correlations, you can now zoom into this kind of contributions and find out which genes affect the metabolite abundance and gene expression. And this will be synonymous a bit with the metabolite correlation, but this is more spatially aware as opposed to the metabolite RNA correlation. So in summary, we have a workflow to align spatial and single-cell transcriptomics, through which we could map the metabolites to the same single cells. We could align those cells to Allen Brain Atlas to enable more fine-grained investigation of the region-specific cell types and metabolic states. We can detect spatially localized domains of both modalities. And then we also found high concordance between transcriptionally identified cell type and metabolic cellular populations. And this is just scratching the surface. This is just the beginning. There's still more to come, but for the sake of time, I could only share just a glimpse about it. But happy to talk more if you're interested. Yeah, finally, I would like to thank everyone who was involved in the spatial multi-omics project, mainly Theo and Charles who prepared the data, Mario and Yaron from Resolve who did the experiment, Jan who provided the data sets, and the group of Julio C. Rodriguez and Oliver Stegley for spatial data and MISTY analysis. And thank you for your attention.
Thank you very much for your talk. Very interesting. So we have I think time for two questions. One is about the HuBMAP that you showed. Actually, at the beginning you showed some HuBMAP of the single cell, and it really showed the different clusters very clearly. And then you show the HuBMAP in the tissue section after multi. So I saw that the HuBMAP are not very distinct. Do you explain how do you explain these differences?
What do you mean by the HuBMAP are not very distinct?
The fact that the clusters are not so far apart in the HuBMAP compared to the single cell.
Yes.
I find it very interesting. Yeah.
Of course, yeah. So on the single cell metabolomics space, the feature space or the metabolites are much more similar than the genes, for example. So if you take at the transcript level, there are distinct markers like for cell types. While in metabolomics, there are more similarities between the metabolites, so you get less variability. And that's why you see that the cells or the clusters are a bit closer to each other compared to the single cell transcriptomics. Yet these still are unsupervised clustering. So as you see with the single cell metabolomics, based on how you tweak the clustering, this is how you get different. And you can get higher resolution, more resolution based on how you tweak. But the reason why they're close together is because the metabolites are more similar to each other than genes.
Okay, thanks.
Yeah.
I think we have time for another question.
The second question, thank you, Bishoy, for the talk again, is about neurotransmitters. So which neurotransmitters are you detecting in this study? And are they patterned in a way that's reasonable compared to the cell types that you're co-measuring? And maybe also how about neuropeptides and correlation between neurotransmitter-related genes and the actual measurement of neurotransmitters, if any?
That's a good question. So I haven't looked exactly into which neurotransmitter I've been detecting. So I'm assuming since we have excitatory and inhibitory neurons, there is a GABA-based GABAergic transmission, but I haven't really looked into the neurotransmitters. So this is something I can look into. And with regards to neuropeptides, so I don't think we don't measure here neuropeptides, but maybe when we align with other data sets, we can comment on that. But otherwise, with just this data set, you don't get as much with neuropeptides.
Thank you, Bishoy. So our next speaker is Lauren Saunders from Heidelberg University. The floor is yours.
Great. Thank you.
Yes, we can hear you.
Okay. Then, can you see my slides?
Yes.
Okay. Perfect. All right. So first, I want to thank the organizers for putting together a really fantastic lineup of talks today. I've really learned a lot. And my name is Lauren Saunders. I am a new junior professor at the Center for Organismal Studies at Heidelberg University here in Germany, where it's evening time. And maybe on the first half of the topic today, my background is really in single-cell transcriptomics, and there will be a little bit of lipidomics at the end. So I'm excited to show you some of that new data. So bringing us back to why single-cell transcriptome measurements are so powerful is because they offer so much more than the transcriptome alone, right? So by measuring gene expression or mRNA in single cells, you can actually measure a whole bunch of different things: transcriptional complexity, cell states, cell cycle status, cell composition, and tissue interactions. And this is so valuable because it spans multiple scales of biology. So by measuring molecules, we get information about cells and organisms. And I want to highlight from some of my work and a lot of new methods to quantify individuals that if we take this cake and turn it into a bunch of cupcakes, we actually get a lot more. And that is by individual sample barcoding. And one of the powerful things this allows us to do is to quantify differences in cell abundance changes in response to environmental, genetic, any kind of perturbations. So as a postdoc in Cole Trapnell's lab, I wanted to be able to do these kinds of measurements with single-cell RNA sequencing, specifically studying cell fate decisions in zebrafish embryogenesis in response to genetic perturbations of key transcription factors. And by measuring dynamics over time and genetic perturbations with single-cell, this is a lot, but the goal that I had was to really scale up to lots of individual animals. And the technologies were really coming together at the time. Now, this we've recently just published last year, so I'll show you how we do this. And the first thing we do is we take individual samples, put them in 96-well plates, dissociate them to cells and then nuclei, and then here's where the sample barcoding comes in. So we can take single-stranded hash oligos, DNA oligos, put them in the wells, fix them in place in the nuclei, and these oligos with poly A tails are capturable with our single-cell transcriptomics profiling. In between that, we can pool them, put them in the freezer. And the method for single-cell RNA sequencing that we use was developed in the Shinduri and Trapnell labs in the University of Washington. And this is a really scalable method that doesn't rely on droplets, but actually uses combinatorial indexing to profile the transcriptomes. So at the end, we have our single nuclei transcriptomes and can match that back to the individual sample embryo of origin and be able to quantify differences not only in gene expression, but in cell abundances. So as a new lab, some of the new technological advances and power of this method is really important. And that's because we have unlimited sample barcoding. We can fix these nuclei and store them long-term, which enables really complex experimental designs. There is no really specialized equipment or reagents that we need. And with the newest protocol that was published in 2022 from the Shinduri lab, the cells are about 1 cent each, which is 10 to 20 times cheaper than 10x. So using this method, the first thing we did was to look at wild-type zebrafish development across 19 time points spanning organogenesis. And this atlas encompasses 1.25 million cells from 1,200 individually barcoded embryos across all of these time points. So once we had this, the goal was to really compare how genetic perturbations impact cellular development. And so this is our perturbation atlas. Really briefly, it includes these 81 different samples. On the y-axis are the genetic perturbations. Most of these are transcription factors that affect some cell lineage development. And at each one of these dots, we had 8 replicates. And this really is the thing that empowered us to quantify the cell abundance changes if we transform the gene expression matrix a little bit. So usually with single-cell transcriptomics, you have gene expression matrices, right, where cells are columns and genes are rows, and the values represent the counts. But as soon as you add a sample barcoding, you can now transform these into cell composition matrices, where columns are embryos, rows are cell types, and the numbers correspond to the number of each one of those cell types. And that allows for a different comparison at the whole organism level. So for example, you can look at the cell abundances in wild type embryos compared to perturbations to look at, how are cells interacting and how is this spanning across every cell type in the animal. So we did this for all the perturbations that I showed you, and we can use the tools that are now really powerful also for all sorts of single cell, RNA, metabolomics, et cetera, analyses to do dimensionality reduction now where each dot is an embryo. And the size corresponds to the number of differentially abundant cell types that we detected. And this gives a really overview picture of, okay, some embryos have big composition changes because they lost a posterior end, for instance, or have small population changes in neurons, and they're much more like controls. If we want to look at the individual cell types and abundance changes, we can visualize this as a heat map where rows now are perturbations and columns are cell types. And when we do this for all of the data at a single time point, we can see really clearly that there are, despite having many different perturbations, there are commonalities among them. And in particular, those correspond to the tissue that perturbation affected. So here, we can, if we hierarchically cluster these perturbations based on abundances, we can see that the mesodermal groups group together, the CNS targets group together. And from here, we can really go and compare how some of these perturbations are phenocopying one another. So this was published, as I mentioned, all of the details and a lot of additional information is in that publication. And now I'm going to switch gears to talk about an unpublished collaboration that I've been working on in Heidelberg. And this is a totally different topic, which is trying to unravel sex-specific liver cancer susceptibility. You'll see the commonality in the methods. And this is a collaboration with a bunch of people in Heidelberg, particularly James Cleland and Jo-Ben Reed and Duncan Odom's lab, Aurelian Dugard at EMBL EBI, Bernie Drutleff at the EMBL Metabolomics core facility, and Edith Hurd's lab at EMBL Heidelberg. And the question really comes down to trying to understand why males in particular are more susceptible to liver cancer than females, given all external factors the same. So then the question that we asked really was, how do intrinsic factors contribute to liver cancer sexual dimorphism? And this is not using zebrafish now, it's using mice. And in particular, a really powerful mouse model called the 4 core genotypes. And in the briefest form, the male of this model has the SRY gene moved from the Y chromosome to an autosome, which means that if you cross him to a wild type female, what you get is a combination of 4 genotypes, 2 of which have ovaries, 2 of which have testes, and within each gonadal group, you have an XX and an XY chromosome. The reason this is really powerful is because when we're studying differences in sex biology, it's really difficult to disentangle the contributions of sex chromosome and gene expression from sex chromosomes versus sex hormones. And this model is really powerful for doing that. So the overall approach was to apply, of course, the individually resolved now per liver phenotyping, both with single-cell RNA sequencing, skyRNA sequencing, in this case, sample multiplexing at RT, because livers have tons of nuclei, and that was really powerful for the quote-unquote icing on the cake, the cupcake, which is that we collaborated with the EMBL MCF to do lipidomics on each one of these 48 livers. So we have the individually resolved lipidomics, not single cell, but individual mouse, with the single cell RNA sequencing from each individual mouse. So again, we make a big atlas with the 48 individuals. This is really showing, the main cell types in the liver. We have about 500,000 nuclei from these 48 individuals, and the 5 genotypes that I showed you. And again, we wanted to look at differences in sex chromosomes versus sex hormones. It turns out that sex chromosome differences are much more subtle than the sex hormone differences, maybe not so expected. And so if we look at how these cell compositions are changing in response to the gonadal status, so whether the mouse has ovaries or testes, we can see that there are a few cell types in particular that are particularly differentially abundant. So this is a HuBMAP now colored by the abundance of the cell type relative of each cell type in gonadal females versus gonadal males. And we can see that in gonadal females that have ovaries, certain immune cell populations are increased, as well as endothelial populations. And there's a specific population of hepatocytes that's especially responsive to whether the mouse has testes or ovaries. Now getting to the lipid information, again, for the gonadal hormones, there are a bunch of differences between the lipid species that are detected in each of these groups, in whether the mouse has ovaries or testes, and bringing this all together right at the end. So there's a lot in this plot, but I think it really nicely illustrates how a bunch of data can kind of consolidate into some really testable hypotheses. So this is a correlation analysis that compared differential lipid abundances with gene expression. And by looking at the presence of enzymes and increase and decrease of these in males versus females, as well as the transcription factors that are present in the hepatocytes. Basically, this analysis points to 4 major CYP genes that the lipid abundances and gene expression data converge on. And so this allows us to follow up and really test how these might be contributing to sex differences in the livers. All right, so in summary, I've shown you a workflow for high resolution phenotyping of embryos and tissues. It's quite flexible, as you can see with these 2 topics. We've made a bunch of atlases, and we're continuing to make more, looking at how perturbations impact gene expression. And this is just a little taste of how adding additional modalities with individual resolution can allow us to compare relationships spanning from molecules to tissues to organisms. We have a new pipeline that's available on GitHub for analyzing skyRNA sequencing data. It has friendly dependency installs, and it incorporates the hash barcode analysis. So feel free to try that if you want to give this protocol a go. And I'd like to acknowledge my former postdoc lab in Seattle and a bunch of people I worked with there, and my new community in Heidelberg, which is the EMBL, a decade said, and UD Heidelberg, and all of the people that I tried to mention throughout the talk. My lab is really working on sulfate specification in the zebrafish neural crest, building new tools, profiling lots of single cells, and we are looking for postdocs. So please feel free to reach out and check us out online if any of this interested you. And I'm very happy to take questions. Thanks for your attention.
Okay, thank you. Very nice talk. And we are going to move to the next speaker. The next speaker is Carsten Hof. Carsten is from the Center for Mass Spectrometry and Optical Spectroscopy in Mannheim University in Germany. So today with his PhD student, James, they are going to present their recent work. And Carsten, the stage is yours. Is Carsten here? Okay.
All right. Welcome, everybody. It's late at night here in Germany. Good to see you all. Yes. So I'm just doing the first part here, and James will actually present the main story. So we are located in Mannheim, Germany, close to Frankfurt, operating a large mass spec lab with various applications. And our team has been busy over the last 15 years in multi-imaging, from designing new organic chemistry, multi-matrices, other derivatization agents, helping companies to develop devices, develop workflows, and most recently also some software tools. And the one application that we want to talk about today relates to Alzheimer's disease. And many of you will know that there are two classical hallmarks of Alzheimer's disease, namely a deposition of various A-beta short peptides of various lengths, such as 1 to 42, and a hyperphosphorylation of the tau protein in axons. And besides those two classic hallmarks, most recently, microglia activation in the brain, a specialized type of macrophage in the brain, has drawn a lot of attention. And that is our topic here. It has been implicated in AD. It's a sizable cell population in the brain, about 10% in some cases. Classically, three different states, M0 resting state, M1 and M2 have been distinguished. Most recently, there's a lot of evidence that there's more of a continuum actually to describe this. But in a classic approach, going from resting to M1 stage, which is a pro-inflammatory condition called M1 microglia, can be mimicked to some extent by lipopolysaccharide stimulation, LPS. And the other state of microglia, M2, is anti-inflammatory and suppressing immune responses. And in our study that also led to a lot of new technology for single-cell metabolomics using multi-imaging, James will describe, but we focus on LPS-induced M1 stage microglia, which has been shown to be damaging to various cell types in CNS, and ultimately also been associated with Alzheimer's disease. So without further ado, I hand over to James.
Oh, I hope you can see my screen now. Just wait.
Not yet. Now, yes.
Okay. Yeah, perfect. So exactly as Carsten mentioned, we wanted to study this single cell, this microglial activation on the single cell level due to LPS induced M1 states. So to do this, we had two basic approaches. Taking an unguided approach, as was discussed earlier on, which would be, this is the entire slide we see here. These are the cells. And the unguided approach would be targeting the entire region here, which in this example would be around 300 cells per hour. This obviously depends on cell density, the cell type used, and the instrument used. So this varies between labs, I guess. So, and the other approach we could use was a guided approach, basically using some type of guidance modality to target every single cell beforehand to acquire these spectra. As we can see, we get more coverage over the entire well we have here. And the left and the right actually have the same number of pixels. So in this example, this is a rough increase of 6-fold, which basically using a guided approach, we don't measure as many background pixels, which we are not interested in this case, and the acquisition time is longer. So basically, for higher throughput studies, we decided that for our use case, we needed some kind of guidance. So the current versions of guidance usually rely on the microscopy step to build a guidance. So it's an optical guidance. This can be based on different modalities such as fluorescent imaging or bright field microscopy. But in general, the cells are taken from cell culture, then an image is acquired, then matrix is applied, and then based on this image, a guidance is created to measure this. This, in general, was developed for lipidomics, but we, in our use case, are actually focused on small molecular metabolites, which we observed to have degradation during this step. So basically, we looked at the TCA cycle, since in microglial activation, it's known that the TCA cycle is interrupted at these parts to produce fatty acids and further activation in general, but this is known. I'll come to that later on. And when studying the TCA cycle, we basically need to avoid metabolite degradation. Since we are observing these small low molecular metabolites, we observed reduced sensitivity, and especially for reproducibility purposes, we need this microscopy step to be removed, since we all know that when performing microscopy, it doesn't take the same amount of time every time we do this. And especially, we had to avoid different workflows that used PFA fixation to focus on these TCA cycle metabolites. So the basic questions that we had were, can we have a guidance without this microscopy step here? So leaving out this step. Well, now we're just basically back to an unguided approach, right? That was a problem. So I came up with this method, which we coined PrismMS. So basically, we took the cells from cell culture and imaged the entire slide at a low spatial resolution of, for example, 200 microns. And then using target signals, such as a fatty acid, for example, we targeted, we built a mask based on this signal to detect where cells are present and where cells are absent on the slide. Using this, we then built a new guidance, which was then used to image this slide with a higher spatial resolution in this case. So we called this pre-scanning imaging of small molecules, and this was all performed using R. So you can see this in action here. So this is actually after the pre-scan step. This is the deep scan imaging now. So the pre-scan step, an example is here. I believe that's 100 micron resolution. And this would be the resulting deep scan we get out of this. I think we did 5 micron resolution for this example here. So the question we had, we obviously need to check if we actually see metabolite preservation. So I did a comparison using 6 different slides, 3 of them using PrismMS and 3 of them using the optical guidance approach. The peaks here are all annotated features via metaspace. And we see several of them being significant for the PrismMS workflow, while others being significant for the optical guide workflow. And the black ones show no significance over 6 samples. So here, one of the significant signals is this fatty acid, which can actually be nicely tracked down to show the oxidation to 9 or 13 HODE. It actually shows more degradation steps, but this is just one example of that. And we can observe that this is actually here higher in the optical guidance compared to PrismMS. Another example would be these peaks. If we look at these features here, they're not significant over our total samples, but they're obviously higher in the optical guided workflow. And this is because they're just present in one of those measurements. and therefore not over a significance threshold. But this is just shows us that the reproducibility is higher for these small molecular metabolites using our PrismMS workflow. So basically what we did from this was I built a Shiny app so others can use this without knowing how to code. It should be pretty easy. I'm currently working on a tutorial video so everybody can use it without having to code. It's released on our GitHub. And yeah, you can access it there. So why did we actually do this? So going back to our microglial cells, we had an activation marker, namely itaconate, and a resting cell marker, which is taurine. This is a resulting cell data after the deep scan. So we've got vehicle control, untreated cells here. And in the other wells, we've got increasing concentrations of LPS to induce this microglial activation. We actually performed subpopulation evaluation using Monte Carlo consensus clustering, which I can't go in here for time reasons. But we found that we get subpopulations that are specific for the resting state and for the active state. And with these refined subpopulations, we could do a comparison. So we see that over the concentration, we get a slight shift from the resting cells, which are taurine positive, itaconate negative, and the pink population, which is itaconate positive and taurine negative. So what did we want to do with this? So basically, we wanted to look at this green population and compare it with this pink population to build this activation profile to see which metabolites actually differ here. So using PrismMS, we found 13 metabolites being specific for the resting state, while we found 49 metabolites being present in the active state. We did exactly the same workflow with this pipeline using the optical workflow and found a way lower number of significant features. And we attribute this as well going back to the metabolite degradation. So the whole point of this was we wanted to compare these activation profiles with tissues. So the question was, does this translate to tissues? For this, we had mouse hippocampal sections that were incubated for 72 hours, which is called slice cultures. So they are incubated as vehicle control again, untreated, and LPS treated. And we had a third population which was clotronate treated, which is actually depletes microglial cells, but I'll not go into this. We needed that for validation purposes. So we imaged these tissues afterwards, did basically the same procedure, just for tissue, not for cell culture, and built a profile based on the resting state and the active state. Keep in mind that obviously we've got complex cell mixture here, while the cell culture is pure microglial cells. We found itaconate again, which was actually really hard to see using the optical guided workflow. And yeah, we see that it's higher in the LPS treated tissue as expected. From this on, we performed a single cell pipeline and analysis pipeline on 2 different iPSCs, human-induced pluripotent stem cells, as well as on CIMA9 cells and a control cell line. And we then compared the metabolite profiles with the slice cultures. One result we got was this. As I mentioned before, this is the TCA cycle that's in active microglia, where we have this interruption, where we get the fatty acid production over itaconate. Every image here on the left side is an untreated tissue section, while the right tissue section is LPS treated. We got all of this information from combining cell culture and slice culture data, and this was a follow-up measurement using 5-micron imaging. And basically what we can see here, what we found, and what we are currently looking into, is that we've got N-acetyl aspartate, which is which we shortened here for NAA. We get a depletion in the active LPS-treated tissue section. And we suppose that this is a shuttling effect going where NAA is in neurons and it's there in times of stress, where it gets replaced and replaces the glutamine, which is subsequently used by microglial cells for energy, for keeping the TCA cycle running. And just a basic summary and outlook. So we've got the first single-cell MSI for small metabolites that preserve them. We found links between tissue cultures and cell cultures. We actually found that this was not a primary objective, but we're no longer limited to transparent slides when doing MSI or guided single-cell MSI, since on gold slides, cells adhere better, for example. We're currently doing cell co-cultures to validate these findings. We're inspecting the data and the pathways. Then we're currently working on identifying cell types in the tissue and via MALDI MSI. And an interesting idea would be a scoring system for evaluating how close a cell line matches the tissue model, the animal model. Since we use different cell lines, we could do a comparison. And yeah, basically we can use this methodology as a test system for other compounds, since microglial activation is not only triggered by LPS. And I want to thank everyone from our team at CMOS and our external collaborators. Thank you.
Thank you very much for the talk. So we have a question, but before I wanted to ask you, I saw that during your presentation you showed that you have some enrichment in fatty acids sometime in the brain in different regions. And so I wonder, do you think there is a specific role of this enrichment of fatty acids specifically?
I believe that's because the activated microglial have been shown to in the activated state they produce fatty acids for I'm not sure for what exactly. I have to look into that, but just during defense or yeah.
Okay. Thank you very much.
Cool. We also have a second question from the chat. Question is, this technique can acquire cells metabolome with high efficiency, while at the same time keep the area without cells. But do you think it is reasonable to measure some background area and take this as background signal to know what compounds are cell specific?
Sorry, could you repeat? I don't.
Yeah. So the question goes, this technique can acquire cells metabolomes with high efficiency, while at the same time keep the area without cells. But do you think is it reasonable to measuring some background area and take this as background signal in order to know what compounds are cell specific?
It could be a good idea, yes, sure, but yeah.
Okay, for sure. We can get in touch. Anyone is welcome to join the Discord that we just shared through the chat and also by email to people who wanted to join that. So in case you can continue this and more conversation by Discord. Thanks again. And I think Joel and Giovanni, the floor is yours again.
So thank you so much. Thank you to everyone. First of all, to the speakers for finding the time over different time zone to join this event. Second, obviously, to our students, we took so much time to organize this. Thank you very much. I would also, of course, want to thank you all listening and staying with us across this whole event. And in particular, I would like to welcome feedback. This is just the first edition. We hope to come back in 1 year. We want your feedback, and this can happen also through the Discord that Luca just advertised, also by email. Please reach out. And then.
Yeah, and so thank you for staying here late, or depending on where you are. One thing that I want to add is that this is an open kind of initiative, and we really think that repeating this next year will be great. But I actually invite all the people that might be interested to jump on board and help people here or collaborate with them organizing this maybe next December. And with this, we just tell you goodbye, and let's keep in touch on Discord and through other means, and let's keep discussing our science.
Yeah, and Happy New Year.
Happy New Year, everyone.